Good morning, Sachin. Sachin, you are able to hear me? Sachin?
I think you should unmute yourself, I believe.
Sambrita, Sachin, can you hear me?
Sir, I'm able to hear you. Sachin also has unmuted himself. I did check with him, but I'm not sure.
Because I don't see him speaking also.
I think he's preparing something.
No, he's not even speaking. Like he's. Sachin? Sambrita, you able to pick it?
Yes, I can hear you, sir.
Can you hear me now?
Yes, Sachin, we can hear you.
Yes, Sachin.
Yeah.
Okay, fantastic.
So I don't know.
Sorry, some issue.
Yeah, and sorry about the delay. It's we are running a little late, but then I think we should start as soon as possible.
No problem.
So, I yeah.
Research is seeing what everybody else has seen and thinking what nobody else has thought. A warm good morning to one and all present here. It gives me immense pleasure to welcome you all for the program on Introduction to Clinical Research and Careers organized by Indian Society for Clinical Research and Kelly College of Pharmacy, Belagavi. Now, I request Dr. M.S. Kanachari sir to give or deliver welcome address and introduction of our chief guest of honor.
Good morning, everyone. At the outset, I welcome all the participants who are logged in online and colleagues who have joined physically in this hall for witnessing the inauguration and being the participating in the workshop. It was an immense pleasure to host this particular program activities, and it is a dream come true for me, particularly getting associated with ISCR with a lot of efforts from Dr. Ullas and his team. And we all know that since very long we were waiting for this because of various reasons. It went on postponing, and finally we are here today to celebrate or to be the part or to witness this unique program. All the industry stalwarts coming forward, coming to a university and addressing the young graduates or young aspiring students. It is a unique coming physically here, witness and gracing this occasion. Second, I would like to thank our KL Academy of Higher Education and Research administrators, including Vice Chancellor, Registrar, and my beloved principal of my pharmacy college, Dr. Sunil Jalalpure. He was so much cooperative, or he was so much in forefront. He was the first person to saying that go ahead to conduct this program. Of course, my colleagues were there always around the clock, and even yesterday night we were preparing the program and we put together the quiz and all those things. Apart from all these activities, as I said, it is a unique opportunities for industries and institutions. We at KLE University, always we are working towards a clinical research, clinical trial since very long, and we have grown step by step with mentorship of Ullas and other members. I can see a couple of panelists whom we all know. We know each other in one or other capacities. At KLE University, we do run a MSc Clinical Research as a capacity building. It is a 4-semester program, full-time program, which is a unique program that we conduct in the benefit of the professionals who want to pursue their career into clinical trials and clinical research. It is an UGC approved program. The program was developed in collaboration with, again, Dr. Ullas and his team and all those things. That's how our syllabus was validated. Apart from Samprita and Sachin and other people, I can see Chandana Paul. She was a quality assessor for one of our activities, which I know personally. I may be knowing other people. So I wholeheartedly welcome all the panelists who have consented to be the part of this wonderful sessions morning. I know that they all are extremely busy, but in spite of all those things, they joined here to impart their experience, knowledge, and skills. Thank you one and all for this inaugural function of this morning, ISCR and KLE College of Pharmacy joint venture for this virtual programs. Thank you one and all.
Thank you so much, sir. Now I request Principal Sir to give bouquet to Ullas Sarvami, sir. I request MS Kanachari sir to deliver bouquet to Principal sir. I request Sidhu sir to give bouquet to MS Kanachari sir. Now I request Satish Karoli sir to give bouquet to MS Kanachari sir. I request all the dignitaries to come on the dais for the lighting up of lamp. Thank you so much. Now I request our guest of honor, Dr. Ullas Sarvami sir, to introduce about today's workshop, that is Introduction to Clinical Research and Careers.
Hello. Hope you can hear me, Sachin, Samprita, and everybody who is online.
Yes.
Good morning, everyone. And thank you for being present in this session. It's been long. We're trying to organize this, and finally we are conducting this. Hope it will be useful for each and everyone who has been here, and there will be something that they can take home at the end of the session. Basically, what we are trying to achieve by organizing this is, when Sachin and I started discussing this, I think at almost 1 year ago. And since then, we've been brainstorming continuously how best it would be to chalk out the program sessions, who to approach in terms of guest lectures and speaking slots and stuff. It's been quite long. We've been trying. So I'm so glad to see this happening eventually. So the objective is what we are trying to achieve is to bring academia and industry together. Obviously, that's the first step. And secondly, to with the help of the experience, skill sets, and knowledge of all the leaders in the industry, disseminate that information among academia and students so that it helps them in terms of expanding their knowledge about clinical research industry, how it is happening in India, how it is happening worldwide. And also in case they would like to take up careers in clinical research organization, clinical research industry, then what would be the avenues, how to prepare, and what should be the starting point. So just a little bit of an insight into this industry, and we would be happy to give this glimpse, and then it would be up to them to take leaves out of this and expand on them. So I'll keep it short with that. And I really thank ISCR for supporting in conducting this workshop, and Sachin personally, and Samprita, I know she was emailing me 10 o'clock, 11 o'clock last night to get all the information and coordinate it all. So thank you very much, Sachin and Samprita. Thank you.
Thank you so much, sir. Now I request Dr. Sunil Jalalpure, sir, to deliver presidential remark.
Very good morning, one and all, and I welcome wholeheartedly to the KLE family. First of all, I would like to thank Department of Clinical Pharmacy thought to organize this unique program. Though it was planned initially offline, but because of this pandemic, we had to make it online. And one more team headed by Dr. Ullas Arbavi and Indian Society of Clinical Research. So they have joined hands to organize this program. So I thank both the Department of Clinical Pharmacy as well as Indian Society of Clinical Research to organize this very unique and need of the hour program. So once again, I welcome all the delegates who all are attending this program online and offline to the KLE Society. As you all know, KLE Society is a big banyan tree established way back in the year 1916. So we celebrated its centenary year in the year 2016. So reason to tell these all things is many of the delegates who all are attending this program may not be aware of KLE Society, because across India, delegates are attending this program. So we run 282 institutes under the ambit of KLE Society. And KLE University is the sponsored university of KLE Society. KLE University has two universities. One is Technical University, which is situated at Hubballi, and this is Health University situated at Belgaum. So as I was mentioning about KLE Society, it takes long time to tell about the story, then history of KLE Society, then activities of KLE Society. Directly we'll come back to our Health University, which is established in the year 2006, and we are now part of KLE University, affiliated to this in the year 2009. Before this, we were in the Rajiv Gandhi University, this constituent unit as a KLE College of Pharmacy, Belgaum. And before that, when we were studying, myself and Dr. Ullas, we were in Karnataka University, Dharwad. So at present, KLE College of Pharmacy is affiliated to KLE University, and it has other constituent units like medical, dental, nursing, physiotherapy, Ayurveda, homeopathy. You name any medical stream which is affiliated to KLE University. Lots of clinical research related things are going on, and our Department of Clinical Pharmacy, headed by Dr. M.S. Ganachari sir, is flooded with activities. And I hope the Department of Clinical Pharmacy, those who are working there, they are also busy enough in conducting these activities. And definitely the students who all are studying in KLE College of Pharmacy are very lucky and taking a lot of benefits and advantages of these activities. Now coming back to KLE College of Pharmacy, Belgaum, which is also very old and renowned institute imparting quality education in the field of pharmaceutical sciences, established in the year 1968. So this is very oldest pharmacy college in this part of India. We celebrated its golden jubilee year in the year 2018. So we started with a diploma in pharmacy. Then in the year 1975, we started B.Pharma course. And now we offer M.Pharma in 6 various departments. And in the year 2000, we started PhD. In the year 2014, we started PharmD course. So considering these all facilities, infrastructure available here, so even accrediting councils also, they have graded us A+ by NAC. And recently we got NBA 6 years accreditation for B.Pharma program. So this is about, KLE Society, KLE University, College of Pharmacy. So I welcome once again all the delegates and especially the officials of Indian Society of Clinical Research, Dr. Ullas, Dr. Sachin, and Samprita, and other officials of ISCR for this particular unique program. And I also welcome to KLE College of Pharmacy, Belgaum. And once again, welcoming all the delegates who all are attending this program online for this program. And I thank once again the Department of Clinical Pharmacy and all the faculty members and head of the department, and thinking to have this program at this particular juncture at KLE College of Pharmacy, though it was planned offline. And Dr. Ullas is in touch with us since almost 6 to 7 months to conduct this program. And I think last month we planned this program offline. We booked 300 hall also. So unfortunately, because of rise in the COVID cases, we had to have this program online. So with this, once again, congratulating all the team members who all are associated with this program, Department of Clinical Pharmacy and ISCR for having thought of conducting this program on this occasion. And I thank giving me this opportunity to speak as a presidential remarks in this inaugural function. Thank you one and all.
Over to you, Samprita and Sachin, to take over and start the program.
Thank you, Ullas. A very good morning to one and all. I'm Samprita Hebley, Associate Director, Clinical Data Management, LabCorp Drug Development, and I'm going to be your host for today. So it's an honor for me to be hosting the Indian Society for Clinical Research event, and I'm extremely happy to share that we've had an unprecedented response for today's event, with almost 500 plus registrations, and I look forward to interacting with all of you and making this event a grand success. The ISCR event is organized by Ullas Arabhavi, who is the head of Clinical Data Management, George Clinical. So Ullas has around 16 plus years of experience across pharma, academic, and CRO industries in India and Australia. Experienced in end-to-end data management activities, he is involved in process, people, and project management, and in providing leadership to the DM teams. So currently, he's heading the global DM team in George Clinical, and his core interests are building teams, setting up collaborations, partnerships, strategic planning, and growth. Coming over to his education background, he is an M.Pharm pharmacology and holds a PG diploma in clinical pharmacy and also a certified MEDRA coder. He is an esteemed member of SEDM, life member and resource person in ISCR annual conference from 2018, and also the board member for studies for PG of Pharma, PharmD, and clinical research in KLE University, Belgavi, Karnataka. I welcome you, Ullas. And then we have Sachin Tonapi, who is the convener of this event. And Sachin is the Executive Director, Global Head, CDM Data Operations at LabCorp. He has a total 18 years of industry experience across pharma, BPO, and CRO in managing large global teams, setting up and learning the FSP partnerships, selling to clients. Responsibilities are spanned across several levels of management as well as long-term strategic initiatives. So prior to joining LabCorp, he headed multiple CDM FSP partnerships at a large CRO. Coming over to his education background, he holds an MBA degree from TAPMI, Manipal, a bachelor's degree in mathematics, Karnataka University, Dharwad, and he's a certified clinical data manager from the Society for Clinical Data Management, and also a certified Six Sigma Green Belt. Towards his contribution for ISCR, he has chaired the CDM BSMW Council since 2021, and he's been the member of ISCR since 2015. So I welcome both of you, and I look forward to collaborating with all of you on this event. I also welcome Dr. M.S. Ganachari, the Deputy Registrar, KLE University, Dr. Sunil Jalalpure, the Principal of KLE College of Pharmacy, and I extend a warm welcome to all the dignitaries present here. I also welcome our esteemed speakers from varied departments across our clinical industry with whom you will be interacting today. Welcome, speakers. And I also welcome the IT team from KLE University who are there with us today to support the proceedings throughout the day. And not the least, of course, our Gen X millennials. Thanks for the amazing response shown for this event, and I welcome each one of you and wish that you'll go back with a clear and positive mind about your future. So this symposium will focus on sensitizing students from life science, medical, and paramedical courses on the basics of drug development, what clinical research industry is about, the good clinical practices, and introduce you to all the career opportunities in clinical research. So without wasting more time, I now request Sachin Tonapi to give us a quick introduction on ISCR for you all to understand better of the objectives of the forum. Over to you, Sachin.
Thank you, Samprita. Good morning, everyone. It's my pleasure to be part of this wonderful session, and I thank all the dignitaries present here and KLE institution for providing this opportunity. It's really an honor for ISCR to partner with KLE and then host this event. I'll spend next few minutes and take you through what ISCR does so that it gives you a better view in terms of what kind of activities we do, and then it'll help us, understand where we are coming from. So let me share my screen. Can somebody help me share the screen? It's disabled right now. Is it possible to allow me to share the screen?
Yeah, it'll be done within a couple of minutes.
Okay, sure.
It is done.
Yeah, it is done. I can share it now. Great, thank you. Can you all see my screen?
Yes, please.
Yes.
Okay, great. So ISCR was set up in 2005. So all of the professionals, in the industry felt that there has to be some voice for clinical research. So they came together and created this nonprofit organization. Right now, we have 39 accredited member organizations. So these are all your multinationals, pharma, CRO, academic institutions, hospitals, all of them together run this organization. So it's all voluntary. So I, Samprita, all of us are part of this, including Ullas. We volunteer to, provide our time and expertise, to various events. We have more than 1,100 life members. And these are people from various walks of life related to clinical research. And we have 7 councils, and I'll take you through those council names, and 4 regional chapters, which are organized in such a way that, we can cater to different parts of India, as well as, different functions. So the vision of ISCR is to promote a compelling scientific disposition and widespread awareness of clinical research for advancement of healthcare. And the mission is to inspire and align different stakeholders to shape and foster an environment facilitating ethical and responsible clinical research for better patient outcomes. So we work with government, we work with industry, we work with academic institutions, we work with patients to make sure that we create an environment where we are helping each other and making sure that the clinical research is advanced in India. Our president is Dr. Shanice Davis. Our secretary, general secretary is Dr. Seema Pai, and treasurer is Anirban Roy Chaudhary for the year 2021-2023. And these are our executive committee members. All of them actually work for many multinationals, other CROs, and then help us, run this organization. If you look at the councils, these are the councils. So we have Clinical Data Management, Biostatistics, and Medical Writing Council, which we represent. So I'm the chair for this council for the last 1 year. Then we have the Ethics Council, the Investigator Council, the Pharmacovigilance, the Regulatory Council, the Real World Evidence Council, the Training Council, and then we have a Media Task Force, which works with the media. Now, each of the organizations which I talked about, the 39 organizations, will send their representatives to each of these councils. So, for example, LabCorp is a member, and then I've been asked to be part of the Clinical Data Management, Biostatistics, and Medical Writing Council. So likewise, we have different people coming in, and then we participate to move the agenda of the ISCR forward. These are the 4 chapters. So we have North, South, West, and East. Each of these chapters work for that region. So we are part of the South chapter, where we work with institutions and people within the south of India to make sure that we collaborate and really have some activities around clinical research here. These are some of the key activities. I thought this will help us understand what ISCR does. So one is shaping the clinical research landscape in India. So work with government, with institutions, and make sure that we create the right landscape. Advocacy initiatives. So we work with government, policymakers, regulators on an ongoing basis. In fact, every quarter we have a meeting with our regulators to take feedback from the industry and provide them some inputs for the framework or the guidelines which they usually help us create to run the clinical research. Then we have media and public awareness. This is very strong, and I think this is one of the important things we do. We dispel myths about clinical research in the media, and we actively participate in providing the right information to the media so that people have informed decisions. Capacity building, educational programs and workshops with various institutions, and this particular session is part of the educational program which ISCR takes from time to time, so that we can create an environment where people are aware of the clinical research opportunities which are available for people from our geographies. Academic consortium for clinical research in India. This is a unique thing which we have started for last couple of years, where we are bringing in academicians to be part of the clinical research. Then we have the Clinical Research Coordinator Certification Program. This is one of the unique programs which has been put in place by ISCR. It is a really successful program. Now it's been replicated in South Africa. And some of the institutions who came here saw it, and they have asked us to actually replicate this in their country. Then we have an annual conference, which usually happens during February, March of every year. And this year also it is happening. So please look up in the website, and there is a huge discount for the students. So in case if some of the students are interested, and even for academicians, the teachers and the professors, if they are interested, they can certainly be part of the annual conference. We also have an ISCR Award, which is constituted by ISCR to help really recognize some of the best researchers in India. So that's what is a core activities I can call for ISCR. There is a journal which we publish every quarterly. It's called Perspectives in Clinical Research, and that's been well received as well. So it's available online for free. So if you are interested, you can certainly go to picronline.org and have a look at that. This is actually contributed by all the clinical research professionals in India. They contribute on a regular basis, so you can have a look at this. What we have done during the pandemic, I think this is an important thing, because when pandemic hit us, we were all face to face in terms of our workshop and events. We had to go back to be online. But I think that is a boon in a way, where we know now to reach out to actually many people. If you see, these are some of the activities which we conducted. Around 11 workshops, we reached out to around 2,500 participants overall. SAE Sugam portal, that's an important initiative from Government of India. I think we contributed significantly to that. Benefit risk assessment tool. We also had a 7-part web series on clinical research during and after COVID times. This was very well received, with an average attendance of close to 200 people per session. So I think that was the kind of activities we did during the pandemic. So who can join? Any working professional associated with clinical research with 1 to 2 years experience can join, including people in the academic institutions. And there is a life membership also available. If you're interested, you can certainly go to iscr.org to have a look. An AMO is a member organization which has certain benefits. So they can actually nominate few people to be part of the council. They can be part of the AMO community and then get all the information. So, if somebody is interested to be an AMO, an organization, they can be part of it. Then there are individual membership and also, the various benefits which I just talked about, are available for all the members. With that, I would like to thank KLE Pharmacy College for us to really give this opportunity. And then I would also like to thank all of the people who are contributing today. These are all people coming from the industry. They are bringing in their experience. So I urge all of you to take this opportunity to ask questions, make sure that you make notes, and also give us feedback in terms of what else we can provide, because we definitely want to continue doing this in the future. We have done this in the past, but I think your feedback really helps us. I know Samprita has put in a lot of effort. Ullas has put in a tremendous effort to organize this. So thank you, Samprita. Thank you, Ullas. There is a quiz which is going to be organized. So I would urge all of you to take that quiz and then make sure that you participate very actively, ask questions, and bring in the best from our presenters. Because the presenters will come in with certain thoughts, but if you ask a lot of questions, make it interactive, it helps us to also contribute at a higher level. So with that, I thank everyone who are part of this. And back to you, Samprita, to take it forward.
Thank you, Sachin. Thank you for giving us a good insight about the objectives of ISCR and what are the various programs ISCR has conducted over the past year, and also giving us a heads up on the upcoming conference, the annual conference. So I encourage all the students to participate in this upcoming event, which will be very informative and educative. So as Sachin mentioned, before we really get started with the technical sessions, I feel it's a good way to get warmed up. So warm up yourself, guys, and gear up your mind to absorb the technical aspects of clinical research. And I feel the best way to do this is having a quiz session. So this will really help us also to understand the pulse of the crowd. And I request the IT team to run the quiz now.
I think all the participants can be the part of this quiz.
Yes, sir. So the quiz will be available for
Yeah,
all participants.
Just now we started. Can anybody text so that it is working in the chat box?
Okay, I can see a yes.
Okay, good.
Just to mention, while people are filling out this pre-session quiz, at the end of the day, we will have the post-session quiz as well.
We also have other speakers who have joined in. So if you can allow them to be part of the panel, that will be great.
Yeah, we will do it.
Okay, thank you. I already see somebody has posted a question, which is fantastic. So please do use the Q&A panel to ask your questions. I think it will really help us make sure that we answer all of them. So thank you.
We'll just give a few seconds more before we close the poll and see the final results.
Next speaker is Chandana Paul. Chandana Paul. 10 to 10:45 slot. Chandana Paul, you should give her the, you should make her come on the. She has not joined yet. Okay.
Few minutes more. So you can continue answering the quiz questions. Few minutes more.
Ullas, can you just check? Chandana confirmed that she has joined.
Sachin, we just checked. So Chandana hasn't joined yet. So we have our IT ready to give her, get her on the panel.
So Chandana has messaged in the group. If you see, I think she's using a different system. And so if you can look at the chat, she's saying, I'm Chandana here. So you can allow her maybe. That person.
Done. She is now on the panel.
Okay, great. Thank you so much. Chandana, can you confirm if you can hear us? You're on mute, I think.
Yes, thank you.
Thank you.
Okay, great. Thank you.
Are we good to end the quiz session and see the final results? I request the IT team to close the quiz session and let us know the final outcome.
Shall we close the quiz?
Yes.
Okay.
So the quiz can be closed, and it would be good if we can see the final results.
Okay. Yeah. Takes time. They're closed. Yeah. Huh? Samprita, can you see the results? No.
No, sir. Not able to view the results.
We'll just. Never. We are sharing the results, but I don't know.
Okay, I'm getting to hear from the chat box that they can see the results.
Yeah.
I'm not sure because I'm not able to view it.
Okay.
Is it possible to take a screenshot and maybe share the screen if possible?
Yes.
Yeah,
I think
that's a good idea.
Also, another reason could be, Sachin and Samprita, maybe the panelists won't be able to see it. Only the
Yeah, could be.
I'm guessing
Yeah.
so, sir. Yes, that
Yeah.
might be
Yes.
the reason.
It could be the
Yeah. Can you see the now screenshot?
We can
we can see a
the
website actually.
website, MSN website
MSN.
is visible.
Ramesh, can you share that?
or you paste it in the chat box.
Correct. Now?
No, sir. We still can see the website, and now it is back to the dais.
Okay, I think if someone can paste it in the chat box, that would be good, I think. Image. Image one. Because also we don't want to keep shifting screens. And if it gets stuck again, when the next session starts, then
I think others are seeing it since.
Yeah, I think only the
Yes.
panelists can't see it.
Yes, only the panelists are not able to view it.
Yeah.
So probably if any one of you can read out the poll results also is fine. If
yeah, that would be
people
good.
can-
So can you increase the font? I can read it out. Now it is there? Okay.
Can you see now?
No, sir.
No, sorry. It's not visible from here. Here, the slight problem with the connectivity.
So probably if anyone can just read out the poll results also should be fine.
Okay, Samprita, I'm trying to read this for you.
Thank you, Las.
Okay. DASH is a set of research ethics principles for human experimentation created at the end of the Second World War. First one, Nuremberg Code, 57%, 39 out of 69 responses. ICH guidelines, 26% people have responded as ICH guideline, 18 responses out of 69. Good clinical practice, 17% of the people have chosen this option, 12 responses out of 69. So total 69 responses for the first question. Nuremberg Code, 57% of the responses. ICH guideline, 26%. Good clinical practice, 17%. And the correct answer is Nuremberg Code, of course. So a lot of people, the majority of the responders are correct. Second question, guidelines for international ethical and scientific quality standard for designing, recording, and reporting trials that involve the participation of human subjects is called as. Single choice question. Total responses 66. 20% have chosen GMP. 18% have chosen IRB. 58% have chosen GCP. None of the above, 5%. Correct answer is GCP, and majority of the respondents have chosen the correct answer, 58%. Third question, signing of informed consent is not mandatory prior to any subject starting with clinical trials. Single choice question. Total responses 62. 24% have said true. 76% have said false. Correct answer obviously is false. So majority of the respondents are correct. The science and activities relating to the detecting, assessment, understanding, and prevention of adverse effects or any other drug related problems is called as? Single choice question. 62 responses in total. Pharmacovigilance, first option, 79% of the
I see that the video is hung. Am I audible? The panelists are unable to hear or see anything.
Yes, Samprita, you're audible.
Yes, Chandana. Seems like there's a glitch.
There was a momentary connection loss. So
Okay.
going back to the screen now. So we were on fifth question. Yeah.
Right.
Yes. So 16% have chosen control for drug and standards center organization. So majority are correct in choosing the right option. Sixth question, medical writing is an umbrella term for single choice question. Total response is 61. 26% have chosen scientific writing. 7% have chosen regulatory writing. 2% have chosen publication writing.
Okay.
Correct answer, all of the above. Number 7, measure of central tendency R. Total responses 63. 10% have chosen mean range deviation. 78% have chosen mean mode median. 8% have chosen range mode error. 5% have chosen median variance and range. Correct answer is mean mode median. So 78% of the respondents are right. CRO means clinical 63 responses again. Clinical research organization 54%. Contract research organization 16%. Both A and B 30%. Nobody has chosen none of the above. So both A and B is the correct. So 30% of the respondents are right. Number 9, DASH combined with big data hold the potential to solve many key clinical trial challenges. 61 responses. Blockchain 5%. Electronic health records 33%. Artificial intelligence 20%. A and C options 43%. Correct answer is artificial intelligence.
We are unable to hear again.
You can have the screen back on, please. 10th, last question.
Yes.
Okay, and the last one. All of the above, 63% have chosen it. Correct answer, of course, is results in hospitalization, is life-threatening, congenital anomaly, or death. So 22% of the respondents were right in choosing the correct option. So that is the pre-session quiz for you guys.
Thank you, Lars, for briefing us on the quiz questions and the correct answers. So thank you, team, for participating in this pre-session quiz. And I see that we are good to go in terms of the correct responses, what we received. But I see that there are a total of 137 participants at the moment, and the response percentage was around half of it, less than half of it. So I kindly request and encourage all the participants to actively participate, ask us questions, and we will also, as mentioned earlier, we'll also have the post-session quiz. So I request all of you to actively participate across all the events. So now that we're all set to get started with these sessions, we now have Chandana Pal, who is the Accreditation and Quality Systems in charge, Apollo Research and Innovations. So she's the team lead and the main contact of AAHRPP, that is the Association of Accreditation of Human Research Participants Program, Apollo Hospitals Enterprise Limited. So she's got 13 sites and 5 ECs, the Global Gold Seal in 2016 and re-accredited in 2019 under her leadership and training. She oversees the regulatory registrations, that is the CDS, SEO, and DHR, and the NABH EC accreditations for all Apollo Hospitals ECs. She's a member of few institutional ethics committee in Hyderabad. She's a certified NABH assessor for assessing the standards for clinical trials in India and an NABH faculty for accreditation and GCP training. She's also the course faculty for the clinical research courses run at ARI. She's been active life member of ISCR since 2011 and now part of the ISCR South chapter as well, and has been awarded the Hall of Fame Awards in 2019. A nursing graduate from PGI Chandigarh, MSc in Clinical Trials from LSHTM UK, and now a research scholar at IIT Hyderabad. On the professional front, she's having 20 plus years of experience in clinical research. As informed by Ganachari sir, she's also associated with Kerala University on various activities. Chandana, I welcome you to the session, and she's going to take us through the overview of the clinical drug development. Over to you, Chandana.
Thank you. Thank you for the nice introduction, and thank you all for calling me over for this session, which is very, very nice to be able to connect to so many participants, so many people who are looking at clinical research, and we all as clinical research professionals, we all feel happy if there are people who are ready to walk in our footsteps and take clinical research as a way forward in their career. It's so nice to be all of us, Amrita here, and all of us who will be attending the sessions with you and sharing our views about us, about the basic activities and the events and the specialities that clinical research has. So I'm sure we'll be very happy to share our view, and we'll be very happy to take comments from you and in the chat boxes, listen to you as what are your queries towards the various fields of clinical research, which we are going to introduce you through the day. And we hope that we'll have you together with us all through. So I'll just share the screen. It'll be what is clinical research today that I will be introducing you today. Give me a moment. Yes. So I'm sure you are able to view my screen, all of you. So let me take you forward to a basic question: what is clinical research? And I'm sorry that, of course, all of you are pharma graduates here, pharma postgraduates, and people taking up pharma from your undergraduate self, and you would be knowing so much about drug development and drug research and how it goes forward. But for the common population, correct me if I'm wrong, I don't think so, the pandemic has actually brought clinical research more to the front round, more into our discussion, which has become a every dining table discussion, a every TV room discussion. Whenever you put the TV on, you have nothing but things about the pandemic and the drugs which are being repurposed and the vaccines which have been started. In 2020, when we are struck with the pandemic, we did not have anything. We were actually scuttling to understand what could it be that can be used, and that is when the repurposing of many drugs were used. There were quite a few clinical trials which were designed to see which would be the best one to tackle the pandemic. And then it took us a little time, but the shortest time ever actually, to come across few vaccines which has definitely shown its effect. And here we are in 2022, when we are saying that with this wave, it has been much better. The mortality has been much lesser and the morbidity has been much lesser, because we have the the vaccines, and we have people who have been vaccinated and already going for the booster. So that is what clinical research is, and that is how it has progressed. And it was not that it started only when we had the pandemic, but it has been there for times immemorial. Not that any of the drug that we have today did not go through clinical research, but clinical research had its own, pathway to follow. It evolved as the research continued. Clinical research evolved. The many questions which you had in your just now done quiz, there were so many questions on which was the amendment, which was the Nuremberg Code, what was it about, what is the IRB, etc. So initially, when research was being done, it might have been, followed for the purpose of research, for the purpose of science. But as we go forward, clinical research took more of a sense in learning what is ethical research, what is scientific research, and how do we put research forward for everybody to know. Though not a part of this, but I can always tell you that we all know about the CTRI. Nobody can go ahead with research in today's day without registering your research in the Clinical Trials Registry of India. And why is that so? So that everybody knows what research is happening and everybody knows what is the outcome of research. We all are science students. We all know when we do research, it's not necessary that every research has a positive end. Then we'll not do a research at all. If we knew that drug A is better than drug B, why will we do a research at all? But why we do a research is only to have a null hypothesis which is proven or disproven at the end of the research, whether drug A is better than drug B, drug A is equal to drug B, or drug A is actually inferior to drug B. And believe me, as we go forward, you will see that out of the tens and thousands of molecules which we start working with, it is only one molecule which sees the day of the light of the day. And that is why clinical research is so very important. So if you look at the drug development process, again, as I said, you are all pharma students, you know it very well. But to take everybody along with us together, we know that we look at the drug discovery, we look at the early development, the preclinical, the investigational new drug, the phase 1 and the early phase 2. So when we do the phase 1 or the early phase 2, the number of sites which are chosen for the phase 1 are very, very limited, because the numbers we need in phase 1 are very few. And the target, that the primary outcome of our phase 1 is also very, very clear. It is, first of all, we have to see safety, because we are introducing the drug to the human beings for the first time. And we are looking at the dose limiting toxicity or the maximum tolerated dose. So we do not really need numbers in thousands. And we have a very cohort type of increase in the volume of the drug, which is given X, 2X, 3X, and we go forward and we look into the toxicities and collect the toxicities and see where do we have to stop. So that is a very, very limited exposure which is done in phase 1. And once the drug actually goes through the phase 1 and we think that, yes, the activity of the drug has actually mimicked what it has shown in the pre-clinical trials, that is the animal trials, then we'll take it forward to a phase 2. And here also in phase 2, we will be looking at how safe it is. Safety is something which will continue through the drug development process. And of course, keep at looking at the efficacy of the drug as well. In phase 2, we may do a phase 2A and a 2B. 2A, we may look at what can be the possible dosage, what can be the possible regimen. And once we are sure of what could those be, we can make a more focused trial on phase 2B, where we pick up the dose which we thought is the best in phase 2A. Going forward, when we know that phase 2 and phase 2B were good enough for us to take it forward, of course, when we look into medical writing, you will also see how the dossier is made. We'll have to present for every phase. At the end of the phase, we'll have to present the dossier to the regulatory authority and make the dossier speak for itself whether the next phase can be approved or not. The sponsor themselves would be knowing whether we should take it forward or not. And the regulatory authorities also will look into the whole dossier to ensure that, yes, it is safe, it is efficacious, and it can be tried for a phase 3 trial on human population, on a larger bit of population. But this population will be representative of the whole population that we have in our country. And of course, it should be a justifiable proposal, which is okay for the country that we are talking about doing the trials in. So as said, in phase 3, we'll have a huge population which will be exposed. And these are multicentric trials. These days, most of the trials are global trials, multicentric trials. Many hospitals in the country will be running the trial with their own principal investigators, whereas the central body The sponsor which is running the trial and the regulator of that country will keep an eye on the study as it goes forward. After we have done the phase 3, and if we think that yes, the drug is really good, we will go for the registration of the trial with the central authority again. We'll get the drug approval. But after approval again, there will be continuous post-marketing strategies. Continuously for the lifetime of the molecule, the drug will be actually kept an eye on how good it is performing, what is the efficacy level, and is there any safety issue to the drug. And if there is any safety issue, however years it might be when it has been approved, it can still be, the approval can be taken off and it can be taken off the shelves from the pharmacy. And if you all, as I said, you're all pharmacy students, maybe you will be able to, if you think of any drug whose registration was cancelled and approval was cancelled in the recent times, you will get quite a few in your mind, including ranitidine, which was taken off in the recent times. Sorry. So this is what I was talking about, the drug development process. If you see it on the screen, there are 10,000 compounds which starts the drug discovery. And once we actually move from the lab to the animals, it already gets reduced to a very, very little number, which you can see is 250 compounds, maybe crossing the preclinical level when we are okay to go ahead with these numbers for the clinical trials. They have shown some promise on the animal studies which are done. And once we know that the animal studies show us a safety and an efficacy data on which we can base our human studies, then on we start the clinical trials. So clinical trials is basically when we start with the first human dose and then go ahead all the way till the time we think that that particular molecule can be taken up for approval by the regulatory body and actually used by the patients. Again, when you see in the phase 1, we have, as I said, very, very few number of patients are exposed, and it's a cohort-based study. One cohort will get a particular dosage, and then they'll have to be looked at till they complete their 21 days, till the time the half cycle of the drug is over, and then we see that everybody is fine and there has been no toxicity, which can be, which is seen to be able to stop this the particular cohort. Then we go back again and increase the cohort and expose another group of patients. Now, it can be a single ascending dose or it can be a multiple ascending dose, how we do it in phase 1. And you must be knowing about the phase 1 of the BAB studies as well. But anyway, in phase 1, it is very few limited number of patients who are exposed, and the primary need or the objective is to see that it is safe on the human population. And the secondary is, of course, to look at the maximum tolerated dose or the dose limiting toxicity, where we say that the drug has to be stopped. And not that every phase 1 actually reaches a DLT or an MTD. There are times when a phase 1 has to be aborted because it never reaches that stage. And we start all over again from the earlier phases. So once the phase 1 completes, we go to the phase 2, and you see there are 100 to 500 patients. The number of sites which are chosen for a phase 2 are higher than we have in phase 1. And the inclusion-exclusion criteria becomes a little more larger. Again, one thing which I forgot to tell you in phase 1 is usually tried in healthy volunteers, unless it is a molecule which is for the cancer patients or it is for an HIV. Otherwise, it is always tried on healthy volunteers. Talk about phase 2, we are not talking about healthy volunteers at all. They are actually patients. And it is only when patients are exposed to the drug, we can talk about seeing the efficacy of the drug. So here we are not only talking about the safety, but also the efficacy of the drug. And as I said, the number of patients are a little more than what we had in phase 1. The number of sites also will be more. And this is when, again, when you have your medical writing sessions today, you will understand how important it is to write a protocol for these phases of trials. These are all independent protocols for each of the phases which are written. And when I say that these studies are done in a global way, in a multicentric fashion, and many of the investigators will be the principal investigators, and there will be so many other people who will be working under the principal investigator, taking care of that particular protocol. Now, why do we think the protocol is so important for each of the phases? Because there are hundreds of people working on the same protocol, and they should follow the protocol like a Bible. A simple thing like taking a blood pressure should not be different in how their blood pressure is taken by a student who is in KLE versus a student who is in Apollo. The blood pressure should be taken in the same way, whether it's in lying position or it is a standing position or it is a sitting position, half an hour after the patient has already reported into the OPD, because it makes a lot of difference in the readings of the blood pressure. And ultimately, these are going to be collated and analyzed for the final reading of the blood pressure. So for a simple thing like blood pressure, if it has to be so very minutely detailed in the protocol, you can imagine how minutely detailed it has to be on when the drug is to be taken, whether it is taken on an empty stomach, whether it is to be taken on after having your breakfast, and then when is the samplings to be done, especially for a phase 1. If you have to do the multiple number of samplings over 72 hours, the samplings every half an hour, 1 hour, 3 hours, 5 hours, 7 hours. So we have to really follow it to the T, so that when the curve is made, we all know that there are not too many outliers from the way the protocol was designed. And that is very important in a scientific point of view, from a research point of view. So once we have cleared our phase 2, we go on to the phase 3, where we have 1,000 to 5,000 patients. Many more number of sites will be involved, many more number of principal investigators will be involved, and obviously many more number of research coordinators, project managers, etc. would be involved. And here again, we have to see that everybody follows the written document so very well and the regulations so very well, so that once the data comes out of our research, it is not only scientifically valid, but ethically compliant as well. And that is why it's so very important to know the ethical regulations of each country and the GCP, which is worldwide, to see that once the research has been completed, the research cannot be questioned on the way it was done or the way the human beings were exposed to the research. Going forward, yes. So it's easy. That was the generic way of how the drugs continue forward. The history of drug development, the clinical trial regulations, GCP, as I said, it has been evolving along with us with the times. And the treatments we have today and tomorrow are only available because of the development of clinical research methodologies and regulations that are governing the same. So if we talk about 1700s, there were not so many regulations, but research was still happening. Science was still taking its own stride. If we know how a human body works, it's because of research. If we know how a bacteria actually affects us and cause those bacterial diseases, and what is the pathophysiology of that disease, it is because people have worked on it, people have done research on it. But then there weren't many rules and regulations governing research, and those evolved as the research itself evolved across these years and centuries. It has been a long journey, starting with 20th of May 1747, on which the James Lind began the first known trial comparing the different treatments for scurvy then common among the sailors in the British Royal Navy. I'll tell you a bit more about it. The subsequent development of these have been influenced by several events and reactive and proactive steps taken by the research community, regulators, and the larger society and other stakeholders. Those days, the mode of communication was ship. And we know that to reach from one point to the other, it used to take months. And it was always that the fittest of the sailors were chosen for the journey. But once they started the journey, it was very common that more and more sailors used to fall sick just immediately after they left the shore. And what was the reason? Nobody could actually understand. So it was in 1747 where Dr. James Lind, he's a doctor himself, a surgeon himself, he thought, okay, let me divide the group into 5 different groups. And he first randomized trial, if we can say so. So that's why he is called the father of clinical trials. The first randomized trial, he divided the sailors into 5 different groups and exposed them to 5 different treatments. One was just the seawater, one was apple and cedar, one was citrus fruits, etc. And then he saw that the group of people who were given citrus fruits were actually recovered within the least possible time. And not only recovered, they were able to help the others who were sick. So that is the story of James Lind, and that is the story of the father of clinical trials, who we, in our notes, as we say, is the first known person who has done a randomized trial, exposed different drugs, if we cannot call it drugs, different food items to these people, those sailors who were sick, and found out the reason for the sickness. So the moment they were given vitamin C, the people who were given all the citrus fruits recovered. That means the disease was caused by deficiency of vitamin C, and scurvy till now is treated with vitamin C. So that was one research which guided the research forward, and people understood that yes, you can expose people you can make groups, you can give them different types of therapy and see who recovers the best. So A is better than B is better than C, or all are equally good. So taking this forward, then came the Nazi experiments, which there's no end to the number of experiments which were done during the war times. The prisoners of war, we all know, do not have any rights to them. Once they are prisoners, they can be treated anyway by the people who have taken them as prisoners. And that is how it was. These prisoners of war were taken for seawater experiments, where they were made to drink seawater. And we all know how salty the seawater is and how toxic it can be for the body. But then how do we know? Because there were people who were experimented upon with the seawater, and they understood that yes, seawater will change the whole pH of the body and the person will not be able to survive. Similarly, today we know that when we go to high altitude, we need oxygen. But how do we know it today? And how do we know that when people go mountaineering or people go for other activities higher up in the altitude, they carry oxygen? That is because these prisoners of war in the Nazi experiments were exposed to higher altitude without oxygen, and then seen what happens if oxygen is not given to them. How does the body react to the lesser oxygen levels which is found in the higher altitudes? Actually, after this, there was a Nuremberg Code which was framed, because here, as we say, they were prisoners of war and they had no right to say yes or no when they were exposed to any experiment. So after this came to the light of the researchers, of the general population, of the people who make the laws and the rules, they made it very clear that you cannot actually expose anybody to a human experiment without the consent of the person who is participating. And the person has to know that it is an experiment, and the person has a right to say no to the experiment only if he says yes, that is, he gives the consent, he can be a part of the experiment. And as and when during the experiment also, he has a right to withdraw from the experiment. So today, if we do any research, we will not call them as experiments. Today, if we do any research, the participant must and should read the informed consent, understand the informed consent, has all the rights to ask questions on the informed consent, and only if he is satisfied, he will sign the informed consent. And once he signs the informed consent, then only research related, any of the activities which are related to research can begin on that person. And during the participation period, anytime the participant feels he wants to withdraw, he has a right to withdraw from the study. Out of goodwill, we will ask what is the reason why they are withdrawing, because it again helps us in our data building. But he has a right not to share why he is withdrawing from the study. So that is what came out of the Nuremberg Code. Nuremberg Code. And there were trials which were held, long trials, and people who were very few people by the time the trial completed. There were very few people who were surviving, and they were actually given the notice that, what was done was very, very incorrect, very, very unethical, and these came up as a Nuremberg Code, and it's still followed. Then comes the Thalidomide Tragedy. So there are many other experiments, or many other unethical experiments, like if we can talk about the Willowbrook study. Now, there was a Willowbrook school in US, and that was the only school which was available for the American citizens, where the children who were mentally incapable or mentally depreciated were actually given as an admission. It was, of course, a government school. But then the Willowbrook people, the doctors in the Willowbrook school, wanted to do an experiment on the children. And then it was such that any parent who wants to bring the child to the school has to agree to the child being exposed to the experiment. And this also was very, very unethical, because the school is not for experiment or research. It is a school for the mentally disabled people, and they were exposed to this type of experiments. If you see the screen, it is on the Thalidomide Tragedy. Now, the Thalidomide Tragedy is a little different. It came through the process of clinical research. There was research done. Thalidomide as a drug was actually very good for treating nausea. And it was approved by many countries, though US FDA did not approve it, but many other countries approved it. And it was an OTC drug for nausea. It so happened that the pregnant women started taking it for their nausea and morning sickness. and they really started feeling relief out of nausea and morning sickness. And many more people started using it and in that way. So what happened during this, after a few 1.5 years of using, many children who were born were fecomelic. So if you see these children, they do not have they have flipper-like arms, so would be their legs. So when the doctors actually started doing a due diligence on all these pregnant women who had normal pregnancy otherwise, but gave birth to fecomelic children, the common history that they found is all these women were exposed or had taken thalidomide during their pregnancy. And though it was an approved drug at that point of time, it was taken off the shelves. And later, after this thalidomide tragedy, it became very important for the sponsors, for the researchers to not only give the efficacy data, but also the safety data. Till then, it was the efficacy data which was more important. Yes, it works in such and such group of people. Yes, it works in, hypertensives, or it works, as I said, in nausea, vomiting, etc. But it's very important not only whether it works or not, but whether humans are safe to be exposed to that drug. And it is the thalidomide tragedy which, during the, as I said, the development of clinical research, during the development of research as such, the thalidomide tragedy brought into an amendment called the Coffer-Harris Amendment, where it was very clearly said that you have to tell us about the safety of the drug before we can give you an approval. And it's very mandatory now to give not only the efficacy data, but also the safety data of the drug before the regulators can give an approval. So the next slide which you see is the Tuskegee trial. Again, a very, very inhuman trial. Here, the Black prisoners they wanted to see what does the disease syphilis does to people. Now, syphilis can be in Blacks, in Whites, in Hispanics, in Indians, in anybody. But they chose only the Blacks who were in the prisons of America and suffering with syphilis. Now, they wanted to see the way the disease progresses, the way the syphilitic person finally, dies. So if we know today, those days, pulmonary tuberculosis, they used to lose, they used to have fever, they used to lose weight, they used to cough, then they used to cough out blood, and then they used to die if they were not treated in those days when there was no treatment for pulmonary tuberculosis. Similarly, they wanted to study and understand what is the route that a syphilitic person follows before death. Now, it's okay to do that, but there are few things when you say, understand the scenario, you will think it is unethical. First of all, as I said, syphilis is not only a disease of the Black population. So that was one minority group. Syphilis does not affect only the people who are in the prison, but the prisoners were chosen. That is another group who do not have any say or any rights. And the third most important thing is, while we were doing the study on Tuskegee, on the syphilis patients, penicillin was discovered. And they clearly found out that the penicillin actually will be able to treat syphilis. You as students today, would you not think that you will give that drug which actually treats the disease for those people who we know that they are suffering with the disease? We would be the first, those people would perhaps be the first people who would be given the drug. But then the researchers of those times thought, if they give penicillin to these people, they will recover. And then these researchers or the scientists will never know how a syphilis patient dies. So just imagine, those are our fellow human beings conducting research, but research was so much in their focus that they forgot the ethical rights of a human being. And these patients were devoid of penicillin and they died their death. The only promise given to them was a peaceful burial. And by the time it was actually known by the population, known by the, I would say, the media, and it was Clinton in those days, he actually went and apologized, and the few members or the family members who were still alive were given a compensation. But can you think, can we ever compensate for what they went through during the study, even though there was a therapy available? So that is why the evolving of research is so very important. We have been doing research, but we keep evolving on how we do research, and it keeps teaching us on what not to do for the sake of science. Science, of course, takes the foremost, but then there are many other things which dictate us or tell us what is ethically possible and ethically valid and ethically not valid to do. And that is when the regulations come into picture. So I'll just take you through another slide, which we have discussed in. So look at this one. So in 1927, of course, the Food and Drug Act was there from the year 1906, and the FDA was created in 1927. Then there was a disaster called the Sulfanilamide Disaster. At that point of time, this was an antibacterial, and they thought that it really works for bacterial tragedies and we can give it as an elixir. And it was made an elixir and given. But this was the cause of actually mass poisoning, one of the largest poisonings ever heard during those days. And from then, it is the 1937 Food and Drug Act came, where the drug safety became very, very important. Then comes the Nuremberg trial, which we talked about, and it gave rise to the Nuremberg Code, where it was important for a voluntary consent, which we still follow. Of course, it has become an informed consent. It was important for a voluntary consent. We can do human trials only after we completed our animal trials. Trials or research can be done only by medically qualified people. Not anybody and everybody who wants to do a trial can start having and doing a protocol and exposing patients on that protocol, but they have to be medically qualified and thoroughly able to do a research, understand a research, and take the research forward in an ethical and scientific manner. Risks should never be more than the anticipated benefits. Now, again, when we talk about benefits, we are talking about anticipated benefits because it's a new drug. We are exposing it to a population. We really do not know what will be the benefits. But at least we, after having done the animal studies, the phase 1, the phase 2, we keep understanding what may be the risk and what is our anticipated benefit. At any given point of time, if we think that the risk is outweighing the benefit, the research has to come to a halt. I'm sorry. Then comes the Thalidomide Tragedy, which I talked about, and it gave rise to the Coffer-Harris Amendment, which really says that the drug safety and efficacy and the reproductive studies have to be submitted to the regulatory authority for the regulatory authority to take a discussion forward and understand whether to approve or not to approve the study. The Declaration of Helsinki, which came after that, made informed consent voluntary. Now, it was a voluntary consent. The person gives a consent when they're told about the research. But here it is an informed consent, and this is followed till now. And if you see any of the informed consents for any of the trials now, you will see why is the patient being chosen, what are the other therapies available, what are the anticipated benefits, what are the risks, how many times does he have to come to the hospital, or what all tests will be done on him, that he can withdraw at any given point of time. So it's a huge document and very, very regulatorily compliant document, which the patient will have to sign before he participates in a clinical trial. For a vulnerable population, it's a special code of ethics which is applied altogether and has to be clearly justified when and how a vulnerable population can be taken into a clinical trial. Again, the trials have to be done only by medically qualified people, and the anticipated benefits should always be more than the risks. And if there is any skewing of the risk-benefit ratio, the study should come to a halt. And mind you, students, what we are talking is what we are following. There's continuously a safety monitoring board which looks at the protocol as it goes forward, and it looks at all the benefits and the risks, the side effects, the serious adverse events, the adverse events, and keeps a close watch on how is the protocol progressing. And if there is anything that is questionable, the trial is put to a halt, or maybe the trial can be stopped as well. So going forward, we had in 1974, the National Research Act, 1974, 1979, the Belmont Report, which looks at the institutional review board or the institutional ethics committee. Now, none of the hospitals can do a study if they do not have a registered institutional ethics committee. So going forward, there was an IRB. Now in India from the year 2013, it has become imperative that every ethics committee has to be registered with the CEDESCO. Respect for persons, justice, and beneficence has to be the core element on which the protocol is built up. And 1996, the ICH GCP came into picture, and we are being ruled by that, though there have been many reviews and updates and additions to it. But yes, any trial has to follow these basic principles when we are doing a trial. So this brings us to the end of the clinical research. What is clinical research? Why do we do clinical research? And how we have evolved in the process of clinical research. So thank you. I will just go back to, I will stop sharing. Thank you.
Thank you, Chandana, for
Much.
letting all the audience know on the various events like the Nuremberg Code, the Thalidomide Tragedy, the Tuskegee Trials, which led to the evolution of the good clinical practices. Thank you for the very informative session. Are there any questions from the audience? I request all the students once again to be more interactive. Please let us know for any questions, any doubts you have, and we are very happy to help you know the responses. Any questions for Chandana on the presentation?
I think all the audience can put the questions in the chat box.
Yes.
They may not be unmuted because of the large number. I request all the audience can text so that immediately we can take the question and request Chandana Pal to respond.
So Rabani says she has a question. Yes. Please do type in your question so that I can let Chandana know.
You can please text your questions in the chat box. We may not be able to unmute you. Okay. Oh, she has disconnected.
So Nabeen asks, how do we get into clinical research after MPharm? So again, so we will get into such questions as we go over the sessions through the day. So right now, I would encourage all the students to ask any specific questions to Chandana related to the presentations we've just had. So I repeat, please do type in your questions for the presentation what we've had. So Rabani says, actually 15 years to take drug discovery, but now we have COVID vaccine within 2 years.
Yes, so that was the need of the hour, and we should actually pat all of us who are in the clinical research arena, that this vaccine, as you said, it takes 12 to 15 years or more sometimes, but this could be made in 10 months, and it is all over the world, all the researchers, including all the clinical research participants, because had they not participated, the vaccines would not have come. So imagine so many people, healthy, unhealthy, with COVID, without COVID, actually participated in research. I would say, it's a greater thumbs up for all those people who participated in because vaccines might have been made in the research process. But if we do not have done the clinical research, we do not have the participants who are ready to take the vaccine when we know it's so very new. They have exposed themselves, they have given us the data that we need, and we could confidently go ahead with the approval of those vaccines, and millions have taken the vaccines now, and that's how we are at a better stage today, I'm sure. So thank you for that question, but yes, it feels proud to be a part of clinical research, to have been able to reach this stage in 10 months rather than 10 to 12 years.
Thank you. So before we move on to the next question, I request the IT team to make Rutheja as the presenter. It's Rutheja Joblecor. So I request the IT team to make her as the presenter. So Chandana, we have the next question. So in which phase do we check the efficacy? I mean, we check the effectiveness.
Yeah, as I said, in the phase 1, we are only looking at safety. We are only looking at safety because in phase 1, as I said, we are also using human volunteers unless it's an oncology drug or an HIV drug. So there we're only looking at safety. But once we go to phase 2, we start looking at efficacy as well, because from then on we take the patients and we can see whether the drug A or the drug B is actually affecting whatever is the disease of the patient, whether it is hypertension, whether it is diabetes, whether it is cancer, whether it is rheumatoid arthritis. So then on, not only safety, but efficacy plays a great role in taking the molecule forward. So phase 2 onwards, it will be the efficacy as well, along with the safety.
Okay. Thank you. So the next question I have is, what is the difference between clinical trial and clinical research?
Good question. So clinical trial, if we say, we are looking at a new molecule, basically. We are looking at approval of the new molecule. When we talk about clinical research, it need not be always, approved by the regulatory authorities. It may be like when we do our studies in the hospitals, for example, the MD doctors have to do a study. They do not have a new molecule. They are working on all existing products and they want to do a A plus B or a A minus B or A plus B plus C type of a study, which can be a hypothesis for a clinical trial later. So these can be called as investigator-initiated studies or academic studies, whereas clinical trials actually involve newer molecules, newer regimen, and they need a regulatory approval as well. So all clinical studies which are done in the hospitals by the doctors, by the healthcare workers, can be clinical research studies. But any clinical trial has to be done by the sponsor and has to have a regulatory approval and then only taken forward, saying that a clinical study can be a basis for a hypothesis of a clinical trial. Yeah. And the numbers exposed in a study is much lesser than the number exposed in trials. We are talking of thousands when we are talking about a clinical trial.
So we have one last question, Chandana. So I hope I'm understanding the question right from Sagar Puli. Please emphasis on post-trial access. So I guess Sagar wants to know as to how the drugs can be accessed post the trials.
That's a very good question, Sagar, and I'm sorry I do not know your background. But yes, post-trial access is something which was there, but made it more by NDCT rules, which came in 2019. The post-trial, post-drug access has become more important. As you said, Samprita, perhaps that's what he is asking.
Yes.
So here, when a participant or a volunteer has volunteered himself or herself for a clinical trial, and let us say the investigator feels that that particular volunteer has actually shown efficacious results of his participation. Now, when we are talking about a clinical trial, the positive effect of the study is not yes or no is a statistical answer. Not everybody gets the right, therapy out of the treatment that he has received. But this is a one-to-one, case-to-case, patient-to-patient decision, which the doctor might take if he thinks that the patient has actually, by participation, the patient has actually benefited, and he will have to take a one-to-one approval from the ethics committee, from the sponsor, get the drug from the sponsor, and from the regulatory authorities also. And it is not, again, a lifetime approval. You have to take time-bound approval per patient for a particular protocol, and that is post-trial access. And this has become, from NDCT rules 2019, ever since we have started following that, it has become a part and parcel, because ethically also, when we feel that if a patient has taken a drug, now let us say the disease of the patient is a chronic disease, you have given the drug only for 1.5 years, you have gotten your results, you moved on, and you left the patient in the lurch. That's not ethical. So taking the ethical point of view and the scientific picture also, the principal investigator, if he feels that the patient has benefited, then he has asked for an ethical approval and a regulatory approval to continue that particular patient on that particular drug. But it is out of clinical trial. He no longer falls into the clinical trial rules and regulations. No serious adverse events are counted, no compensation, nothing is applicable for a patient who is on post-trial access. I hope I could answer that. Thank you.
Yes.
Yeah.
Thank you, Chandana. So I do see many questions coming up in the chat box as well as the question box, but in the interest of time, we'll move on to the next session. So I just want to notify everyone that I'm making note of rest of the questions, and as we move through the other sessions and based on how much time we have, we will surely try to address all the questions. So thank you, Chandana, for the very informative session and patiently answering to all the questions.
Thank you so much. Thank you all. Have a great day with us today.
So we'll move on to our second speaker for today's session. So we have Rutheja Joblecor, who is the director, SSM AstraZeneca. So she's into site management and monitoring in AstraZeneca, India. She has a total of 15 years of clinical research industry experience, project management, risk-based monitoring, quality assurance, and team management. She's executed various roles ranging from a CRA to a project manager, line manager, as well as an auditor. She's worked across multinational studies across various phases, 1, 2, 3, and 4, with wide range of therapeutic areas like oncology, ophthalmology, endocrinology, cardiovascular, neurology, infectious diseases, vaccines, and so on. So in her current profile, she's working as therapeutic area lead for general oncology, along with people management activities. So she's been an active member of ISCR Training Council. So apart from ISCR, she's also involved as training faculty for CDSA, NCG, BIRAC, and ACE Institute. So today we have Rutheja speak about the overview of good clinical practices in continuation to the presentation we had from Chandana. Rutheja, I welcome you to this event and over to you. Rutheja, are you able to hear us? So I get to see that Rutheja has not been added as a presenter. I request our IT team to kindly add Rutheja Joblecor as the presenter for the upcoming session.
Done.
Rutheja, can you kindly check now?
Yes, hi Samprita. I just joined in as the presenter, so if you can just give me 1 minute to connect. Yeah,
sure, Rutheja.
Correct.
Okay. Thank you so much, Samprita. And thank you for making me a panelist. Now I can actually, I'm able to talk and present my screen as well. So thank you so much for that. Thank you, Chandana, for taking us through the, okay, I think we have lost. Okay, so thank you, Chandana, for taking us through the background of clinical research. So why clinical research is important, what are the basics of what is the history and other aspects. So I would like to continue from the point where Chandana has left, and I'll quickly present my screen. So today I'll be talking about overview of GCP. Now let's talk about ICH GCP. Now, Chandana gave us a very good background about what exactly was the history, how different tragedy had happened, what were the shortfalls or what were the events that led to GCP, so development of GCP, right? Why ICH was put in place at the first place. So today, what I'll be talking about, first I'll be giving you introduction to good clinical practices. I'll be talking about the amendments. Those were made to E6R1. Now we are calling the amendments as E6R2 and its implications on conduct of the trials, as well as I'll be talking a little bit about guidelines and regulations for conduct of the clinical studies. So now to begin with ICH GCP. So now what ICH stands for? So ICH stands for International Council for Harmonization of Technical Requirements for Pharmaceutical for Human Use. Okay, so ICH is basically International Council for Harmonization. I've underlined the term. Now let me give you some background about ICH. Now we saw different events at the last, so when Chandana showed us the slide wherein she had put in a slide with all the events together, and then she talked about ICH being developed. Now, the concept of ICH first was initiated. So the root or the thought process of ICH, or having a harmonized approach or bringing some regulations, some guidelines to conduct a clinical trial, started somewhere in 1980s. So that was started, or that was initiated by the Europe region. So European Union was the first pioneer to start or think about harmonization concept. Then what happened in 1990, there was a meeting that was being held in Brussels by regulatory agencies of Europe, Japan, and US. So all of these together, what happened is that so that's where we say is the birth of ICH. So birth of ICF actually happened in 1990, and from there on, the ICH guidelines started to develop. And finally, we see that first ICH guidelines were released in year 1996. Okay, then as we are progressing into clinical research, there are a lot of developments, there are newer technologies which are coming in place. So there is continuous improvement that is happening. And we saw a second guideline, a revised guideline, so addendum that was been released to the first guidelines that happened in 1996, released in 2016. So in 2016, 9th November 2016, the second guidelines were released. Now, to give you a brief background, now I told you initially that ICH was initially started by European Union, Japan, and US. So now currently, if you go and see the ICH GCP website, ICH GCP or ICH has, they have differentiated the members who are associated with them as members and observers. Okay, so currently there are few members. I wouldn't like to go through the complete list, but just to give you an idea, we have, apart from Europe, Japan, and European and US, we have Canada, we have Switzerland, we have Singapore, China, Brazil are also the members of ICH. And apart from that, we can see we have the Regulatory Authority of India. Okay, so CDSCO is an observer for ICH. So we observe the ICH GCP guidelines. The other countries include UK, Argentina, then South Africa, etc. So there are a lot of other countries who are part or who work as an observer, or who are included as an observer of ICH GCP. Now, why this ICH GCP was being first established? So ICH GCP brings together regulatory authorities. As I said, there are members as well as there are observers. So not just members and observers, there are also pharmaceutical industries, pharmaceutical teams who are pharmaceutical companies who are associated with GCP or ICH. So what happens is that they bring together regulatory authorities and pharmaceutical industry to discuss the scientific and technical aspects of drug registration. So everyone who is conducting a clinical research is on the same page, follows the same guidelines. It achieves, because of that, it achieves a greater harmonization worldwide to ensure safe, effective, and high quality medications, which are developed as well as registered, and that too in a resource efficient manner, right? So wherever the studies are conducted, the data across the regions where the studies are conducted is acceptable. Okay, and then GCP in ICH. So we saw what is ICH, International Council for Harmonization. And GCP stands for Good Clinical Practice. So together, we say we follow ICH GCP guidelines. Moving ahead, now let's understand more about, so I gave you a background about what is ICH and what is how it was being formed. Now, basically, what is ICH? Okay, ICH basically talks about the ethical and scientific quality standards. Why? For what? To design, conduct, record, and report the trials that involve participation of humans. Okay, so that is very important. So all the aspects, all the quality standards are being defined by the ICH GCP. Okay, so those are guidelines. Why it is needed? Again, we saw, first of all, to create a harmonization, and definitely after seeing the past events that Chandana described today, I think it was very much required to ensure the right safety and well-being of the trial participants, as well as to ensure the credibility of the clinical data that has been generated, right? So ICA-GCP will help us to achieve both of these things. When we are required to follow the GCP, so whenever we are generating the data for clinical trial, which needs to be submitted to the regulatory authority. So the intent of generating the data is to submit it to the regulatory authorities. And it also may be applied to other investigations, clinical investigations, which have direct impact on the safety and well-being of the human participants. So when we say ICA-GCP, we always say we put the patients first. So everything goes around the patients. So to safeguard the life, safety, and well-being of the patients is the prime most important thing for all of us in the industry, whoever is involved in the clinical research. Right, and why it is developed. So we saw the public disasters that have happened. We have seen cases wherein there are serious frauds, there are data forgery which happens. So in order to avoid these, as well as abuse of human rights, right? So we saw how the Nazis conducted the experiments on the prisoners of war. So that was abuse. So to stop all those things, these guidelines were developed, and then there are countries who follow these guidelines. So all the countries, if you go and visit the clinical ICA-GCP website, you will see all the members as well as all the observers who are following the GCP guidelines. Moving ahead now, how the GCP guidelines work. Now, the GCP guidelines are divided into 4 categories. So QSEM. Okay, so QSEM stands for, first Q stands for quality topics, S stands for safety topics, E stands for efficacy topics, and M stands for multidisciplinary topics. Now, what is quality topics? So quality topics basically those guidelines are basically developed to give or talk about the pivotal milestones. So pivotal milestones in the sense, I can give you examples of stability studies. So how the stability studies should be conducted, what are the guidelines that needs to be followed, all those things will be described in the quality guidelines. Okay, then safety topics. So all the topics which covers the safety of a product. So relating to in vitro, in vivo preclinical studies. So specifically nowadays, the non-clinical testing strategy to assess the QT interval, okay, is followed as per the safety guidelines. And this is the main QT interval is now becoming the main hindrance for most of the withdrawals from the drugs from the market as well, as on date. So most that's the because of the QT interval strategy, it's the market, the drugs have been withdrawn from the market, right? So safety topics talks about the safety guidelines, talks about those parameters, how you are going to perform or how the safety parameters needs to be done. Efficacy talks about relating to clinical studies in humans. So now safety talks about how the design conduct of the study needs to be done. What are the safety reporting criteria that needs to be followed in clinical trials? And finally, the multidisciplinary. So what they have done is that wherever they are not able to classify all the guidelines, they have put it under multidisciplinary topics. So cross-cutting topics which do not fit uniquely in any of the other 3 categories. So that is quality, safety, and efficacy are put in the multidisciplinary topics. So example for multidisciplinary topics is the MEDRA. So that is MEDRA, you will find it under multidisciplinary topic. Now, when I'm talking about ICA GCP E6 R2 guidelines, so which I showed you earlier, that I'm going to talk about E6 R2. Now, when I'm saying E6, okay, E6 refers to efficacy topics. Okay, so the guidelines that I'm going to talk today about ICA GCP E6 R2. 6 in the sense is the 6th guideline which is available under the efficacy topic. So if you go into ICA GCP website, you have to search under E category under QSEM, and in that the 6th guideline will talk about ICA GCP E6 R2 guidelines. Okay, that's how we read it. That's how we look at it. Moving ahead, now a brief history. I have already given you a brief history as how it talks about. Now, as I told you, the first guidelines, E6, for good clinical practice was finalized in 1996. Okay, so what does E6 describe? So E6 describes the responsibilities and expectations of all stakeholders who are involved in conducting of clinical trials. Then GCP covers aspects of monitoring, reporting, and archiving of the clinical trials, as well as addenda for essential documents and investigators brochure. Now, on the right side, I have put up a picture over here, which talks about the content or table of content of the E6 guidelines. Okay, so good clinical practice. So good clinical practice, ICH has been divided into eight different sections. So those are called as sections. So first section is about glossary, which gives you definition of all the terminologies which are being used. Then second is the principles of ICGCP. I'll be talking today about principles of ICGCP. Then third is the ethics committee responsibilities. Fourth are the investigator and the responsibilities. Then is the sponsor responsibilities. Then ICH talks, or E6 talks about clinical trial protocol and amendments, investigators brochure, and finally the essential documents which are required. So whatever I have mentioned over here are covered as separate sections in E6 guidelines. Then there was a need, as I said, we are continuously evolving. There was a need of addendum that had to be created for the E6R1 guidelines. So that was been created in 2016. That was been released on 9th of November 2016. So that is the addendum to encourage implementation of improved and more efficient approach while continuing to ensure human protection. So now let's understand what exactly was different in E6R2 guidelines. So E6R2 basically talks about a few parameters. So first, adopting a risk-based approach or having a risk management approach involved in conduct of a clinical study. Now, what is risk-based approach or what is risk management? So risk-based approach or risk management is to identify the risk right at the start of the study, to anticipate what can be the risks that can come when we are actually conducting the study and how we can better manage them, how we can put a tab on the risks that might come when we are conducting the study, and observing those risks as the study progresses. There could be additional risks which can come in. So then how we can manage the risks which are coming up, which are not pre-identified by us. So all those things will help us to enable a better oversight of the study which is being conducted. A small example which has been given is like, let's take example of a site or a study which is being conducted wherein there are a lot of protocol deviations which are happening, or a lot of deviations which are happening. So deviation is a risk for a sponsor. Okay, so if there's a deviation, probably the sponsor will have more oversight. They will conduct more monitoring visits. They might conduct an audit on the site because of the risk parameter. So there are different risks which are being pre-identified and those are being monitored. So that risk-based approach is something which the industry had started following, and ICH GCP has now also included the risk-based approach, not just in conduct, but also in designing the study. So when you're designing the study, the sponsor has to have a risk-based approach. Then promoting risk-based and centralized monitoring in managing the studies. Now, what is centralized monitoring? So just to give you again an idea about a lot of us now moving from paper-based records to electronic records. So when we are moving to electronic records, the things can be made available very quickly. So now the data management. So in data management, earlier we used to use paper documents to capture the data, paper case report forms. Now it's been moved to electronic case report forms. So we have the data that has been collected. That data, there is some data that can be monitored remotely, that can be kept a tab on remotely, the number of adverse events that are happening on the sites, any serious events that are happening on the site. So any significant changes that are happening. So there are a lot of data, medical data, as well as other associated parameters where those data can be checked centrally by the sponsor. The sponsor it's not required for the sponsor to go and visit the site. Okay, so that is what we call as centralized monitoring in managing the study. Right, so a risk base is something which has been stressed upon in ICGCP E6R2 guidelines. Then addressing, reporting, and follow-up of significant non-compliance. So there's a altogether different addition of quality management, which has been given or which has been added in the E6R2 guidelines. So GCP or E6 ICH wants us to focus more on how the quality has been managed, how the quality management process has been conducted. So that's a very critical parameter, and you will see a lot of quality things are being put in place or been implemented, how it's been monitored throughout. So quality is a very, very important parameter, right? So it also helps us to have the credible data which has been generated. Okay, so credibility is established because of that. Then next thing is very important. As we say, now we are moving, technology is becoming a very common thing. So we are moving into newer and newer technologies, electronic systems. So we have to adapt with the newer requirements, newer technologies. So has ICH adapted. And E6R2 talks about addressing the technological issues. So how the system needs to be validated. So there should be a backup, there should be safeguard. So all those things are being required now as per E6R2 as well. Then not just that, but also E6R2 now is stressing more on the responsibilities and the oversight of the sponsors as well as of the investigators. So investigator oversight and sponsor oversight is a additional, or investigator and sponsor oversight definitely is a very, very important aspect for conduct of any clinical trial. But now it's monitored. It has to be visible that both sponsor as well as investigator has proper oversight on the study that is being conducted. And definitely, if we look into all these things, finally what E6R2 is trying to achieve is improving the data integrity, right? So if we are going to achieve all these things, definitely a data integrity is definitely going to be improved. And as well as the most important point is that the investigator and the sponsor should have access to study data as well as the study documents. Now, it's important that when I'm talking from the sponsor's point of view, access to the study documents which are there on the site is mandatory. So the sponsor should have access to that document, as well as the data which has been generated by the sponsor. By the conduct of the study, the investigator should have access. So it goes in both the ways. So everyone, it should be a transparent process that needs to be conducted. Now let's talk about a very important parameter about the principles of ICH GCP. So the ICH GCP has got 13 core principles. Okay, now 13 core principles, I have tried to divide it into 7 main areas. Okay, so let's look at those 13 core principles. What are the 13 core principles? So first principle is ethics. So under ethics, what does the principle state? So first principle talks about the trial should be conducted, which should be consistent with GCP and applicable regulatory requirements, right? So whatever studies are being conducted should be compliant with the ICH GCP E6R2 guidelines as well as the regulatory requirements. So for us, for India, we are following the NDCT rules 2019. So for us, we should be compliant to both of these parameters. Then the benefits to conduct the trials should justify the risk. So Chandana did talk about the risk, the risk and safety ratio. So what are the risks associated? What is the efficacy parameter? What are the safety parameters? So that needs to be considered. That has to be there. So if a trial is going to be conducted, we have to make sure that efficacy is something which is definitely overweighing the risk associated with the molecule, with the investigational product. It could be different for different therapeutic areas. It could be different for different molecules. So that risk safety ratio that needs to be verified. Then most important about ethics, as we talk again and again, the right, safety and well-being of the subjects prevail over the interest of science and society. So patients should come first is what GCP talks about, right? So patients' safety, right, safety and well-being of the patient should be the first priority over science and society, right? So it should not be compromised at any cost. Then when a trial has been conducted, so what is a protocol and what is the science behind the conduct of a clinical trial? So non-clinical and clinical information on investigational product should support the trial. So if you see the trial, we always have a requirement of investigator's brochure. So there's a complete section in GCP which talks about investigator's brochure. So now investigator's brochure will describe about the molecule, about the preclinical studies, about the clinical studies, about all the data which is available to support the conduct of the trial or to support the design that the sponsor has proposed for a particular molecule for a particular study. Okay, so that should be present. Then clinical trials should be scientifically sound and described in detail in the protocol. Now, if we actually go through the protocol, there are different things that are required to be present in the protocol. So just to give you an idea, what is a rationale to conduct the study, what is the design of the study, what are the objectives of the study, what are the inclusion exclusion criteria, what are the events or the schedule of assessment that will be conducted. So everything along with that, other documents that will be used, other scales that will be used, everything is detailed, described in detail in the protocol, right? And that is what is required. So there is a guideline and it's how the protocol should be, what are the basic requirements when you're designing a protocol. Then the next parameter comes is about responsibility. So now we saw ethical considerations, we saw the protocol and science. Now we have a protocol. Now what happens is that all the ethics committee should approve the trial prior to the conduct. Okay, so the trial cannot be initiated if the ethics committee approval is not received. So not just ethics committee, even regulatory approval is required, but principles basically states about the first is important is ethics committee approval. Then medical care should be taken by qualified physicians only, right? So when we see about the medical care responsibilities which are given, or probably we say that the principal investigator is finally responsible, principal investigator can delegate it to a qualified individual only, right? Then individuals involved in clinical trials should be qualified by education, training, and experience. So there is a lot of emphasis which has been given on the education as well as training of every person who is involved in the clinical trial, not just from the hospital's point of view, not just from the but every stakeholder. So it could be ethics committee, it could be the sponsors, it could be the regulatory agencies, it could be the investigators team as well. So everyone should be qualified with education, training, and experience. Then the most important again is informed consent. So we saw about the Nuremberg Code, right? So informed consent should be freely given by every subject prior to participation of the study. So informed consent is a very, very important parameter while conduct of the study. So that is the 9th principle of ICH-GCP. Then principle 10 and 11 talks about the data quality and integrity. So accurate reporting and interpretation and verification of clinical trial information is important, right? So it is not only applicable for paper, but also any media. If it's been collected, if it's interpreted in any media, in any format. So everything should be properly done. There should be data quality as well as integrity should be managed in reporting. Then finally, it should protect the confidentiality of records and respect the privacy rule. So patient subject confidentiality is very, very important over here, right? So we should protect the subject confidentiality. So usually a subject has been given a code. So every subject will have a unique code by which the subject will be identified, the patient will be identified. Right, so data privacy, or it should protect the confidentiality of the patient as well. Then investigational products. So all the efforts that are being put around conducting of clinical trial is definitely for investigational product. So even if the investigational product is not a marketed molecule, it's under investigation, still the manufacturing, storage, and handling of investigational products should be as per the GMP guidelines. Then finally, the quality control, then the systems with procedures, there should be proper systems and written procedures to ensure quality at every aspect of the trial. So there's a continuous quality management that needs to be performed by the sponsor and as well as whoever is involved in the study in order to have a proper quality control of the study. Right, so these are the 13 core principles of ICH-GCP. So everything that is described later on in ICH-GCP, all the sections are based on these 13 core principles of GCP. Then this brings me to my last slide. So then when we are talking about conduct of clinical trial, or when we're talking about GCP, so GCP is a framework or guidelines. It could be a guideline. What we talk is ICH GCP is a guideline or regulatory framework. So NDCT are rules. Okay, so there will be guidelines to conduct of clinical trials. So first of all, we have to follow the local country guidelines and regulations where the study is conducted. So we are going to conduct the study in India. So we are going to follow the NDCT rules 2019. We will be following the ICH GCP guidelines E6R2. Then international regulations where the data is intended to be submitted. Now, for example, we are submitting the data to US FDA. So we are going to follow the international guidelines which have been given by US FDA as well for conduct of the study because the data is going to be submitted over there. Then local clinical study sites SOP. So every site, so site in the sense of hospital where the clinical research or clinical study is being conducted, that will have its own SOPs. So that needs to be followed. Whatever are the institutional norms, institutional requirements, sponsors SOPs needs to be followed. And finally, very important, the protocol needs to be followed. So whenever a clinical trial is being conducted, we have to ensure that we are following all of these guidelines in order to conduct a clinical trial in a better way. So that brings that, as I said, that was my last slide, and I'll give it back to Samprita. So if there are any questions that we can take, it was just a brief overview of GCP. So now people who are experienced, they will agree with me that having a session in GCP in 45 minutes is difficult, right? So probably a whole day or a 2-day session is also sometimes not sufficient to talk about GCP, right? So what is important for us to remember is about the principles of GCP, how we are going to implement it, what is the basis of having GCP brought at the first place.
I completely agree with you, Rutheja, and thank you so much for providing us with the overview of GCP and the 13 core principles and how important it is to uphold the rights, safety, and well-being of our subjects. And the most important, the key theme of the E6R2 guidelines, a very informative session. So now I open the session for questions from the audience. So as Sir mentioned earlier, you can please type in your questions because we will not be able to unmute you all. So please type in the questions in the Q&A box and we can take it from there. So I repeat, audience, the Q&A box is open for you all to type in your questions. Please do let us know any specific clarifications you all have on the ICH session we've had from Rutheja. So we've had one question again from Sagar Puli. So participant or subject, which is the right term to represent in clinical trial proposal?
Okay, so participant is something that is the industry has started using now, but if we refer to GCP, it talks as it states as subject.
Yes, thank you. So one of the participants wants to know the practical problems you have faced while using an ECRF, as we do not get to know more about in any literature.
Okay.
So I will repeat, Rutheja. So the participant wants to know what are is there any practical problems we face while using an ECRF? Because they are unable to find more details about it in literature. So the practical problems faced with an ECRF.
Okay, so I would like Samprita also to add on, because she is coming from the data management team itself. So probably I can tell you from the operations point of view, what could be the basic challenges which the site might face is that, first of all, for ECRF, we need a good connectivity. Right, so having a good system in place, having a proper internet connection is sometimes a problem, specifically if you talk about areas, because still for us, here in India, we are developing, right? So the requirement or the speed that we need for internet might not be the, or that might have some issues, that might be on downtime, if there is power failure. So everything, when it revolves around electronic systems, we need to have the basics in place, right? So that's a very important criteria. That's a practical problem, I would say, which has been faced, specifically if the sites are a little remote in the remote areas. So that's a common problem that has been faced. Apart from that, I think we have to be a little good, familiar in using the electronic media, right? So getting ourselves up to date on the use of the media is very important. Or how the electronic system will be used, because every sponsor might opt for different electronic systems, and every electronic data or EDC data will be based on different programs, or it will be based on different systems, wherein the application could be different. So learning and implementing those are sometimes a situation, but I think we are sailing through it properly, and definitely there is a huge amount of improvement that I can see. And as a country, I think we are doing pretty good in using the electronic systems. So Samprita, if you would like to add anything on this.
Sure, Rutheja. I completely agree to your point, and I must say that every day is a new challenge whenever we have new systems in place, because every system has its own specific features, be it in terms of the way data is being entered into the system, the way we perform the review process, and most important is the challenges what we face is with regards to the reports which we run. So data management is a field wherein everything runs through metrics, right? So we give a lot of importance to metrics, which is a very important component of a clinical trial process. So this is where we face challenges on a day-to-day basis, and we have to have ourselves accustomed to the new tools, to the new features as we progress and as we advance through the various new systems. So we have another question. So how long is a GCP certificate valid for?
Okay, so GCP certificate, so as per GCP, there is no, so nowhere it mentions about how long it is going to be valid, but it's always good to have a refresher. What we expect is at least once in every 2 years. So once in every 2 years, the GCP refresher course can be done. If you are already familiar with GCP, a refresher course is always better, because you're using those guidelines, you're working on those guidelines. So it's always good to stay updated and take a refresher course. So that's what the industry usually expects, and that's what you will hear from various sponsors as well as CROs, that at least once in 2 years is what you need to do.
Thank you. So the next question is, in India, do we have the proper on-site technologies available?
Okay, so as I said earlier, yes, as a country, I think we are doing great amount of progress, and we all should be really, really proud of the way in which we are moving towards the technology. And like I can give you an example is the data speed that we can get in for internet connection at a very low cost is pretty good now. So 100 Mbps, it's a common thing now, which has happened everywhere, but we are moving in the right directions. The resources are made available to us. We are ensuring that it is we are well equipped to be to probably say that we are able to perform the study equally as the other global counterparts for us. So I think overall in India, we are on the right track, and yes, we have the required technologies, what is required for now.
Right. So yeah, I would like to add to what Rutheja said. So I was remembering my times 10 years back when we used to wait for specific instructions from our global counterparts to perform any kind of activity. So now I'm very proud to share with all of you that we are the global hub. India is the hub, and we've seen a boom in the clinical research industry, and we are equipped to perform end-to-end activities across all the departments, across all the phases. So that's how we have progressed, and that's a good a move what we've seen in our industry. So we do not have any further questions. So I thank Rutheja once again for the detailed updates regarding the ICH and the GCP guidelines. Thank you, Rutheja.
Thank you so much.
So now, as per the agenda, we will have a quick 15-minute break, and we shall assemble again in 15 minutes from now. So yes, let's have a break, guys. Thank you.
Hello, Samprita.
Yes, Ulas.
You're online, right?
Yes, I'm there.
So do we have Payal joined?
Just checking.
Yes, yes, I have joined.
Okay, so IT is giving you the presenter access.
Okay, sure.
Sure, thanks. Sorry, Payal, can I ask as what name you've joined?
It's by the name of Samprita only, because while I'm joining the link that I received, I'm not able to join as a panelist.
Oh, that's the reason. I think they are not able to find your name in the list.
So it's a co-host. Maybe you can just... And I'm not also able to change the name or anything. Oh, wait a minute.
Probably, Payal, you'd have received the link once you register. Yeah.
Yes. Samprita, I did join by that link.
No, I
but
think they
then,
got you.
I have just changed the name.
Okay, good.
Thanks.
So I think we still okay. It's 11:50. I think we can cover during the break, 30 minutes break.
We were supposed to get started with our next session at 11:45. So we are 7 minutes past that schedule. So we will surely try to cover.
Yeah, I think we are ready to start. I mean, you can give Payal presenter access, Ramesh.
Okay.
Payal, you should have it. Please, can you check and let us know?
Not yet.
Yes.
Yeah. Okay.
We can see your screen, Payal.
Okay, great.
Great. So we're good to start?
Yes, Samprita, please.
Okay, so welcome back, everyone. So we've had a good start with regards to how the clinical trials were being conducted way back then and what exactly led to having specific guidelines in place to uphold the rights, safety, and well-being, and what exactly are these guidelines. So as we move on, so you'll get to hear more about the clinical research industry now. So this is a very vast industry with multiple stakeholders, with various departments in place. So we now have our speakers who will take you through the various departments who play a very active role in the clinical research across all the phases. So we now have Payal with us. So Payal Jain has started her career with IQVIA as a CTA. So this is previously known as Quintiles. She then moved to a CRA role and now working at ZTI. So currently she's working as a senior project coordinator at Zydus Therapeutic Inc., that is Zydus Cadila Healthcare. She oversees the project management primarily for clinical trials conducted in the US. She has around 11 years of experience in the clinical trial industry, where majority of her roles have been into ClinOps, that is being a CTA, CRA, regional CRA, and protocol lead CRA. When it comes to her education background, she's completed her master's degree in biotechnology, followed by 1-year PG diploma course in clinical research. So Payal, I welcome you to the session today. So the next session is about Introduction to Clinical Operations from Payal Jain. Over to you, Payal.
Thank you, Samprita. Hello, friends. It's an honor and privilege to be amongst you all today and introduce you all to the clinical research and primarily clinical operations world. You know, everything has come on paper, the protocol has been designed, the guidelines have been framed. Now, what does the clinical operations do? Clinical operations basically help in executing that trial. So, you know, this industry has been working silently over years, playing a pivotal role in serving the human mankind. But of lately, clinical research has become super famous, all because of COVID. People have suddenly started taking interest in my career. Wherever I go, people ask me, hey, are you working with Zydus? When is the vaccine coming? And, all such sort of questions. So not diverting much from the topic, let me take you through what clinical operation does and who are involved in this. So topics that we will cover today is the clinical trial key stakeholders, the sponsor and their team, clinical operations team, operations life cycle, operations as a career, and lastly, the summary. Now, before I jump into further slides, I would like you all to take cognizance of some of the general terms that we use day in and day out in our job. So what is a sponsor? A sponsor is an individual, company, or an institution who takes responsibility for the complete management and financing the clinical trial. And what is a CRO? A person or an organization contracted by the sponsor to perform one or more sponsors' trial-related duties and functions. What is a site? A site is a hospital or clinic where trial is conducted. You would be happy to know that KLE Hospital itself is a site, and there are various clinical trials conducted at KLE. In fact, I, as a monitor, got a chance to visit KLE and perform a monitoring visit at KLE Hospital. Who is an investigator? An investigator is a person who's responsible for conduct of the clinical trial at a trial site, basically a doctor and his team who conducts the trial. And lastly, the CRA, very important, clinical research associate, also commonly known as a monitor, who supervises, monitors, and supports the administration and progress of a trial. Now, for any industry, there are several key stakeholders involved in smooth functioning of it. For clinical trial industry, following stakeholders are involved. Sponsor, investigator, we currently saw the definition of all of them. And the ethics committee. Now, what is an ethics committee? Ethics committee is an independent body constituted of medical professionals as well as non-medical members whose primary responsibility is to ensure that the protection of the rights, safety, and well-being of human subjects, something about which Rutheja spoke in her last talk, and how important it is to, maintain the rights, safety, and well-being of patient. So that is the role of ethics committee. Then the study subjects, also known as participants, patients who basically participate in the trial. Then the regulatory, the bodies who have the power to regulate the trials. Like specifically in India, we have Drug Controller General of India. In US, we have FDA. So these are the key stakeholders in the clinical trial. Now coming to the sponsor, like I'll give you a small example. I have my brother's wedding. What do? Either I manage it on my own or I hire wedding planners. Either I give them the complete responsibility to manage the wedding, or I outsource some part of the wedding functions to them. Likewise, what does the sponsor do? They have a molecule now, they want to conduct the clinical trial. Either they outsource some of the activities to the contract research organization, or they give the complete responsibility to the CRO, or they themselves manage all the activities. So that is the sponsor. And coming to the sponsor team, what comprises of the sponsor team? So there is a pharmacovigilance department, there's a project management, there's clinical operations, regulatory team, site startup team, data management, as well as statisticians team. So, project management and clinical operations work very closely together. Sometimes it is very difficult to differentiate between the project management and clinical operations. But for now on, we will just focus on the clinical operations team. clinical operations' primary responsibility throughout the course of trial is to consistently ensure that the trial is being conducted at the sites as per the study protocol, the GCP guidelines, and the regulatory requirements, and the protection of rights, safety, and well-being of the patients is maintained throughout the trial, and the data that is generated out of the trial is accurate and real. Basically, there should be no fraudulent data. So that is the primary responsibility of the clinical operations department. So let's see, who all are there in the clinical operations team. So there's a startup team. They basically help in identifying, qualifying a particular site, hospital for the trial conduct. There's a clinical trial assistant who's based in-house, based in office, and supporting the day-to-day activities for that particular trial. The clinical research associate who basically travel to different clinical research units where the trial is being conducted and ensure that trial is running correctly and smoothly. Then there's clinical trial managers who manage the teams or running of the trial. And lastly, the operations head, head of this particular group who owns complete responsibility of smooth functioning of the trial. So this is just the basic designations that I have covered upon. There could be various sub-branches to it, depending on sponsors to sponsors, contract research organizations. So out of these members, there's one team member who plays a vital role in monitoring the trial and who is a monitor. They are the main communication link between the sponsor and the investigator. As I just recently mentioned that they're traveling to different sites very frequently. So, they are the sponsor face for the investigator or for the site. So I will explain you how, trial is being conducted at several locations. So as I mentioned that monitor travels to these locations, reviews and monitors the data, and then they report it to the manager, the project manager, as well as the clinical trial manager. They prepare a visit report that is a detailed document and write down whatever they have reviewed and observed at that particular site. So that is their primary function. In short, CRAs have responsibilities that cover the full life cycle of a clinical trial. So coming to the clinical operations life cycle. So though a lot of work is done in the background, operation group comes in picture with the site identification and the startup. Site monitoring is an ongoing activity once the site has been initiated or started. Then lastly is the site closeout. I will talk about these more in detail in my subsequent slides. So coming to the site startup. Now you all are students. When you choose your college, what all you see? I'm sure you will have your checklist in place and you do a lot of research before going to a college, like what course you want to do, whether it is available at that particular college or not. What is the faculty? What is the infrastructure at that college? What is, the facilities that college is providing you? What is the connectivity of that college to your home? And whether it's a private or a government setup, whether, what is the affordability, whether you can afford the fees of that college or not, how are the placements, etc., etc. So I'm sure these are the basic pointers that you would pick upon before you select the college. Likewise, this startup team has their own checklist. They have a huge database and from which they identify suitable hospitals, doctors who are qualified and have good experience where they would like to conduct their specific trial. So once they identify the site, there's a mutual decision taken by the project management, the clinical trials team, and the CRA to select that particular site. So once the site is selected, we start the site, which is typically called the site initiation visit. where clinical operation teams perform following activities. As I mentioned, the clinical trial associate, they are largely based in-house. So what they do is they prepare the site master binders, which includes all essential documents. This is clearly explained in the ICH GCP guidelines, the essential documents you would see in section 8, and then they send it to the site. In clinical trial, there's a famous saying that if it's not documented, it's not done. So these documents are very important. And also they send required equipments to the site. And once the site initiation visit is conducted by the CRA, what they do is they train the site team on various aspects of the trial, like protocol, and they also review the site infrastructure and ensure that whatever facilities are available at site are sufficient to conduct the trial or not. And what also importantly they do during the site initiation visit is they discuss patient enrollment recruitment strategies with the investigator, that is with the doctor and his team. And the trial manager post site initiation visit, what they do is they review and approve the site initiation visit report, which is submitted to them by the CRA. And they also manage any query or anything that comes up from the CRA. So again, once the site is initiated, there's a frequency that is set up by the project management team that, okay, this site, you will, the CRA will go for monitoring visit every quarterly, or, once in every 4 weeks, or once in every 6 months or something like that. Frequency is set up. So a monitor travels to that particular site, they verify the source documents. Now, what are source documents? The doctor's prescription, the patient lab reports, all those documents comprise of source documents. And then they match it with the entry in the patient data system, like ECRF. Largely, most of the sites, most of the sponsors have moved to the electronic patient data system. They also review the consent forms and process the informed consent forms that has been completed by the study subjects. And they also check the overall compliance of the trial. The study drug compliance is very important, how much study drug site has received, how much study drug is currently stored at site, and how much study drug has been given to the patient. So all that compliance checking is very necessary to ensure that there's no misuse happening with the study drug. Then training site members if required. So this is an ongoing process. And CTA, before the site monitoring visit, they provide any required documents to the site. They also track the finances for that particular site, clear site payments, very important. And then if there's any ongoing study update, then they share it with the site. Trial manager, again, they review and approve the report. And if monitor is unable to resolve any issue that they see at the site, then it is escalated to either the project managers or the trial managers, line managers. So these are the various functions of CRA, CTA that they perform during the monitoring visit. Now, lastly comes the site closeout visit, wherein a monitor again visits the site. Prior to the site closeout visit, it is very important that the patient data system, that is your ECRF, is completely clean. There are no queries in the system. So that is ensured prior to the site closeout visit. Then during the site closeout visit, as I mentioned, the study drug reconciliation is done to ensure that no study drug is being misused. And they collect the required documents from the site and ensure there are no outstanding payments. Otherwise, that becomes a major problem while closing out the sites. Then they collect and ensure that all essential documents are available at the site. And lastly, the archival of the study. because there might be a case that there would be a future audit inspection from the regulatory, or could be a sponsor audit. So it is very important to archive the documents. So, you must be thinking that out of all these, what can be a potential career path for me? So just giving you a glance of opportunities that you can grab as freshers if you are looking to make a career in clinical research operations. So clinical research associate, in-house CRA. In-house CRAs are basically, based out in office. They don't travel frequently, because of all this COVID situation and everything. So things have turned remotely. There's a remote monitoring done. So in-house CRAs can manage those things. So terms might differ for a sponsor, for a contract research organization. So there's a site startup associate who basically helps with all the site identification and all those processes. Then there are centralized monitors. Again, they manage, the trials in-house. So there's not major difference between an in-house CRA and centralized monitors. Again, the term and designation might matter. Lastly, the clinical trial assistant. So basically, coming to the summary of the entire presentation, what is an operations? Operations, they perform a practical work or of something that involve the practical applications of principles or processes. At the end of the day, that's what clinical operation does. The practical job of carrying out the last step of drug development, a step, if that is performed correctly and effectively, can ease the suffering of thousands to millions of patients. From site startup to site closeout, clinical operation teams ensure that proper planning, conduct, patient safety, and data quality, basically, they execute the trial efficiently. And monitor is the main sponsor representative who acts as a bridge between the sponsor and the site team. And as we discussed just in the last slide, there are various career opportunities for fresher in the clinical operations. So that's it from my end. I would be happy to take any questions that you might have.
Thank you, Payal, for taking us through the various stakeholders who are involved in the clinical operations, the importance of documentation. So giving us details about the site initiation, monitoring, and closeout, as well as the various career possibilities within the ClinOps team. So it was a very informative session. Thanks once again. So for the audience, we have the questionnaire session open. Yes, I can see questions coming in. So Sagar again says he's an MSc, PhD medical pharmacology graduate and work in DST SARB funded clinical trial in Jimper. So where do I fit in clinical operations setup? So Payal, I repeat, he's an MSc, PhD medical pharmacology graduate and works in DST SARB funded clinical trial in Jimper, JIPMER. So where does he fit in the clinical operations setup?
So thank you, Sagar, for your question. As I just mentioned, for freshers, you can take up a role in the site startup, or you can take a role if you are excited to be in the field. You can take a role in the CRA since you have a MSc PhD background. So companies would be happy to hire you as a CRA. Generally, the freshers, it's typically difficult to get into the CRA role, as a fresher, but giving your MSc PhD background, you can quickly get into that. So yeah, that's what.
Also, Samprita, sorry to interrupt, but I just would like to remind we have a panel discussion at the end of the,
Yes.
that's the last program where career opportunities will be discussed more specifically. So
right.
if your question is related to career opportunities, then please, you can save it for the last panel discussion, yeah?
Thank you, Ulas, for that. So any specific questions with regards to the presentation we've had on clinical operations? Audience, please shoot your questions. So we have a question from Abhilash. So he wants to know the difference between SDV and source data review.
Thanks, Abhilash. So source data verification, I would say, is that. So you have a document in your hand wherein your vitals are written, or say suppose your vitals are written, and there's an electronic system in front of you. So what you do is you match the data, that is what is written in the prescription that you have and what is entered in the system. So that is typically called the source data verification that you are doing. And source data review is typically not different from source data verification. Source data review would be the document that you have in your hand. You are reviewing that, all details and everything is entered correctly. There's no crisscross done by the investigator or by the site team. So that is your source data review, basically. Okay, I see a question coming in. How can a monitor ensure proper documentation during the site visit? So that is an interesting question. I can go on and on for that because I have been a monitor for almost 6 years. So, you have to ensure that there's an ALCOA principle about the documentation, where you have to ensure that the data that is present in front of you is accurate, is legible. So if there's a data which you cannot read, so doesn't make any sense. So it is very important, that the documentation is as per the ALCOA principle, that is, accurate and legible and contemporaneous. So the documentation should meet that principles.
Any further questions for Payal? Okay, so thank you, Payal, for the wonderful session. So it's now time to move on to the next department, that is my arena. So we now have the next speaker, Ullas Arbhavi, who will give us a heads up about clinical data management. I welcome Ullas to the session.
Thank you, Samprita. Everybody can hear me well and see the slides on screen?
We can hear you, Ullas. We are unable to see your slides.
Now?
Not yet.
Yes?
It's coming up.
Okay, good. Did that load up yet?
Not yet, Ullas. Not yet. It's just showing as Cayley University has started screen sharing, but it's blank. So just wanted to check with the other panelists, is it the same case with you all, or is it just me who's seeing, okay, there we go. Yes.
Yes, okay. I think now it's on, right?
Yes, Ullas.
Okay,
Right.
sure. Thanks a lot. Okay, I welcome you all for this specifically data management oriented session. Out of all clinical research, you've been hearing talks on GCP, ICH GCP, clinical operations, overall drug development process. So let's go into clinical data management specifically now. So if you can go to the next slide, please. Yeah, so I would like to take you through broadly through these points. We will start with history, then look at the regulatory landscape. What are the governing bodies we have? Then the principles of data management, core aspects of data management, and how the flow of work, in data management and the people involved in this flow of work. What are the various systems we use? What are the phases? Typically, the phases. When I say phases, it's not phase 1, phase 2, phase 3 of clinical trial, but the typically in a study when we start, what are the various phases, then the significance of clinical data management overall, where it sits in the broader scheme of things in clinical research. And then I would like to leave you with the take home message, what CDM can do, and leave you with this message, like, so that you can go home and build on this thought. Just before we go any further, just a quick disclaimer, that all the views or the information that I'm sharing here is only my own views and not really associated with any of the organizations, companies that I may be associated with, right? And this is entirely for educational purposes also. Next slide, please. Okay, just to start with, I thought, it would be good to start with a little bit of a history of clinical data management, right? Quick reflection on the timelines, major timelines. Okay, so how it's been, and when you look at the history, people might put a different event in different categories. So I thought this would be what I may consider as major, right? So around 1960s, 1970s, I can say was the beginning of clinical data management more systematically. So the field of clinical data management started to become a profession. Like, people started getting into it and started to manage a few things. But of course, it was all paper based then. A lot of manual efforts. When you say paper based, people had to manage a lot of information, lot of data manually, right? And obviously that was all being done using basic in-house databases, and most of it, as I said, was paper, right? Then come 1980s, 1990s type of era, where the field began to grow a little. So then the arrival of SCDM, for example, that is just, I put that as a milestone there, because the Society for Clinical Data Management did a lot of things and tried to bring things under one stream and lay down some standards and bring people together. So concept of electronic data capture also started evolving in the second half of 1990s. So around 1998, 1999, we started to see a lot of electronic data capture systems. So what was before paper-based, now people started moving into handling things more electronically, right? So this was kind of a growth phase for us, and which saw also the first half of 1990s was probably more customized in-house database. What was basic in-house database before became more customized in-house database, and I will share you some examples of the systems in the coming slides, right? And around 1997, technology started evolving. Big change towards paperless trial started to get pushed, and this brought in institutions like CDISC. You can refer to the abbreviations, like, the acronyms at the bottom there. So CDISC and FDA also brought in 21 CFR Part 11, which is one of the major regulations that we have to keep in mind when we do things in data management. So standards started coming in, regulations started coming in, So which led growing, the interest started growing in electronic data capture systems, right? Then coming into 2000s, around 2000, 2002, Society for Clinical Data Management brought in a document called as Good Clinical Data Management Practice, right, which sort of tried to lay down how we should do things in data management. There are numerous chapters in there. So right from the beginning, when the protocol comes to us until the database log, what happens? So chapter wise, they try to describe and give some sort of guidelines so that the process becomes a little bit more streamlined, right? And certifications in CDM kind of like, started to become a trend. So for example, accredited clinical data manager, right? Certified clinical data manager. So this started around 2000s as well. So, and along with that, more and more EDCs started coming into market. So EDCs are like, they are like, brands, like they are basically it's a software, right? It's a platform using which you build a database. So previously, the second half of 1990s, there were not that many, but then come in 2000s, there were many vendors with their own EDC software, right? So accordingly, when you see a lot of new things coming in, you also see new regulations also and standards coming into picture as well, because you need to manage those new things that are coming into the market, right? Then 2019, 2020 and beyond. So what I'm trying to say is last 2-3 years, and from here on, what it is really evolution phase. We are into this big evolution phase right now, where big paradigm shift is kind of happening, if you like me saying that. The Society for Clinical Data Management also brought in white papers on this whole evolution process, where the data management, what used to be called as data management, is now being referred to more and more as data sciences. So of course, data science is a very vast term, which includes a lot of things under it. But then yes, by bringing in these three, the white paper in 3 parts, they try to describe it a little. So if anyone is interested, they can refer to the white papers that SCDM has brought in, right? And of course, the pandemic. So when we say last 2 years, whatever happened, when we are looking at it, we cannot exclude COVID-19. So what COVID-19 has done to not only us in clinical research industry, but also to the society as a whole, it has shown us few things, what we can do, what we can achieve, that otherwise before COVID, we didn't anticipate or we didn't know. So for example, work from home, for the very example of this virtual workshop, what we're doing. So we know 500 people have registered. They're sitting behind the screens of their respective laptops and mobiles in their respective homes, bedrooms, kitchens, wherever they are. And this is being broadcasted. Before 2019, if someone said this type of virtual workshop could be held, not many people would have believed. Some people might have actually laughed off. This is not going to happen, but now this is happening. That's what pandemic has done to us. So there is a positive thing in everything. So of course, pandemic has been distressing. There is a lot of negative impact happened on people and society and professions and careers and everything, but there is also a positive side to it. If we look for it, we have tried to identify what else we can do. Okay, this is a challenge, but how can we overcome the challenge? So the introduction of virtual trials, hybrid trials, decentralized trials, whatever you call them, it has started becoming reality. Of course, these terms were being discussed from past 5-10 years, but then what pandemic has done is brought it forward significantly. So what we might have seen after 10 years, because of this pandemic, it has made us act quickly on these new technologies, new concepts, new way of doing things much quicker. So we are adapting. So the pandemic has acted as a catalyst to the whole process. So welcome to the virtual world, including this session. We are all in this virtual world right now. So what we are going to see more and more in future is the real world data, real world evidence, artificial intelligence, machine learning. That's what you will see more and more. These terms were all discussed even before, but from here on, you will see these terms being discussed more and more in practical also. Not only being discussed, but also you will see them in practical life, So you might be soon joining a company where a clinical trial may be running, and you might be using some concepts of artificial intelligence or machine learning to conduct your respective jobs. Next slide, please. So, okay, we saw the history. We saw how it's been evolving, what we have done and what we are going to do. But then this is all being governed by few people, few institutions, few organizations. So we need to stick to few things when we do things, right? There is a rule book. So ICH GCP, it's not government body, it's a industry standard guidelines, ICH GCP. So that's there in the middle, in the center of it, right? So there are principles that CRAs have to follow: project managers, sponsors, PIs, data managers, safety associates. So they have to do their things as per these ICH GCP guidelines, right? But then there are country-specific guidelines as well. Like, I mean, more government guidelines and rules and things. So FDA, as you know, and I've tried to put in only the major ones here. FDA, as you all know, it's a US specific, I mean, but it governs most of the world. So FDA is US, it's got a lot of regulations according to which the clinical trials are being conducted. And then there is EMA, European Medical Agency. So that has got rules to go in more specifically in EU and globally as well. Then there is more Japan specific PMDA. Again, all the acronyms are there for you to read out yourself. Then TGA for Australia. And then we have our own CDSCO for India. So there is DCGI as per CDSCO, who governs, who looks at things and gives the licenses to initiate the trials, conduct the trials, and reviews things and audits things and make sure things are being done in a proper way, ethically, with consistency and quality, right? Few important regulations in data management are 21 CFR Part 11. For the scope of this talk, because it is going to be introduction to data management, I will not go too much in detail about what these regulations are. What I thought was, if you know these names, you can then go and search them up, because information is there on the web. It's all there. If you just Google, it's all there. It will tell you what 21 CFR Part 11 is, what GDPR is, what personal health information is, what HIPAA is, right? So, but these are few things that you should be aware, like, you know, in data management, right? What they do is they will make you work in a systematic environment, in a consistent way, and keeping in mind the requirements of the regulations, right? Next slide. Okay, we saw the regulations and regulatory bodies, and then I would like to come to the principles of clinical data management, and Payal kind of referred to it in the previous talk. The principles of ALCOA, right? ALCOA initially introduced in 1990s by FDA, according to which, you know, the data that you are handling, or that you need to make sure that it is attributable, legible, contemporaneous, original, and accurate. So this is what the central piece of it is. So the words are self-explanatory. So I don't really need to go into too much in detail to tell you what each term means. Attributable is who did it, when did this happen, and where did this happen. So you need to attribute the data that came to you to something, right? This is the data for this subject. This site coordinator collected it using particular system or whatever. So it needs the data that come to you needs to be attributed to something, right? Then legible. Legible is a very much self-explanatory term. You just need to understand it. It should be human readable, machine readable, if based on it, whether it is paper based or electronic data. So can you read it, understand it clearly, see it, make sense out of it, right? So that's legible. Contemporaneous. So did it get recorded when it actually happened? So in say, I mean, data cannot get recorded anytime you want. So it needs to be recorded more like, you know, when it happened, right? Timely recording of the data. So it needs to be contemporaneous. It cannot be out of the context altogether. Then original, of course, yes, it has to be original. It cannot be duped. It cannot be copied. It cannot be like, you cannot bring in any piece of paper. Even if it is a copy, it has to be a true copy with proper stamp and everything, like a signature saying that this is a true copy of such and such document, blah, 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 So originality of the data. Then comes accurate, yes, truthful, free from errors. Truthful is the actual data that got generated in any clinical trial, as accurately as possible. You need to capture it now. But when you are capturing the data, it's not that, in ideal world, free from errors, saying free from errors is easy. But when you imagine yourself in the site environment where nurses are juggling between the patients and the subjects and the doctors and a lot other things are happening. So there could be a few errors, but whenever there are errors, there should be, there are certain ways how you can correct errors. Like you cannot mask it, you cannot use the white paint, or you cannot erase things and do it again. So clear strike, note down the actual reading next to it, sign it, date it. So that's correcting the error. So when I say free from errors, please don't misunderstand. There can be errors, but there is a way to fix errors, right? So that's maintaining the accuracy of it. So this was around 1990s. But then as we moved on, people started thinking it is necessary to bring in few more concepts to support ALCOA, right? Okay, previously we laid down these guidelines of ALCOA, but then we as time progressed, this is the evolution process, right? So we started feeling like, okay, we need to add more principles to it. So complete, consistent, enduring, and available. So more like 2010 onwards or so. So complete as is, data as is, no deletions, no omissions, right? Even if it sounds like, this can't be it, but you still need to collect it and show it. The person who's going to analyze it, it is their responsibility to make sure what it is and how it is and all that. But no deletions, no omissions. Consistent is expected sequence, right? Consistency. This happens first, then this happens in chronological order. That kind of a consistency, right? Enduring, controlled environment, and tools. So it cannot be an open environment. Like, people cannot come in randomly and contribute to the data or generate data or capture data. So it has to be a controlled environment, fixed people doing fixed things, tools wise as well. So you cannot bring in, for example, any random USB or a CD or a DVD, or you cannot record like a blood pressure measurement or a vital signs on a piece of paper, like a random piece of paper, a rough paper, and then there are source documents, there are proper CRF pages if it was a paper study. So there is a section where the data should go on that piece of paper. So use a dedicated, authorized way of collecting information. So that's enduring. Then available. Is it available for me to view or review? Because, okay, you might have collected world-class data, Nobel Prize winning data, but if I can't see it when the auditor goes there, and if they can't see it, what's the use? So it needs to be there when required for viewing and reviewing. Okay. Right. Then coming to the core concept, now that we have seen principles, now the core concepts of it, what we do in clinical data management, right? So one thing we need to understand is we are dealing with data that affects people's health, people's lives. So you cannot, for example, there are industries, data science is a vast field, as I said in the beginning. So data science is, concepts of data science are used in different sectors, consumer industry, geology, mining, defense, or, there are so many industries where data science concepts are used. So similarly, health science also is utilizing the concepts of data science, but here we need to be a bit more careful. So consumer industry, say you might have all used Amazon, Flipkart, or all those online shopping sites, right? So once you buy something there, then the next time you log in, it will also automatically start showing you, giving you suggestions. You bought a particular shoe or a particular shirt or a pant, then it knows your interest, what this person is going to buy based on the previous purchases. So it suggests something. So no big deal. You bought Nike last time, you ended up buying Reebok this time. Doesn't matter. It's not going to kill you. It's still a shoe. But here you cannot you need to be a little bit more careful, because the data you're dealing with the data that is people's health. So what you do, it is going to have a bit more larger ramifications and consequences. So people who take up careers in clinical research as a whole, I'm going to say, not restricting it to data management only, they need to be they need to act more responsibly, more diligently, more eyes open, attention to detail kind of environment. Like, because why we are saying this is, the data you generate is of no use if you haven't stick to these four things, like data integrity, data security, privacy, and quality. Data integrity is really necessary. I mean, you cannot just post any data. You need to substantiate your data, endorse your data, saying that it is, the integrity is all there. Like, there is no malpractice or a fraud or a misconduct or anything. So integrity you need to demonstrate, right? And then there is data security. You're dealing with people's date of birth. You're dealing with people's blood pressure data, whatever, so it's all personal health information. So you cannot leave it anywhere and everywhere just like that, randomly floating. So it needs to be in a secured environment. So it needs to be captured, processed, cleaned, reviewed, and analyzed in secure systems and secure environments by authorized people only. So that will govern the security aspect of it. Then the data privacy, of course, security and privacy overlap a little. So there are certain things that you cannot reveal to everybody, a larger public. So you need to maintain those confidentiality aspects as well, which GDPR, the regulations that I showed in the previous slide, out of those regulations, GDPR governs the privacy aspect of it. Security, integrity are covered by 21 CFR Part 11. Then comes data quality. I mean, of course, yes. I mean, you're storing it all in a secure environment. You're maintaining all the privacy aspects of it. It's all good, but data is not of quality. Then it is not telling story. After the analysis, you come up with your results and stuff. If the quality is not good, then there is no confidence in the results, right? So then it's not of any use, right? The whole purpose of conducting clinical trial itself will get lost if the quality is not good. So maintain a good focus on the quality as well right from the beginning, and start alerting the people that are associated with this process of generation of data. You need to alert them by looking at the trends, looking at anything that's going wrong. You need to alert them so that they can tweak the process and the quality again comes back on track, right? So this is a very much like a balancing act, maintaining the integrity and quality. So it's a nice balancing act. It is a little bit of science and a little bit of skill. So you need to be aware of that. And your decisions are only as good as your confidence in the data, right? Normally, like, again, you can take it back. I actually, like, when I explain things, I like to use day-to-day examples so that you can understand it better. Okay, you want to buy, again, I'll go back to shopping. Shopping gets into head properly, really easy, especially ladies. You want to buy a piece of cloth or a shoe, right, or a bag. You would have researched first. Who else has this kind of bag? Which company makes this kind of bag? How much it costs? So you have the data with you before you enter the shop. You have the information, you have data with you. So based on your research, what type of data you have generated, your decision is going to be good. Decision is which bag you will end up buying. That's your decision. If your data is not good, your bag is not going to be good because of your poor decision. You ended up making a bad decision. You ended up with a bad bag. You will back to the shop again quickly. So now extrapolate this scenario to the clinical trial. You're to have good confidence in, so that you can take proper decisions, is possible when you have good data, quality data that sticks to all these 4 points that are there on the slide. So Privacy and integrity is ensured by security though. So I just thought I'll put that, make it a little bit, little colorful so that, you know, they are all interweaved kind of bullet points. So you cannot isolate them. You cannot see them separately because they are all interlinked. Okay, so now coming to a little bit on the operation side. So the flow of events. So what actually happens in data management? What do people do here, right? So let's start with data entry at the top. So data gets entered, the data that is generated at site, right? That gets entered into a database. The database is a central place where all the data gets dumped by all people who have data with them, right? So data entry comes into the database, which would have been programmed by a database developers, right? So I've given the people associated next to the circles as well, right? So data entry operator enters the data into the database, or the data can come directly imported, like there is a handheld device or a BP machine or something. So from those individual machines, electronically data comes into this central database. That's also possible. Manually also data gets entered into the systems. So that's also possible. So then it gets monitored. So that's covered by covered previously by Chandana and Payal. So how the CRAs and the PMs go in there and do source data verification and monitor the data to make sure whatever is in the source documents is what has been transcribed into the electronic CRF, right? So that's the monitoring process. Then comes reviewing and cleaning, where data managers sit there and then they look at the data, they look at the protocol specifications, what the protocol is asking us to do. Is the data that has come in is conforming to the protocol requirements or not? If not, they will raise a query, ask the site questions, the questions get resolved, and the data gets cleaned. So that's whole data review and cleaning process, right? Then the data gets coded. So there is some specific requirements to code certain data, not all data. For example, adverse events and concomitant medications or medical history. There are certain specific medical dictionaries. Again, I will not go into those details as this is only introductory, only kind of a session. So using those dictionaries, you code the data that has come to you. Okay, so the two dictionaries that you use are MEDRA and WHODD, WHO Drug Dictionary. MEDRA you use for adverse events coding. WHODD is used for medications coding. So there are again dedicated medical coders who are trained in this type of activity. They code the data, and eventually the data goes to analysis. So it could be statisticians, SAS programmers. They run their program on the data that is now available, which is more cleaner, consistent, quality kind of data. They do their calculations and analysis and everything, and they come out with a story. The drug is statistically significant, blah, blah, blah. This is the effect. This is how the drug is showing effect or not showing effect, or whatever it is, the statistical significance and all that. So that is the cycle. So eventually after analysis, the data goes to the clinical study report writer, the medical writers. So the report gets written and that goes to the sponsor. So that completes the cycle. Now, initially in my second slide, I think in the history slide, I mentioned that I will show you the systems that are used in clinical data management. And these are some of the systems. And by no means, this is not a comprehensive list. There are so many systems out there, and I've tried to put in some of them here. So you can just put electronic data capture systems in Google and you will end up with hundreds of results, right? But what I would like to mention on this slide is on the left-hand side, it is more in-house databases. They are not EDC systems. They are not electronic. They are in-house databases, customized databases. On the right-hand side, they are electronic data capture systems, ECRFs, if you like. So more in the olden days, like 1990 or before, Oracle Clinical, ClinTrial, DMSys, Fosco, TrialDB, probably they were in use, out of which I have personally used ClinTrial and DMSys. They were in-house databases. Nothing was electronic. People could not see real-time data like sponsors, PIs. They did not have access. Only the data managers could tell them, okay, this is how it is looking. This is what the data is coming and stuff. And then shift to 1997, 1999 kind of time onwards, these electronic data capture systems started coming in. Paperless trials started. So Medidata Ray, Viva, Clinical One, IBM Clinical Development, Medrio. These are all the platforms using which we build clinical databases in these systems. So site staff gets access for these sites, for these systems. They log in like you log in any of the social media websites like LinkedIn, Facebook, whatever. You have your user ID, you have your password. You use that, get into the system like you get into Facebook or LinkedIn. Then you can enter data, you can review data, you can code data based on the level of access that is given to you. All this is covered by one person, who is called as database administrator or a super user. Different companies call them with different names. So if I am given the access of data entry only, then I can do only data entry. If I am given the review access only, then I can only look at the data, review the data. I cannot enter data. If it is a medical coder, he or she can only code the data using the dictionaries. So role-based access to the system. Right, that's what happens. And as soon as the data is in these systems, people can see it because it's all cloud-based technology. It's all online. So the sponsor who has read-only access to the system, as soon as the site has entered data, they can see. They log in and they can see how the data is coming along, right? So that is the advantage of the EDCs, and that's why there was a big shift late 1990s from paper to paperless studies. So I mentioned the phases of the study. So typically we divide the study into these three phases: startup, conduct, and closeout. So when the new protocol comes to us, when the new study starts in our department, the new protocol comes to us, we start with kickoff meeting. We look at things, how, what are we going to do, how we are going to do, what is the timelines, who all is involved in the team and all that. So and after that we typically do these activities. CRF gets designed, ECRF gets designed, database gets built with all the programming in it, edit check programming and the dynamics and the logics and everything. Data management plan gets written and data validation plan gets written. So these are the two major documents in data management, which will tell you how the data is handled by, in data management department. Of course, there are a lot of other DM documents that I haven't mentioned there, but these are two main ones, right? Then coming to the conduct. So database is built, documents are written, and the database goes live. That means data starts coming in, the real data from patients or subjects. So that gets reviewed and cleaned, and queries get raised and they resolved, and data gets going cleaner and cleaner and cleaner. Eventually, reconciliation activities, if necessary, they happen, because sometimes the data that is in the EDC that needs to be cross-checked with some other data elsewhere, then that is called as reconciliation. So someone else is maintaining safety database, for example, elsewhere, and the information in the safety database needs to be matched and cross-checked with the information that is present in the clinical database. So that is called as reconciliation. Then coming to closeout, once we are all fully sure that we have reviewed it, we have cleaned it, there is no other questions, we fully confident, okay, we are ready. Then closeout, what happens is by liaising with other functions like clinical operations, safety, stats, and all that, we start addressing the final issues, and eventually we take the database to the lock, which is called DBL, database lock. Once you lock the database, our job is done in data management department. So database gets locked, data gets exported finally to the stats and SAS programmers. 99% of our job is done. A little few things will be remaining after that, like archival and stuff, but that's for the purposes of this discussion, it's not required here. Okay, we looked at all these. Now, what is the significance? Just to touch quickly on the significance of it. I will go a little quicker. Think over time. I would like to emphasize on the clinical aspect of the entire word, clinical data management. So data management, as I said, happens in a lot of other sectors also. Here, I would like to emphasize a little bit more on clinical, because people need to have some sort of clinical knowledge to be able to efficiently manage data, right? Just to give some random, just a quick example, 11 million medical data security breaches happened in 2015 in United States. And you can imagine medical data security breach, what it can lead to in terms of claiming insurance, for example, or there can be big impact of such breaches. So you need to know what type of data you are handling, and using that knowledge, you can maintain the confidentiality and stuff, security and stuff. So it becomes really important there. Time. Save time by getting content and review right at crucial stages. So your clinical knowledge enhances the review process, and content wise also, what can you review and how quickly can you review. Obviously, if you are able to do it quickly, you're going to reduce cost. Okay, any cost that inadvertently gets added to the whole process of drug development, you are going to avoid it if you're going to do it quicker. Just some facts there on the right hand side. Average drug development cost ranges anywhere between 1.3 to 2.6 billion. So you want to bring a new tablet into the market, from the initial researches to the time it comes to it hits the shelves in the medical shops, it is something like 1.3 to 2.6 billion. It's a lot. And then imagine if you're conducting a clinical trial and your decision making is delayed, and you're submitting your data to the regulatory agencies for licensing to market the drug. You wanted to apply for licensing today, but if for some reason you couldn't do it today, you went to tomorrow. The cost of 1 day delay, it's about 600,000 to 5 million. Now put that into the context and see how significant it is to clean the data on time. If someone didn't perform their activity on time, every day you are adding around 600,000 US dollars to 5 million US dollars based on what type of drug it is. Continuing the significance, CDM is expected to address the growing complexity and volume of data in the modern clinical trials. So traditionally what we've been doing was different, but what we will be doing in future is going to be different. Volume is going to increase significantly because data will come from all different directions, from wearables, from all sort of machines, and along with the manually entered data. So data will be coming to you from literally every directions, right? Amount of data is going to increase, and also the complexity, because it comes from varied sources. Every source has its own complexity. So complexity will only get increased. CDM is expected to play a role in detecting and avoiding frauds. We should keep it at the top of our list and close to our hearts, because we cannot afford doing things in shortcuts and in wrong ways. And especially we in health science sector, we should be focusing on ethics more than anyone else. We rather fail with honor than succeed by fraud. Not my quote, but I just thought I will have it there just to put things in context. So continuing from there, connected issue, data integrity associated warning letters issued by FDA by country is in that table. You can see for yourself. During those years, what was WLC's warning letters issued by FDA, and those warning letters were obviously issued for certain reasons. They audited and they found something wrong or some malpractice, some fraud or whatever. Those many warning letters were issued. So what I'm trying to tell you is things happen, errors happen, because I think Chandana mentioned in her opening session, like we are into research. In research, nothing happens as per plan. There will be challenges, and you will have to go on exploring and coming up with answers for the challenges. So nothing will go as planned. Otherwise, it will not be called as research if everything goes as planned. But having said that, no shortcuts, and we should keep records and proper documents for everything, whatever is being done. Last slide. My take-home message for you all is we are all responsible for managing subject data generated in the trials, which is crucial. Better data management will lead to smarter real-time decision making during trials, not after, long time after the trial is finished. So what is more important is during the trial itself, because sometimes trials are 2 years, 3 years, 5 years. During the trial itself, you need to go on modifying your decision making. Sometimes you thought initially something, but then you need to quickly change and do something else. So keep on, improving your things. So that needs decision making in real time. So that is possible with effective data management. Then CDM contributes to the drug development process by using technology in combination with clinical knowledge. Like I mentioned in previous slides, how clinical knowledge is important. Of course, there is technology also. What I mentioned in the history slide, artificial intelligence, machine learning, real world data, this is all new technology that we are going to see, or we have started seeing already, in fact. Combine that technology with clinical knowledge, you are going to contribute significantly to the whole process of drug development, right? So the world, just to give you things a little bit more detail about enormity of data, world's data keeps doubling every 2 years, just as an estimation. It's not 100%, but just an estimation. Global wearable device traffic is expected to have surpassed 40 zettabytes at the end of 2020. We don't know what it is, 2022, maybe someone has more recent figures, but it is expected to have surpassed 40 zettabytes. And 1 zettabyte is there, you do the calculation yourself in your mind, is 1 trillion gigabytes. So 40 zettabytes, that was in 2020. We are in 2022. I don't know what other new term has come to explain that. And the way data was traditionally handled will definitely change in the coming days, of course, and with more advanced methods, which will make it even more quicker, cleaner, and more consistent. Last thought, I would like to leave this thought with you. Without data, you are just another person with an opinion. You can't say anything meaningfully or that makes sense if it cannot be interpreted in a proper way, in the right manner. Thank you, everybody.
Thank you, Ullas, for the detailed session on clinical data management. So we got to hear about evolution of data management through the years, the Alcoa CDM principles, the framework for data integrity, the core concepts of data management, and the importance of data privacy and security. We also got to hear about the various CDMS systems, wherein we had a question previously as to how equipped we are in India to handle the new platforms. So thank you so much, Ullas, for letting the audience know as to what data management is. So we already had certain questions coming up. So in that we have, what conditions the database may be unlocked, and does the data manager has the authority to do it?
Yes, database can be unlocked during certain situations. But of course, I mean, that question will come only after locking, and the locking happens only after we would have thought all the data is cleaner and neater, and we would have taken care of everything. So mostly it will, if at all it happens, it is at the end of it, in case we would have missed anything to check or some, yeah. But yes, unlocking happens. But whenever it happens, what is more important is to document the reason for unlocking, correctly documenting what corrections we are doing after unlocking, and the date and time of relocking.
Thank you, Lars. So the next question is, just the way we have guidelines for the electronic documents, that is 21 CFR Part 11, on the same lines, do we have any guidelines for IRT, IVRS, and IWRS?
If I get this question correctly, there can be company-specific work instructions and SOPs to manage these processes. Regulatory guideline, I think 21 CFR Part 11 encompasses these aspects, because 21 CFR Part 11 is whatever you do using an computer. and IWRS happens using a computer. It uses a electronic system to allocate randomization number and stuff, right? So it still is kind of governed by 21 CFR Part 11, but specific steps of performing these activities, I think, in my opinion, is covered by the individual work instructions and SOPs of individual organizations.
Thank you. So what is hard lock and soft lock?
Okay, good question. Hard lock is when we lock it final lock. Everything gets locked, pretty much everything is done, and the data goes to statisticians, right? Soft lock, on the contrary, is study hasn't finished yet, but in the meantime, we want to look at something. So in the momentarily, we want to stop data coming in. We want to freeze the screens. We don't want the site to enter newer data. Whatever data come until now, say today I want to soft lock this database. So I want to have a look at all the data that has come in until today by the site, but and clean it, make sense out of it, and start, and give it to the statisticians to start analyzing it. But in the meantime, I don't want any newer data to come in the EDC. So we have frozen, momentary freezing of the database and the forms in specific. It can be specific forms in the database also, but it is not like a real hard lock that happens at the end, which means study would have finished. Soft lock can happen when the study is still undergoing. That's the difference.
Thank you, Lars. Any further questions on clinical data management? Okay, so I take it as a no. So thank you, Lars, for briefing the audience about what clinical data management is and what the core principles are. And I'm glad to say that we all are part of the clinical data management team, where we form an important bridge between our clients and the ClinOps team and the various other stakeholders. And we take in all the friction which happens through the clinical trials and processing the data.
Yeah.
So
yeah, feel proud if you're in CDM. That's what I would say.
Very much. Yes.
You can say I'm biased, but yes, still I'm going to say that.
Very true.
Thanks for the opportunity.
Thank you so much, Ullas. So as we explore the various departments within the clinical research industry, we now move on to pharmacovigilance, where we see a lot of inclination from the M-Pharma guys. So to give us an introduction about pharmacovigilance, we have Dr. Aniket Patil, who is the country safety lead for Pfizer India Drug Safety Unit, located in Mumbai. So he's Indian Pharmacovigilance Officer in charge, Indian QPPV for Pfizer India Drug Safety Unit. Coming over to his professional experience, he has around 13 years of the industry experience in the area of pharmacovigilance, including his previous position at Amgen. So he also held experience in clinical practice for 4 years. Coming over to his qualification, he's a registered medical practitioner and holds the medical degree of BAMS from Maharashtra University of Health Sciences. He also holds postgraduate diploma in clinical trial management from Billcare Clinical Research, Pune, and also completed MBA in healthcare services. Aniket, I welcome to today's session, and over to you.
Yeah, thank you, Samprita, for that kind introduction. I just want to check, hope you can hear me clearly.
Yes, Aniket, you're visible and we can hear you clear.
Okay, so I'm just sharing my screen. One of you can please let me know once you can see the screen.
Yes, clear.
Okay, thank you. So first of all, thanks to all the organizing committee, ICR and Calais University colleagues, and all the team members for giving us the opportunity to speak in this very important workshop. So without further wasting time, I would straight away come to the topic. So just before I begin, a disclaimer: the comments, views, opinions expressed in this presentation are of mine. These does not reflect any opinions of my organizations to which I'm affiliated with. Okay, what we will cover in today's session is the pharmacovigilance terminology and somewhat about the history, why we need pharmacovigilance, what are the aims of pharmacovigilance, and you will also see some acronyms. So very important is pharmacovigilance. It's PV or PHP also called. So aims of PV, pharmacovigilance key components, adverse event definitions and sources, responsibilities of pharmacovigilance professional, and somewhat key pointers about pharmacovigilance in India. Okay, so what is pharmacovigilance? If you see the word pharmacovigilance, it involves two words. Pharmakon, it's a Greek term which means drug, and vigilare, it's a Latin word. So that means to keep awake or alert or to keep a watch. So to keep a close watch on the drug, how it is behaving, it's basically the pharmacovigilance. What does the definition which is stated by WHO, World Health Organization? The science and activities relating to the detection, assessment, understanding and prevention of adverse effect or any other drug related problem is called as pharmacovigilance. Now here see again the very key important terms here: the detection, as you can see in this funnel, assessment and understanding, and prevention of the adverse effect. So combining the science and activities which relates all this is called as pharmacovigilance. So let's learn from the history. I know that Chandana has already stated the background about this thalidomide tragedy during her presentation. So I will not go into deep, but just I would like to have your attention into some of the key points with respect to pharmacovigilance. So as you can see here, after the administration of the drug, and actually have the launch of this thalidomide into the market, the first report of the adverse event, that is a birth defect phocomelia, which was reported, that was in 1959. And post that, there were several cases, around 5,000 to 7,000 infants, phocomelia cases were reported. So after that, in 1961, thalidomide was taken off the market. So from the from the year of launch and the time it was taken off the market, it hardly few years. And that is how pharmacovigilance is very, very important to identify those key aspects, and if there are any risk or any safety concerns while drug is actually given to the patients. So of course, the immediate impact of that was to have a very stricter and tougher, regulations and the rules in place in the developed countries. And also we see in India, we have a stricter regulations now for pharmacovigilance reporting requirements. Why do we need a pharmacovigilance? So we'll just see some of the aspects, some of the reason about need of the pharmacovigilance. Reason one is, of course, there is a humanitarian concern, because after drug, once it's entered into the clinical trial, so if we see the phases from preclinical up to the last phase, that is a phase 4. So before it actually goes into the market, so in between, of course, there is a research which is conducted, clinical trials are conducted, and during that time, of course, there is a limitation of safety data which evolves during that phase. And therefore pharmacovigilance is a science which is actually applicable throughout the life cycle of the drug. That means from the preclinical up to the drug whenever it is in the market. During the clinical trial, of course, we have the limitation like insufficient evidence, of course, in the animal experiments also, and in the first phases also. So due to all this limitation, a common or a layman person can think, okay, the drug is good in the clinical trial phase, but how effective, how safe it would be when it comes to the market and when I consume it. So that's a typical humanitarian concern is there. But pharmacovigilance is like a science which actually evaluates the risk of the product identifies that throughout the life cycle. Reason 2, medicines are supposed to save lives. So dying from a disease may be inevitable, but dying from a medicine is unacceptable. That is what WHO has said. So as you can see in this diagram here, what is acceptable? So yes, high benefits of the drug, but if risk is more, the benefits are high and risk is low, then it is acceptable. But if a drug has higher risk and benefits are low, then that is unacceptable. So that is how pharmacovigilance is a very, very important science to identify this. And the last but not the least is the reason is ADRs are expensive. So if we see the cost to the country basically in terms of the medicines and managing the adverse event reactions of the drug, so it's really expensive, and that's why we see there are very, very stricter regulations in terms of the compensation. As some of you also may know that in India also we do have a very strict compensation requirements for the clinical trial related adverse event or the serious adverse event which are reported into. Now we'll just see some of the aims or the key aims of the pharmacovigilance. First and foremost, to improve patient care and safety, to improve public health in all and safety, to contribute assessment of benefit and risk medicine throughout the drug life cycle, as I mentioned in my previous slide also, to identify previous unrecognized adverse effect or change in their patterns, which is very, very important aim of PV, to promote understanding, clinical training, and effective communication to the SCPs, that is the healthcare professionals. To promote effective communication to the public in all is also one of the very important aim. And lastly, to promote the rational and safe use of the medicine, which is very, very important. So all these are the aims of pharmacovigilance. Now we will see some of the key components of pharmacovigilance. Now, if you read, through, there are many, many components, but here I have just presented 4 key components from a marketing authorization holder, MH, it's acronym, marketing authorization holder's perspective. So what are these 4 key components? Adverse event case management, including the expedited reporting, which is a very, very key concept and a component in PV. Aggregate reporting, which is again one of the very important aspect. As we know, individual case safety reporting, identification and submission that of that into the timelines is important, but there are regulations which also required aggregate reports of specific type which are to be reported to the health authority within a specified timelines. So again, aggregate report becomes a very, very essential component of PV. Signal detection, again a very key component, and this is identified again, as I said, throughout the life cycle of the drug. Whatever are the adverse event reported in individual case safety report format in the global safety database, these are analyzed and signals are evaluated, and then further actions are ensured. Last but not the least, risk management. Again, for all products, if the risk is identified, how to mitigate that risk, how to manage that risk. It may be, globally we have to manage, or it may be country specific, some of the risk minimization measures that a company has to take. So all these 4 are very, very essential components of pharmacovigilance from a MH, or I would say from a pharmaceutical company perspective. Okay, we will see now some of the key terms, and that is the adverse event. And within adverse event, we will see what is AE, that is adverse event, versus what is adverse event reaction, and what is serious adverse event. As you can see here, adverse event is defined as any untoward medical occurrence in a patient or a clinical investigational subject administered a pharmaceutical product, and which does not necessarily have a causal relationship with this treatment. I have underlined a statement here, which does not necessarily have a causal relationship with this treatment. We'll just see the difference between AE and ADR just in a moment, but we will see, the next definition. What is mean by ADR? ADR is defined as a response to a drug that is noxious or and unintended, and occurs at a doses normally used in human for the prophylaxis, diagnosis, and treatment of a disease, or for the modification of the psychological function, physiological function. As causal relationship between the drug and the occurrence is suspect. Now you can see here the diagram. So, and I will also correlate this with the underlined text here. So adverse event, as you can see, which may or may not have the causal relationship with the product or the treatment, and adverse drug reaction, which always have a or a suspect or a causal relationship with the product which is administered. If I have to give you an example, so let's say if patient is taking drug A for indication fever, and he develops a fracture, and as per the physician, there is no causal relationship between the fracture and the treatment, that is drug A which the patient has taken. So it's a typical adverse event. But let's see another example wherein the patient is taking drug B for fever, and then he also had nausea, and his physician assesses that it is related to the particular product. Then since it is related, it has a causal relationship, and that's why this particular event is called as ADR. So as you can see here, all ADRs are of course adverse event, but within adverse event which are having the causal relationship with the product are called as ADR. Serious adverse event, so it is defined as any untoward medical occurrence that at any dose which results either into death or is a life-threatening condition which requires inpatient hospitalization or prolongation of existing hospitalization, or it result in persistent or significant disability or incapability, or also is a congenital anomaly or birth defect. Like we see the example in the thalidomide, phocomelia is the birth defect. And from MH perspective, some of the important medical events which can classify into this particular categories are also conservatively categorized as serious events into the global safety database. Moving on to the adverse event key elements. So 4 elements are very important when we see adverse event definition, or basically if I have to put it in another way, to determine the valid adverse event, these are the 4 important elements which a PV professional evaluates before assessing that particular event. What are those? Patient, reporter. Again here reporter, it can be healthcare professionals, anyone. It can be a caretaker also, or sometimes patient himself or herself can also report the adverse event. So second is the reporter, which is important element. Event, after that product, what is the event that patient has experienced? So event is the third important element, and the product, of course, which is very, very essential element. So all these are the 4 important elements which makes a particular individual case safety report or adverse event to have that as a valid criteria. Now we have seen some of the key terminologies like A's, ADRs, SAEs, what it means. Now this slide depicts the various sources of adverse event or the serious adverse event which are reported towards the companies. So couple of them we have already seen, that is caretakers and relatives, healthcare professionals. But as you can see, internet, other media is also nowadays very, very important source from which there are many adverse events which are reported from this medium. Literature, patient of course reports adverse event. Clinical trials, licensing partners, medicine agencies, patient insurance, lawyers, they also report adverse event. Other pharmaceutical companies, as a best practices of any pharmaceutical companies, they do report the adverse event to the respective company. Company staff, now company staff also have the obligation to ensure the adverse event reporting within a stringent timeline, so that further the drug safety profile can be assessed in the required standards. So these are, as I mentioned, these are the sources of getting adverse event reported at the PV department. Now we will just see the case management workflow here. I'm not going to go into detail since the objective is to introduce. I will just focus on some of the key aspects here. So whenever a case is received to the pharmacovigilance department or a pharmacovigilance professional, whomsoever are involved into the case management aspect or the department. So this is a typical, I would say, flow of a case into that department. Now, company to company, again, it may vary and it depends on their particular company's SOP. But just to take you through the key steps here. So whenever the adverse event is reported through all those sources, so the A or the adverse event gets received at the case receipt department. So it's kind of called as a case receipt, but basically it receives to the PV department of the company. After receipt of the cases, of course, the particular professional or the person has to evaluate to go to the next stage, that is the triaging. In triage, of course, there are many aspects which are taken care, like if the case is a duplicate case, or if it's a follow-up case, or if it's a new case. And then further, after triaging, it gets processed into the global safety database of the company. After triaging, the next step is the event assessment. Now, in the event assessment, precisely the causality assessment is evaluated, and causality assessment again from two perspectives. One is what is the reporter causality and what is the company causality. So, depending on the process of each company, the causality by the company is assessed. There are also different aspect and criterias of evaluating the causality assessment, so which are applied while doing these steps. Then processing the follow-up information. So for already initial reported case, if there is any additional information which is either reported by the reporter or it is obtained by the company, then the follow-up information is also gets processed into that particular case in the database. And once the complete version of the case get processed, the next steps occur. Now, in between, we can also see here the small step, but very key step, that is the risk and benefit analysis. Now, this depends on the overall data which is received into the whole database for that product, and the particular subject matter expert, or typically known as SMEs into the risk management aspect, they evaluate this. But for a single case report, before the last step, that is the regulatory submission, or I would say a distribution of that particular single case to the different stakeholders. It may be regulatory for a clinical trial case, it may be the investigator, ethics committee, and the health authority. So before the distribution, the final quality check is done, and then the case is distributed to the respective stakeholders depending on the type of case. So that is how a typical case management workflow looks like. And now this is I think very important for all of you. What are the common responsibilities of a pharmacovigilance professional? So of course, the timely detection and notification is a very key here. Appropriate assessment of the entry of the data into the global safety database, and doing that, prioritization and processing of the data is very, very important. So that's another aspect of the responsibilities of a PV professional. Expedited and periodic reporting towards the health authorities, again a very, very important aspect and a regulatory compliance driven activity of a PV professional. Maintenance of PV system in compliance with the local regulation. Now the developed markets may have a very, very stricter regulation, but sometimes country specific local regulatory requirements are also we have to ensure. For example, if I have to tell you here, so in all countries it may be the 15 calendar days, which is the common accepted or required timeline for reporting. But in India, for clinical trial, serious adverse event reporting, we have 14 calendar days of timeline. So that's how local regulation and ensuring the compliance to local regulation is a very, very important aspect for a PV professional. Evaluate risk mitigation and benefits, again a very key aspect in the PV professional responsibilities. Monitor the safety profile of products. Now both this, that is the risk mitigation and monitoring the overall safety profile. So again, depending on the SME within that area, within PV as well, those particular department, the responsibilities are delegated, and that is how those are only the department who ensures and evaluate both this aspect. And lastly, provide applicable stakeholders with important, up-to-date, accurate and timely safety information. So as I said, within the stakeholders, it may be regulatory authority, it may be investigator site, it may be ethics committee, or it may be the head of institution. As we know, in India, we have to report to head of institution as well. So to all the stakeholders, the PV professional has to ensure that the individual case safety report, or if required, the aggregate report has to be reported, in a timely manner. Okay, so that was, I would say, in terms of common PV understanding some of the concept, but we should also know what is the pharmacovigilance aspect in India. So I will cover some of this aspect, in the interest of time. I will just go into the, I would say, key points. So first, we will see some of the background about acts, rules, and guidance, which gives the understanding about what are the pharmacovigilance requirement in India. And as you must have seen in earlier presentation, act or the rules is something, the local regulation, which is a mandatory requirement, and guidance is again, it's yes, good to follow. And every MH of course has to ensure that guidance document are also followed, and conservatively, yes, whatever maximum we have to follow in the guidance requirement, we have to follow that. But as always, acts and rule, mandatory or the legal requirement. In the previous presentation, we have already seen that Drug and Cosmetic Act, which is a law of the land with respect to the clinical research, and even for PV, the aspect which are mentioned, we have to follow that in India. And CDSO is our health authority, and DCGI, who is the head of CDSO, that is the health authority, and it is located in New Delhi. So in terms of acts and rule, new clinical trial rule, which is the latest, I would say, amendment. So and this is again with respect to the PVM. I have mentioned here a few points. So you can see the latest requirements in the NDCT rules published in 2019. Of course, Drug and Cosmetic Act as a whole, amendment, and some of the important schedules like Schedule M, or some of the important GSR which are part of this Drug and Cosmetic Act, like for example 287E. These are something very, very important than the medical device rule. This gives the guidance about what are the pharmacovigilance requirements in India. In terms of guidance document, of course, pharmacovigilance guidance document for the marketing authorization holders, which is published by PVPI, it gives a complete guidance about not only the common terms, but in India, what is the requirement. Guidance document for industry for PV requirements for biological products like vaccine and other biological products. So this is again one more guidance document is available from the health authority. So moving into the next, I would say, precise element from all these acts and rule as well as the guidance document, I will cover that in next couple of slides. Pharmacovigilance officer in charge, acronym as you can see, PVOI, at a marketing authorization holders, we stand for MH. So this is a requirement of Drug and Cosmetic Act, and as you can see here in the snapshot, gazetted notification which is part of TNC Act, which is published, and this entails all this requirement that who can be the PVI and what are his or her responsibilities. So qualified and trained personnel should be authorized by the company management as a PVOI with responsibilities for dealing PV activities at marketing authorization holders, that is the company. Medical officer or a pharmacist trained in the collection of and analysis of ADR reports can be a PVOI in a company. PVOI should reside in India. Roles and responsibilities should be clearly documented, and duty in charge to work in absence of PVI is also, it can be documented. Now, the last three, as you can see here, are given in the guidance document, in the PVPI release guidance document. It is available in the public domain. Of course, you can also you all can Google it and see that more on this. So this is one important requirement. Moving on to the next, a few important requirements. So I will cover that in the next two slides in two aspects. One is what are the regulatory requirements for pharmacovigilance while considering from post-marketing, and especially about the reporting of adverse event towards the health authority. And in the next, I will cover about the clinical trial adverse event reporting requirement. So in this post-marketing pharmacovigilance reporting requirement by a marketing authorization holder, for ICSRs, all serious ADRs are required to be reported. That is what is the Schedule M, which talks about. And so now again, just to let you know, ICSR, it's again an acronym, a common use acronym in PV sector. It's individual case safety report. That is a single adverse event report basically. PSUR, it's a periodic safety update report. So for PSUR reporting also in India, we have a strict requirement. So it tells about after the drug gets the marketing authorization approval, then PSURs are required to be reported 6 months for the first 2 years after approval, and for the subsequent 2 years, these are required to be reported on an annual basis. So this is a typical, I would say, reporting requirement which all the MH must abide with. And beyond this particular period, of course, if drug regulatory authority of India requests to submit any additional PSURs, then of course that company has to submit that. Moving on to the subsequent slide about individual case safety reporting. Again, I will just sort of touch onto the very key aspect, because this is the requirement in India. You all must be aware about this important elements. So for reporting of individual case safety reports to our health authority, that is to DCG office, what is required? What to report? As I mentioned in the previous slide, all ADRs are required to be reported, and all serious unexpected ADRs are also important for submission towards the DCG office. Who should report? Sponsor, marketing authorization holder must report all these ADRs for a marketed product. Whom to report? DCG office. It's a PV division in DCG office, and they have their dedicated mail ID also. So we have to report to them. What's the time frame? 15 days after the initial receipt of the information about the adverse event to the company. Typical format which is accepted for the post-marketed adverse event report, that's the COMS form. Now we also have the PVPI, and I will come to what is PVPI and what's the role in the next slides. So so to PVPI office, again, as per the guidance document, company has to report. So what to report? What is the timeline? As you can see here, spontaneous adverse event or the ADRs are required to be submitted to NCC PVPI office. Who should report? Sponsor, of course, a marketing authorization holder who has got the permission, they can report it. Whom to report? NCC PVPI office. Again, there is official address and the email ID which is available in the public domain to whom they have, the company has to report. What's the time frame? For serious cases, within 15 calendar days of the receipt, and for non-serious, it's within 30 calendar days of the receipt. Typical format, it's a E2B XML format which is accepted by the PVPI office, and this office plays an important role to also feed this particular data into WHO database, that is through Vigiflow. So, and that's why this is a typical format, E2B XML format, which they accept. Now, moving to the next topic about clinical trial pharmacovigilance reporting. Again, I will only focus here about the individual case safety reporting in the clinical trial. So we will just see and glance about what are the cases, type of cases, stakeholders in this table. So what to report? All serious adverse event during clinical trials has to be reported. Who should report? investigator, sponsor, ethics committee, they have to report it to the DCG office. Whom to report and what is the time frame? And this is very important. Investigator has to report it to DCGI, ethics committee chairman, head of institution, and sponsor within 24 hours of the occurrence of the SE. And due analysis report, of course, investigator can submit within 14 days. Sponsor's timeline, sponsor has to also submit the report of the analysis within 14 calendar days to the stakeholders, that is health authority, that is DCGI, ethics committee chairman, and head of institution. Ethics committee also has the responsibility to submit to who? To DCGI within 30 calendar days of the receipt of the SE to them and to give their opinion. Expert committee. So now DCG also has the expert committee, especially for the death cases or the related cases. This is the requirement. But again, it's up to the health authority who sort of ask the particular therapeutic expert which they have appointed to give them the opinion about a particular case. What is the format for reporting of the clinical trial cases? The format is given in the NTCT rule, that is table 5. And for a sponsor, of course, COMS is the accepted format for the due analysis report. And the very important is there is a pre-screening checklist for all the SE submission. So, as a sponsor, it is very important that we stick to this format. And if there is any element for which the information is not submitted by any company, then of course DCG office can issue the query to submit that particular aspect. And recently, of course, some of you may know that Sugam portal has been implemented for the module of pharmacovigilance reporting. Of course, Sugam portal was already there, but this PV module has been implemented, and now investigator, ECs, and of course even the sponsor companies has to report the clinical trial serious adverse event through this portal. There is a provision if any of course trial is not on the Sugam portal, then for offline CTs also there is a provision for reporting. Now just a few aspects in last couple of my slides. So PVPI, Pharmacovigilance Program of India. So what is that? So National Coordinating Center, which is situated in Delhi. So it has Pharmacovigilance Program of India, and this is I would say under the IPC and under the aegis of Drug Standard Control Organization, that is CDSU. So our health authority has come up with this program of PVPI, and this is the center of this is in the Ghaziabad. As for your information, I have just pasted here the org structure. As you can see at the top, our Ministry of Health is there. Then we have Indian Pharmacopoeia Commission and our Director of General of Health Services. And under that, we have our health authority, that is CDSCO, and under whom the PVPI. Here you can see it's a PVPI program, and the center under IPC. And within them, of course, PVPI operates, as per the guidance document and the actions which are, or the aims which are set by CDSCO. And under CDSCO, of course, there are state drug control organizations also. So that's how PVPI also plays a very key role in terms of assessing the safety in terms of the Indian population for the adverse event which are reported to PVPI office. As you can see here, a snapshot of the pharmacovigilance guidance document, which is issued by PVPI for all the MH, and this is also available in the public domain. Here you can see some of the key important modules which are there in this guidance document. I'm not going into detail, but just for your reference, pharmacovigilance system master file, which gives complete guidance about the PV system in an organization. Collecting, processing, reporting of individual case safety report. Again, this particular module's full understanding and full guidance about how to manage the ICSR, what are the reporting requirements, what are the key elements in the ICSRs. This provides the guidance. Preparation and submission of the PSUR reports. So what are the structure of the PSUR, what are the elements, so how to prepare, what's the timeline. Fourth is the quality management system in MH organization with respect to PV. Fifth module is about audits and inspection about PV system at MH organization, and the sixth is about the risk management plan. What is the guidance? So basically, all the 6 modules gives the detailed guidance for the MH in India. So if any of the small company has to also start up any PV department, this guidance document is very, very useful. I think in the introduction of PV, apart from India, we also must know at the international level, basically what are the guidelines and what is the requirement, how PV stands. But in the interest of time, I have just mentioned here what is the WHO role and what is this program about. So as you can see, WHO program for international drug monitoring has started in 1966. Their office is located in Uppsala, Sweden. It's a collaborating center basically for maintaining global adverse drug reaction database. It's a VG base, that is what they call. Roles of WHO collaborating centers are identifying early warning signals of serious adverse reaction to the medicines, evaluate the hazards, undertake the research into the mechanism of action to aid the development of safer and more effective medicine. Some of you must have also seen in news, WHO representative comes and sort of gives some of the keynotes, especially in terms of the pandemic. So that is how WHO role is very, very important role in terms of overall monitoring of global ADRs and what is the risk, if there is any risk. Okay, this is my last slide, and I think for all of you who have joined as a participants, I think it will be helpful for you. So from pharmacovigilance career perspective, what are the opportunities? So there are many opportunities. Few I have mentioned over here. So pharmaceutical companies, of course, which has a PV department, it called as local safety unit, local drug safety unit, DSU, or different terms. So within that department, of course, there is a very good opportunity for pharma graduates and also those who have had the study in the healthcare background to explore their career in PV or the pharmacovigilance. Biotech companies, clinical research organizations, very important, and many, I would say, special positions in which the one who are interested in PV can pursue their career. PV consultancy. So after having experience, of course, someone want to have the freelancing. So nowadays we are actually see that there are many consultants also who provide niche service in terms of some of the key components in the PV. So there are few specialized PV centers also, few organizations. So in that also people can have the opportunity. PVPI, which I mentioned, that is pharmacovigilance program of India, which is under the aegis of CDSU. They also have adverse event monitoring centers in the respective zone in our country. So they also require data entry operators, data entry specialists to feed those data and report it timely from the adverse event monitoring center to the PVP office. So again, it's a good opportunity for someone fresher also who want to pursue their career in PV. In terms of different job opportunities or the designation, there are many from the starting. If I have to mention, drug safety associate, drug safety assistant, safety expert. Once you grow into your career and have the experience, of course you can see and go through this profile: safety expert, safety manager, PV data manager, case safety reviewer, safety surveillance specialist. Now again, here few are the I would say relevant to the core component of the PV, like safety surveillance specialist, medical reviewer, aggregate report analyst, safety team lead. Someone after having a good experience may also want, can be a good team lead, so safety team lead. QPPV, qualified person in pharmacovigilance. Again, as you must have seen some of the legal requirement in our country, QPPV is one of the mandatory requirements. So if you want to pursue and go through the PV and have the career, of course you have a good opportunity to be a QPPV or a backup QPPV also in the beginning at any of the organization or the pharmaceutical company. these are just a quick references from where this topics of the presentation which I have shared now. So once again, thanks a lot for listening, and I'm happy to take if there are any questions on my presentation. Thank you.
Thank you, Aniket, for the detailed explanation so that the audience get to know as to what pharmacovigilance is. So the importance of it, the aim of pharmacovigilance, the case management workflow in terms of PV reporting, as well as the career perspectives within the pharmacovigilance team. Thank you for that wonderful session. We do have questions in the chat box, so let me go through them one by one. So So we have, I request audience to please refrain posting any questions which is out of context. As we know, there are various scales like UMC scale, Naranjo scale. So which causality assessment scale would you recommend?
Good question. So if this has been asked by a student, then see, it depends actually, because if you are working into a multinational company or any Indian company also, so the respective company, they have their own set of process or a standard operating procedures or a work instructions, and they have, of course, they also take essence from this various available scales or the examples which is mentioned about the causality assessment. So WHO scale or the Naranjo, the examples which are just stated. But typically, while working in an organization, the respective PV professional has to follow the company processes. That is very, very important.
Thank you, Aniket. So the next question is meta-analysis considered a part of PV?
Again, I would say yes, because it depends. Meta-analysis meaning what? Because it's just a term, but where we are using. Because if it's used in a whole clinical trial set, then again, what is that data wherein the analysis is required? If it's the PV data, then yes, definitely. And meta meaning the whole set of data which has to be analyzed. And coming to PV again, typically it's not used because see, as I also mentioned during the presentation, pharmacovigilance is a science or it's a process which is throughout the life cycle. So till the drug is in the market, we have to analyze that data. So that's all.
True, very true, Aniket. So any more questions? We do have certain case scenarios, but in the interest of time, probably I can take this question at later point of time. So Sagar, I have noted your question and we'll surely revisit it in the coming times. So if there are no further questions, I thank Aniket once again. And as per the agenda, we will now break for lunch. So we will have a quick 30-minute break and let all of us assemble back. And let us keep the time in mind, considering that we are slightly delayed. And I expect all of you to be back by 2:25. Thank you.
Thank you, Samprita.
Thank you, Aniket. Hello, IT team from Kerala. Am I audible?
Yes, Amrita, you are audible.
Yes, Sachin. I request you all to give the presenter, the panelist, access to Dr. Hetal Shah, who would be presenting the next session, please. It's Dr. Hetal Shah. I kindly request for the panelist access for her.
Hello, Sachin. I think
Yes.
Hetal probably joined in as Samprita.
Yeah, so she's joined in as a MV writer. So she's there. She has just messaged. If you can add her, that will be great.
Yeah, I think the IT guys couldn't figure out the name. They were looking for Hetal.
It is.
Okay.
Yeah, it is there. She has just messaged in the chat at
Chat
2:20.
box ನೋಡ್ರಿ. Okay, give access to that name. Done.
Did you get it?
Yeah.
Hetal, can you kindly check and confirm?
Yeah, hi, this is Hetal here. Thank you.
Yeah, we can hear you now. Yeah.
Hi, Ethel. Good
Hi.
afternoon.
Hi, Sachin. Good afternoon.
Hi, Hetal. Good afternoon.
Hi, Samprita. Good afternoon. Thank you for arranging everything.
We'll just give a few minutes until we have all the participants back.
Sure.
Thank you.
So if you're looking for people in the hall here, not everyone may be back, because they were participating from the central hall, but then now they may be participating from their respective laptops and mobiles.
Okay, okay. Even in terms of the participants here, sir, I see that there are only 70 of them. So they were 100 plus.
Yeah, 120 around. They were 120 to 130.
Yes. So probably we'll just give a few minutes to it so that we'll have more number joining in.
Yeah, maybe 1 or 2 minutes, and then we should start, I think, because we're running 35 minutes late.
Yes, yes, sure, Las.
Yeah, no problem. And we can cover up some of the time in the panel discussion.
Yes.
Yeah, that is meant for 1 hour. If
right.
we can cut down some of the time there, if there are not many questions, and then eventually we may go over by only about 10-15 minutes eventually at the end of the day.
We'll surely try our best. Yes.
Thanks a lot for coordinating everything as well. It's been really nice so far, and response from this side is really good.
Great to hear this, Ulas.
Yeah.
Thank you. So probably in the interest of time, we can get started. So welcome back, everyone. So we are here for the post-lunch sessions, and as we are hearing more and more, we are inching closer to better understand as to what clinical research industry is about, and who are the stakeholders, and what are the various departments who participate in the trials. So on the same lines, we have Dr. Hetal Shah, who would be presenting to us on the introduction to medical writing. So I did observe during the pre-session quiz, most of you all got the the answer to the medical writing question wrong. So we have Hetal Shah, who will present to us on medical writing. So to let you all know about her, she is a PhD pharmacologist with over 17 years of professional experience in the field of clinical research and medical writing. She's an independent medical writing consultant and trainer since 2003, the founder of Merit Healthcare Consulting, which is based out of Ahmedabad, that provides medical writing support for authoring complex regulatory documentation, scientific publications, and medical communications to multinational pharma and CROs. She's also the recipient of Women Entrepreneur in Pharma and Healthcare Award by AIC, LMCP Foundation in January 2019 for her entrepreneurial venture. She's gold medalist for manifold academic achievements, more than 70 scientific publications and presentations at national and international forums to her credit, an adept and experienced medical writing trainer and faculty for medical writing workshops and various scientific events. She's also an active member of ISCR, MW Council, and the Medical Writers Group of India. On the personal front, I'm happy to share that Hetal is a trained Bharatanatyam dancer performing multiple Indian dance forms, a yoga fanatic, and a culinary enthusiast in her free time. So we do have a multifaceted person. A warm welcome, Hetal, and over to you.
Thank you, Samprita, for the kind introduction, and good afternoon, everyone. At the outset, I would like to thank ISCR and KLE College for putting up this amazing program together, especially for our student community. And I thank each one of the organizing team to have this opportunity for all of us. I'll start sharing my screen at this time, Samprita, and please let me know once you are able to see.
Yes, Hetal.
Okay, great. So Samprita mentioned most of you got the one question on medical writing wrong in the quiz. And so probably this is the kind of expression quite a few of you might be having if asked about medical writing right now. So my intention over the another 20 to 25 minutes would be to make sure this expression changes to a slightly pleasant one. On that note, I welcome you to this session on medical writing, and my intention over the next few slides is to give you a bird's eye view on what medical writing is within the clinical research industry, what and why of medical writing, why the need and demand, what are the future prospects, and if at all, if it interests any of you, how you can get started. There are not a lot of technical details involved here, so that I do not really dose you off the post-lunch session. However, if you need any more information, feel free to post your comments or your questions in the chat box anytime. So while we get started, let me hear out from each one of you. And at this point, I request all of you to go to your chat boxes and let me know in one word or maybe in a phrase, why do you think it is important to write in the field of science? I would take a moment here and I will try to look at the chat box if I can. But if not, Samprita, I would request you to help me out to read out if we have any responses.
Yes, I'm keeping a watch on this, Hetal.
Great.
I'll help you on it.
Okay, so I presume most of us is the student community, something I really closely relate to, and I'm sure there would be much of the information coming in. So I do read it out here. It says obviously to pass information and knowledge. Yes, of course. Any further feedback?
Any feedback, guys? The chat box is open for you all to comment.
Awareness, absolutely. Why do you write? If you are a B.Pharm student or you are a postgraduate scholar or you are a PhD researcher, I'm sure you would have much more to contribute to this one. To provide information regarding your work, right? To share research related data, to document, absolutely, in Word to document. So what we are doing in the purpose of all this is we are actually either trying to explain something, publicize your results, inform something, share, ask for approval, tell, or either document. And the key word there is communicate. So medical writing is all about communicating. Communicating the scientific and clinical data to a range of audiences in a wide variety of formats. So these are the pillars of medical writing: the data, communication, audiences, and your format. The one that probably some of you got wrong was this. So medical writing has become a more commonly used term, perhaps, but it does cover on to say scientific writing or research writing, regulatory writing, publication writing, or quite a few other terms. However, what it relates to is at the end of the day, in terms of functionality and operations, it is usually referred as medical writing in the clinical research industry. Now, you have been hearing some of, about some of these domains since the morning, and I thought I'll just give you a snapshot of how the, a transverse section, if taken across a clinical trial conduct look like. So there is clinical operations, which you probably heard this morning, and we have various other domains on the other side of the game, which is data science, safety, and regulatory, where all the other domains play a role, from which medical writing is one of them. And as a function, medical writing sits very well across all the other collaborative efforts also. And it's like needed that medical writing team does not work in silo. They have to be very well connected and work with each element of the clinical trial, domains or functionalities to make sure their jobs are delivered. So in a way, you as a function, medical writing is very versatile and always needs to be, in discussion or communication with all the stakeholders. Now, why medical writing? Three major, I would say, recipients or entities that are influenced or are beneficial, and these are, of course, the part of our industry stakeholders also. So, first and foremost, from pharma and healthcare industry point of view, medical writing as a function is important to ensure that there is a proper alliance for approvals, communication, and continuation of development of new drug products. For medical professionals, it is a tool to enhance or communicate their knowledge, also propagate new ideas, as well as understand about therapeutics much more. And obviously the core to the clinical research industry, that's our patients and general public. Medical writing is a sole source of health related information or getting new updated discussion about what is happening about new drugs, so on and so forth. And I think the scenario in COVID is such that has proved that transmitting or communicating this information has become of utmost important. So that precisely explains the function and importance of medical writing in the industry. The need and demand. So if we look at why medical writing has now emerged over the past 2 decades, if I can say, the reason is research for new drugs have always been continued, right? And with moving time, the submission requirements, the stringent regulatory requirements through documentation has led to a rise of a need to write documents, to prepare reports, or to submit data in a particular format. More so, we also have now data transparency initiatives wherein as a healthcare industry, you are supposed to provide your study results to the lay people in a public domain. So all these has together led to a rise of more reading, more readability among healthcare, and more requirements for sharing, either for approval or for the general public. And that's where the role of medical writing flourished, and pharmaceutical companies recognized that, this is a function that would be imperative to the drug discovery process. If you want to look at some commercials out here, so some projections, obviously a CRO market or the clinical research industry is always a booming one to be in. And the annual growth rates here are just to indicate that medical writing is still to reach probably its commercial maturity, and it's on the upscale out there. So probably we do expect this field to grow along with the industry as a whole. Why to India? That was a global, chart, but let's look at why even within India, we think this is a great, domain to be in, or offers like wonderful career options, because India has always, though this survey is slightly outdated, but it stands valid even at any given point in time, because India as a destination has always been looked upon for medical writing outsourcing, along with a lot of other things that come to us. And it's partly because of the elements that we already know, like a huge pool of skilled professionals, our competency in English as a non-native speaker, the different time zones come to our aid. And so what has happened is what started as a very simplified medical writing outsourcing jobs have now advanced to become, India to become a hub for even writing highly complex and high priority documents. So that kind of shows the growth of the domain within the industry. So having said that, how does the current industry environment look for medical writing? So we now have full-fledged developed teams within various stakeholders, like for pharmaceutical or medical device companies. They are either having individual writing teams, or they are embedded within what we call medical affairs or regulatory department as part of their clinical operations offering. Even pharmacovigilance has one segment of writers involved, and of course with marketing and product development. In the CRO, medical writing is an individual service line by itself now and offers a wonderful career opportunities. And so we have now medical and healthcare communication firms which exclusively deal only with medical writing related services. There are also developments of new players like the consulting firms and of course freelancers and publication houses that also get involved with this domain of the clinical research industry. So overall the industry environment is quite favorable and has been matured enough to develop this as a individual domain or a specialty within the operations. Just an example to show you some prototypes, like we have publication houses, Elsevier and alike. We have pharmaceutical companies whom we call sponsors. We also have CROs who are the service providers for medical writing. And we also have individual firms like Kynaps or Cactus, to just name an example, to show that these medical writing in itself has flourished as a single service line too. On that note, what happens in these companies or what happens into medical writing generally, I thought I'll show you a spectrum of what medical writers do across the drug R&D. So writing is involved and required as early as the preclinical development all the way till the post-approval obligations. Here, what I've tried to do is give you some examples of the prototypes that medical writing teams prepare. So the writing function is involved right from sometimes animal study protocols or analytical reports to the major component of medical writing in the industry, which is during the clinical development phase. This is the phase which involves preparation of clinical study protocols, study reports, your drug submission dossiers, package inserts, so on and so forth. And the writing function's responsibility does not end there. So we have teams who also work on the post-approval obligation requirements, like the PV writing teams would work on the safety reports. We also have publications being written based on the clinical study results available. And of course, lot of other things which do not fall into the regulatory requirement or the publication writing is now emerging as what we call the medical communications, which is like patient information, writing newsletter, visual aids, slide decks for education, so on and so forth. So kind of entire gamut. You may know all of this individually by different terms, like say PV or safety writing or publication writing or regulatory writing, but eventually it is all considered as medical writing in the industry. So what happens from a writer's point of view, when you move down from left to right on the spectrum, your requirements for writing change from being highly regulated and structured on the extreme left, which is during the drug development process, which is especially for regulatory submission most of the time. You move towards a more lightly regulated and creative content when it comes to post-approval obligations or for the purpose of public use. So this is just an area to show how the writing requirements as well as technique changes. These are just few examples if you have a question in mind, like what do medical writers do? So these are typical prototypes which regulatory writers would prepare or medical writers would be exposed to during the drug development process or during the clinical trial conduct. What is another major chunk of writing is scientific publications, which quite a few of us are exposed at the academic levels in our B.Pharms or M.Pharms or PhD levels. So these are various types of publications which also come under the purview of writing when you come into the industry. So for example, if you have a primary research data, obviously it goes as an original research manuscript. And there are also meta-analysis, literature reviews that are done as part of the process. So all this again falls under the writing team's responsibilities. A snapshot to give you how and what happens or what needs to be done for for doing it at a grassroots level, for example, if we say. So if we like take a transfer section again of what a document is or what medical writers do, basically it's about the art and science of the product on hand. So what we do is we combine the content into a particular structure which is guided by n number of guidelines and presented in a scientific manner. So this is put very simply the three vortices of medical writing deliverables: the content, the structure, and the presentation, keeping your purpose, your audience at the core. So if you fit any of your writing deliverable into this, you will have the guidance of what you need to do. Now, as such, the clinical research industry, I'm sure you have realized, is a highly regulated domain, and we have a lot of guidances and regulations to adhere to. So if you need to, just know or wish to know more, these are the regulatory bodies and the regulatory guidances available around what medical writers do, and we kind of follow them to the T when it comes to delivering the documents. So these are some guidances available for preparing regulatory documents. And so on. We have a nice array of guidelines also available for the research publications. So if you wish to read more, these are all in the public domain, free for access, and you can definitely go and have a look at them. Now coming to the industry roles for you. So since this session was more for the students, I thought I'll bring you to what is involved in medical writing, how is it, bifurcated, or what are the specialty subtypes, and similarly the roles have evolved in the industry. So while we still call ourselves medical writers or medical writing team, what also happens is there are specialist roles and career paths that you can follow. So we have a medical or the regulatory writing teams, which typically has associate medical writers, senior medical writers, principal medical writers, like types of role. Same for PV writing. I think Dr. Aniket already mentioned about it. We also have special teams who work only on publication writing. They are called the publication writers. And of course, the external medical communication writers are also there. What is also very beautiful about this domain is there's a lot of scope of having freelancing activities out here if you can develop yourself accordingly. So the role of freelance medical writer is quite established, and there are good future prospects for them also. And eventually it helps you to convert yourself into either a consultant or an entrepreneur, or put up your own writing firm. So on the left of the screen is individual contributors role. If you take up career in the industry, there are obviously also managerial and leadership positions available as you grow up the ladder. So if you think how one should get started, I think there are two things out here that you should consider if you are interested in exploring medical writing as a career option: the knowledge and the skills. Now, the knowledge would be one that you have through your formal qualifications, and of course, the domain knowledge. And skills will again be some which are required to get started, and some which can definitely be acquired. So let's look at it in brief. Some surveys again, which were done when the industry was being developed, and it still stands valid, is an education by degree. So obviously qualification in pharmacy, life science, or medical science is something that would definitely help you to get started with medical writing. And we see a mix of all levels of qualifications. So that is really not a hindrance, but obviously an advanced degree is definitely an added advantage for future prospects. That's when it comes to domain knowledge. So apart from what we do as part of our regular qualifications or degrees, particularly with clinical research is a domain where you need industry experience or training or gather some kind of domain knowledge, which is an added advantage. So overall, what is required as part of the domain knowledge is a good understanding of scientific, pharmaceutical, and medical terminologies, which would mean you should be able to get acquainted to any therapeutic area. You should, in general, be aware of the drug development process. It is important to know the national and international guidelines, as I mentioned, because the entire research industry is quite, highly regulated. So it's important to stay abreast with the guidelines and the regulations coming up. And obviously that would put you, that can be got through either on-job trainings or experience, or there are also various workshops being conducted by various bodies across the country, which can help you get some more domain knowledge. So in general, it would be a combination of your education as well as your domain knowledge. Now comes the skills. So as I said, there are some skills which you should be, which is kind of a requirement if you wish to explore this. So the ones on the left again are kind of, I would say, someone should go through or check for themselves whether we fit in, because unlike any other function, medical writing is one which is a skill-based job, and it involves a slight of, as I always say, art. It's a combination of art and science. So if you do not have the ability to read across a lot of literature, or if you're not very fond of writing per se, probably this would not interest you much to explore. So language competency is must, a flair for writing definitely, a lot of reading skills, active reading, literature hunting is what all the medical writers need to do at any point in time. So that is obviously something you should look for. Analytical and critical thinking. So you have to look for a lot of data through a lot of literature and connect the dots. So that analytical kind of thinking definitely helps. Obviously, communication skills, apart from writing, your verbal communication, your interaction with others would also matter. Computer skills must, because nowadays all our documents are being submitted or done only electronically. So it makes sense to have a very good hang of, computer skills for sure. And my favorite being eye for detail. So that is something what a medical writer need to, it's have or develop. So as writers, you would not just write or draft your content, you are also your first reviewer. So you should always have that editing and proofreading skills also within yourself. Apart from that, the ones that can either be acquired and obviously are good to have is statistics. A basic know-how definitely helps, though we do not want to, step over the statistician's role out here. But as writers, when you write and interpret the data, this really comes to your help. And in general, other skills which either you are exposed to or, you learn over time is time management, organization, good team skills, ethics, and obviously a correct attitude. So overall, this is a list of kind of acquired or required skills that you can, work on if you wish to, explore this domain. Now, if you feel there's, something like that is already lacking, but you need to develop on, those kind of, avenues are available. So you can work on building your skills in terms of language trainings. Even there are certification and workshops especially available for the subject of medical writing. So that can come to your help. Obviously, networking, being part of the community helps you to, know and grow along with the community. So that's in just about how you can get started and what, medical writers generally do. I would leave you with these details. Like, these are just a few organizations or some certifications, for example, available. However, this is not all. There is a lot out there on the internet and in the research space, which you can, help yourself with and upgrade your knowledge or, explore getting into one of the roles. Okay. So in the nutshell, I would say medical writing as a career, as a function, involves juggling a lot of elements of research, be it the scientific or the domain knowledge, having good writing skills, at the same time being up to date and, stuff like that. So it is not just one element that would get you through this function. It is a mix of lot of skills and knowledge that you need to juggle at any given point in time for being in this field. Right, so that's a brief about medical writing in general. These are your resources if you wish to, explore or learn or read out anything in detail particularly. And with that, I would thank you for your time, and I hope we have some time for the questionnaires. And this is just to show you how our day jobs or day lives look on every morning and evening, if I can say. So thank you for being with us today. And if there are any questions, we can take it at this time.
Thanks for the wonderful animations, Hetal. And yeah, that little girl with that surprised look, I'm sure everybody will have a smiling face, just like me, after hearing take you through the complete medical writing session. So I completely relate with you with the present picture being displayed as to how it feels like for a medical writer after they complete writing the details for a page. So I really thank Hetal for taking us through the nitty-gritty of medical writing. She did give a very a good, clear detailing as to what is the career prospects within medical writing, in-depth knowledge about the qualification and skills which is required for you all to be part of the medical writing team. Thank you so much, Hetal.
Thank you, Samprita.
So we look forward for your questions. The chat box is open. I request all of you to type in questions which are relevant to this session, please.
Samprita, until you get questions, I just thought I mentioned this. Definitely, I can resonate to the picture that was at the end. At the day end, that's how I will be for sure.
Very much.
Desk itself. That was a good one. But Hetal, thank you for the very nice, informative session. I had one question just to help the broader community here as well. You mentioned during your talk there would be national and international guidelines that medical writer would need to be familiar with. So where can the young aspirants or the students who would like to build career in this?
Okay, if I may just go back to my slides in that case. And so these are the ones that I have tried to enlist, which we usually follow for the regulatory prototypes or for the publication prototypes. So the attendees can definitely take a snapshot or the screenshot of this slide if they wish. So this is one for the regulatory documents, and this is the one for the research publications, which is like in one slide we have everything that a medical writer needs.
Okay, thanks for that. So that was more specific about national and international requirement in terms of format and template and things like that. But then I also had another question in my mind, more generic question, some other skills which we need to be careful of anyway while writing anything basically. So things like, for example, plagiarism, the general skills. So where can people get information about that, how to be careful that we are actually, because sometimes plagiarism we don't know, we may not realize that we are actually doing plagiarism.
Right. So the guidelines out here, all of them have elements of ethics in writing embedded. So there's no separate thing that you need to kind of go and refer to. For example, if I'm writing a scientific publication, my go-to guideline is ICMJ. And when I go to ICMJ and I try to follow that for my publication writing, I would be taking care of plagiarism or not ensuring there is any conflict of interest or acknowledging properly if there are any authorship issues I can resolve. So unlike any other domain, what happens with medical writings, like these are very document specific guidelines quite a few times. And then there are general guidelines which have come as part of the industry's initiatives. So if you see here, we have AMWA, European and American Writers Association. We have ISMAP, which is International Society Society for Medical Publication Professionals. We have COPE, and we have an N number of guidelines out here on Equator, which kind of relates to every single kind of document you wish to write, be it a case report or a systematic meta-analysis or an interventional clinical trial. There are absolutely amazing checklist as well as guidance is given, which would kind of cover you from all, making sure that you write an ethical and scientifically sound document.
Sure. Thanks for that, Hetal. Thank you.
Thank you, Hetal. So
I think I see some questions on the chat box. Would you want me to take that up, Samprita?
yes, so I am just letting you know on that, Hetal. So we have one of the team asking if you could share details on workshops regarding medical writing.
Right. So workshops regarding medical writings are, conducted by various bodies, including ISCR. So, for instance, if I have to quote, all upcoming workshops for ISCRs are always announced on LinkedIn and on the ISCR web page. There are also other bodies who conduct such workshops, and the information is either relayed through LinkedIn or through the medical writing community groups. So, it is a good idea to get into the community, into the groups, be more active on LinkedIn, follow the correct portals so that you are aware of when the workshops are coming up.
Okay, thank you for that, Hetal. Do we have any other questions related to the medical writing presentation? Do I take it as a no? Okay. So thank you, Hetal, once again for the amazing session on medical writing. I look forward to talking to you in the panel discussion.
Thank you, Samprita. Thanks.
Thank you. So moving on to our next session. So the next session is on Introduction to Biostatistics. Sorry. And we have Anadya Prakash Tripathi, who is a senior principal biostatistician with Abbott Pharmaceutical Division. He has more than 15 years of statistical experience in the pharma industry and public health research, and a special interest in design, analysis, and reporting of clinical trial research. So basically, he uses SAS programming, data standards, and SAS stats, SAS graphs, and SPSS as data analysis tool. So during his career, he served many pharma companies like Novartis, Eli Lilly, AstraZeneca, as well as service provider companies like TCS and Cognizant. He was also associated with George Institute for Global and Public Health Research. And at the moment, he is associated with Assam University, Silchar, as research scholar and doing his PhD on the topic sample size estimation in contraceptive clinical trial, an empirical approach. He also holds a master's degree in biostatistics from Banaras Hindu University and based out of Hyderabad. I welcome Anadya to the session. Anadya, are you able to hear us, and can you confirm the panel access? Hi, Anadya, are you able to hear us?
I think Samprita, again, the problem is with that name, the IT person cannot find anyone.
Okay, probably I will just message him.
Anatya, can you please ping in the chat so that the IT person can recognize you?
You
Yes.
can probably start your video also, Anadya, if you're hearing us, so that we know which name you are logged in under.
Right.
So the participants can't actually share the video.
Okay.
Probably, Anadya, you can just leave a message in the chat box and we'll pick it up from there.
Yeah, he's there now. Yeah.
No, that's, I think, Praveen.
That's Praveen, yes.
No, not that, the other person, Anadya. Did he put some, can you see something in the chat box?
No, no, Lars. Nadia, can you please leave a message in the chat box so that we get to recognize your ID, please? Are you able to hear us? Please excuse me for a moment. In the interest of time, probably until I get to reach out to Anadya, since we have Praveen available with us. Praveen, are we good to start with your session?
Sure. I hope I'm audible.
Great. Yes, Praveen.
Yes, you are, Praveen.
Great. Thank you.
Thank you for the support, Praveen. So, team, we now have Arokya Praveen with us, who will be taking us through the session on the latest developments in clinical research. So Praveen is a senior leader with a proven track record of leading multifunctional teams internationally in the CRO and healthcare industries, with a total of 17 years of experience. So as a solutions partner, he works with internal stakeholders across all levels in multinational organizations in identifying and executing improvement opportunities. In his current role, he is the Associate Director and Head of Global Project Management Team in India. So in this role, he holds the responsibility of managing a team of 375 individuals who work on tasks leading to site initiation, sample management, creation of investigator manuals, alerts management, and kit production. So Praveen has presented in India and global conferences over the past 10 years as a speaker, chair, and a debate panelist. So I extend a warm welcome to Praveen, and over to you.
Thank you, Samprita. I hope I'm
I'll be sharing the screen. Praveen, you want me to share the slides?
Yeah, that will be helpful if that's possible.
Yes, I'm just doing that. Let me know if my screen is visible.
Yes, it is.
Okay,
please put this
here
on
we go.
slideshow.
Yes.
Yeah. Thank you so much. So the topic that I'm going to talk about today is the latest trends in clinical research. So I personally have been a person who's seen the changes in clinical research over the last 17 years. So, a lot of people would relate to what I'm saying. So when we started working as an associate 17 years ago, it was more around paper trials and, things around paper trials and how you review data using paper trials. A lot was paper. And again, over the last 10 years, we've seen, a tremendous growth in the way we've become more electronic and digital. And, that's what we're going to talk about today in more detail. But as I start the slide, I would be touching certain items which are related to more of a regulatory aspect and the current trends in the world we live through a pandemic. And then we will be talking more about the, highly interesting topics like, AI, IoT, etc. So with that, I would just try to start the presentation here. If you can move on to the next slide, please, Samprita. So we are in a pandemic situation, and again, all regulators would like to see how we can, how they can help us. Sorry, I think we moved another slide.
Sorry.
Can you go to the previous one?
Yes.
Yeah,
Yes.
please. Sure, thank you. So we live in a pandemic world, and again, we need to have support from our regulators. And as everybody says, the pharma industry is highly regulated. And again, a lot of clinical research goes on for years before you could get something approved. But in a world where you wanted a vaccine quickly, and again, if the regulators are not going to support you with that, it's going to be a challenge, right? So basically, the regulators, both India, and other parts of the world, have actually stepped up to the game, and they have actually given us some support through different guidances. So if you looked at the first item that I've mentioned here, it's more around the electronic informed consent where available. This was a guidance that was provided by FDA in 2020, as soon as we were going through the first wave of the pandemic globally. So you can see, though it is a regulated environment, just to support the latest scenario or the latest happenings in the world, the regulators come forward to see how they can also help with something which can help us quickly. So electronic informed consent was not something which was a very regular thing in the past. But as we started moving towards more and more emergency authorization of COVID vaccines, we had to run quite a few trials, and probably electronic informed consent became very important. And these guidances definitely helped us there. And looking at the second point there, the smart wearables. So, we currently work in a virtual world, and a lot of patients were not able to go to the clinical sites to attend to the clinical visits that were part of their trials. So we had to provide smart wearables, and in terms of smart wearables and bringing them to more of a once in 2 visits or once in 3 visit kind of a schedule for the patients in a virtual world. The rest of them were physical. So obviously we needed to do something around it. So the emergency use authorization for COVID-19 pandemic and the smart wearables definitely helped the overall community to get faster and get a lot of approvals done faster for the COVID-19 trials. Remote data collection and monitoring, you could see, there were several opportunities made available. And again, these definitely helped us in terms of getting the vaccine trials faster. And FDA also created a coronavirus treatment accelerated program for SARS-CoV-2 therapies, and leading to fast track review of protocols, sometimes within a 24-hour cycle. And if you see the number of studies that had gone through this 24-hour cycle for a review of protocols, I've listed them there: 50 plus antivirals, 60 plus cell and gene therapies, 130 plus immunomodulators, and 60 plus neutralizing antibodies. And FDA expects discontinuation of patients, delivery of IP delays or failures, IP administered at home, and affected COVID patients to deviate from protocol requirements, because certain protocols definitely ask the patient to go to the site. But in this current pandemic situation, this was not possible. And how do we document protocol deviation is something that the COVID-19 time, the regulators did come forward and help us with a proper guidance around that. Import of foreign vaccines, if approved for emergency use in other countries. So that was also something, you must have all seen it. We have some foreign approved vaccines which were actually imported into India. And, in the past, there used to be a lag time or a window time that used to be there before something was supposed to be brought into India for import. But in this case, since it was an emergency situation, obviously the DCGI as a regulator did help us and come forward in getting these vaccines into India, thereby supporting the overall population. And finally, but not the least in any terms, so the Internet of Things. So when I speak about smart wearables, so we needed to connect them through a virtual network and still be able to run trials and not compromise on any of the data integrity or data interoperability issues. And that is definitely something, the regulators did help us with the guidance. In June of 2019, though we were still getting into the pandemic, but the Internet of Things definitely helped us fast track some of the protocols and fast track some of the events that led to the COVID-19 vaccine emergency use. So, first and foremost, I do want to tell you in emerging trends that, the first and the pharma industry being more of a regulated industry, we needed the support from the regulators. And here you see, these are the latest trends that have been provided by the regulators in making us, more agile in terms of running all our clinical trials faster and getting them to emergency use faster. So this is, the latest emerging trends from the point of view of regulators. Let me move on to the next slide, please. Samprita, if we could move on to the next slide. Yes, sure. Thank you. So we speak a lot about artificial intelligence, reading human behavior, writing that back into an algorithm and, learning, multiple forms of data, understanding natural language and putting that back into a, algorithmic format so that it could be automated using the remote, the RPA systems. So how could we use artificial intelligence in clinical trials? And this is something over the past few years has been spoken in various conferences, various forums, and they're talking about how we can get faster clinical trials done. And I've just touched upon 3 areas where artificial intelligence is currently being used, and there are some case studies available on the internet if you would like to go and review that. So from a trial identification point of view, at least from the US point, US as a region, the electronic health records, they've been trying for a lot of years to see if that can be available as one source. So what do we call as electronic health records? So a patient goes into a clinical visit and the electronic format of the health record is maintained at the clinical site or the physician's office. So if I as a person go to different hospitals, can I still take my electronic health record when I visited another hospital? So this is something that they're trying to make it more universal, at least from an US point of view, and see if they can share that data along with a more of a blockchain kind of a ledger where it will be safe and secure while they transfer that data to the patient. So at different points of entry, they have provided a secure version of understanding the clinical data and trying to bring that back together in one place. And while I am going to another hospital, or maybe if I'm going to be part of a clinical trial at another site, how do I identify where do I have relevant clinical trials? So, there is more and more patients, especially in the oncology area, access to data where they would be able to find out, if there is a relevant clinical trial that's happening closer to them. So artificial intelligence as a boon is trying to help us, in terms of trying and the patients, in getting them the right clinical trial available for them. And the electronic health records we spoke, and again, these electronic health records have a massive amount of data that could be reviewed, and again, probably if you were going through a human review of all of this data and identifying a probable match of a clinical trial for a patient, this would take weeks. But again, using an algorithm or a natural language processing algorithm, you could just review it in minutes and identify a proper clinical trial for an individual. So these are benefits that we see, or latest emerging trends we see in terms of trial identification. You must have already heard while the Google CEO speaks in his yearly annual reviews, he keeps telling us that you can actually assess or identify a clinical trial using just Google Maps in the future. So this is something that is going to happen over a few years, because we also need to see how the data can be more regulated when he says that statement. So that is something that we are trying to closely watch as a industry to see how that can be possible. But you can see from a point of view of it takes weeks and months to identify a clinical trial, to a point of view of just click on an app and you're going to see, if I'm a patient, can I go and find something which is relevant for me closer to my house within 5 miles. So this is something where AI is going to play a major part, and we are all looking forward to it as we move forward into the future. Study enrollment, and again, if you find a clinical trial, and then what? So now there is a non-structured data signals review that can actually happen using AI as a methodology. And telehealth services, which a lot of us would have seen during the pandemic, where you could not go and visit a doctor for other ailments, but you were made available with telehealth services. So artificial intelligence is also trying to help in a lot of ways, where clinical trials could take advantage of the telehealth services, and data can still be transmitted back faster through different data sources, and finally into systems that are able to review data and get the trials moving faster with clinical decisions. The geosynchronization with trial location is something that I spoke when I mentioned about the example of Google. So they are actually trying to see if, based on your geolocation, if you can identify all the different trials that are happening closer to you and see if any trial will be useful for you or anybody else that's going through an indication which needs treatment. Bot support based on trial requirements and counseling. So like I mentioned, today's protocols are being amended just to mention that, like you don't have to go to the clinical site every visit, but you can go like maybe like once in 2 or once in 4 visits. And those amendments are also, required to be supported by some digital systems. So what has happened so far is we've seen a lot of bots or robots being created, which help this amendment requirements and also provide patient-centric counseling, as patients go through each of these situations more virtually. So patient care has always been more physical, and that is expected as a need in a lot of indications and therapeutic areas. But now they're trying to see how they can bring in augmented reality or any other such technical solutions that will help, counseling of patients more virtually. And coming to medication adherence, this is something that was a challenge, and there is many use cases available on the internet, and we can definitely see that digital phenotyping, face recognition, speech recognition, and smart medication bottles. Actually, the video is taken when a pill is being swallowed by a patient, and they actually can see if the pill was actually taken or the pill was spit out after maybe like 2 minutes, and using facial recognition and eye pupil movements and things like that. So what has happened in a lot of cases is the medical adherence during a clinical trial has increased by at least 65% in many places, just because of the use of artificial intelligence. And again, I just also wanted to mention, artificial intelligence just, does not stop with tools, systems, and, algorithms. Artificial intelligence also plays a vital role in trying to understand patient behavior. And, one particular trend that is currently, more of a smart trend, I would say, is basically around Internet of Behavior. So why do I use a particular tool? How many times do I touch that tool or my phone screen on a particular app icon? Or what makes me more adherent to a medicine? Am I happy when I take a medicine, or this medicine is not the one which is going to support me, but obviously I'm just taking it, or maybe I'm faking that I'm taking a medicine. So even though I might record a video of a medicine being taken, but the facial recognition or the pupil recognition or the retina recognition is actually something that is helping in terms of understanding how this medication is more of a behavior, that has been seen with patients. So I definitely see artificial intelligence as something that is not just going to stop with more of an algorithm or tools or system. It's also going to affect human behavior and see how human behavior can be more predictable while we run clinical trials, thereby increasing the number of trials that can be done and for a particular indication, and more medical solutions available for patients. So with that, I'll stop on this slide and let me move on to the next slide, please. Okay, so I am part of clinical labs, at least for the last 3 years, and I definitely wanted to bring you the latest trends that are happening here within the clinical laboratory space. So we have heard of blockchain, and a lot of people sorry about this. So in the past must have just thought the blockchain was just useful for financial industry, and it was just more of a how do I maintain a financial ledger. which actually transmits a lot of information to stock market and a lot of these regulated financial instruments. But later, though it was a little slow for the pharmaceutical industry, the blockchain also became more of a boon for the laboratory space, especially within clinical trials. So today, if a patient sample is being collected, at say at home or at a particular draw site, so that can actually have a crypto-controlled ledger receipt for the patient. So the patient actually just goes in, provides a draw sample, and he or she will get a receipt, more of a blockchain receipt, and then that is used to track how the sample travels from the lab to the point of delivery, to the point of analytics, and to the resulting. So a lot of this resulting is being used across many clinical trials, especially from, how can I use this, sample parameters for other studies that might be, a source, and this patient could become more of a in-sourced patient from one trial to another. So those kinds of information are also being analyzed, and then they are utilizing the same. Rewarding patients for improved biomarkers. So basically, the blockchain is also helping in terms of moving away from the needle. So, a biomarker, something like, if you have to have a better blood pressure, or if you have to have any other, physical parameters that would help the patient, have lesser needle draws. So the blockchain is actually helping provide that, using the behavior reviews of that individual, trying to make them more patient-centric, and thereby reducing the need for more needle draws. So the data is actually taken from different sources other than needle draws, just because the patient is being rewarded for an improved biomarker. If he or she is able to, say, for example, keep the blood pressure under control for a period of X days, then there is this message that comes in saying, maybe we don't need a needle draw right now. Your blood pressure is going to be reported as normal, and then we will be taking away the data required for the clinical trial. So this also increases the clinical trial participation by patients. And supply chain is managed by blockchain ledgers, helping sample tracking and expiry timelines management. Very important piece, especially in countries which are highly regulated while during the import of items. So if you look at, say, Latin America, Eastern Europe, Japan, there are countries where import licenses take a lot of time. And probably if you had a supply chain managed by blockchain ledgers, a lot of this information which restricted has now become more easier to track, more easier to comply with. And hence the supply chain moves faster. And this was more important during the COVID timelines. Basically, you needed to move the frozen samples, and before it loses stability, you will have to drop at the analysis site. So how do I do it if I have a very huge timeline for import license, especially when humans cannot go to the regulators and get that approval before you import a particular raw material? So these are things that were helped by blockchain over the pandemic time, and now it's going to be more centralized, and more and more decentralized trials are going to see how this blockchain as a technology could be used for supply chain movement, and also to track the samples through its life cycle. Home self collection, we spoke about it in part. So direct to patient kits is something that is a technology, I would say, or a particular practice that is being now more common and acceptable, and a lot of labs are providing that. And kits are delivered to investigators and or patients directly. The regional solutions may vary, just like I said, due to restrictions on legislations and regulations. But a lot of kits are now not directly sent to investigators. If you can send to a patient directly in countries, they're trying to send it directly. So thereby reducing the real-time visit of patients to investigator site. And also I mentioned about drop-off samples at local pharmacy store with a blockchain receipt. So we spoke about it. So it's becoming more and more easier and more patient-centric while we collect samples. And customers are increasingly adopting innovative DCT assay modalities. So nasal swab, saliva, and dried blood spot are more common. So earlier, you need to give us a sample. But now in the current pandemic situation, we would have seen some of the trials going on hold. But what has become a more lively trend is the dried blood spot. So you can actually send a dried blood spot. It's more around a tissue paper, and a blood spot is kind of made available on that, and it's dried and it's sent to the lab. And even using a dried blood spot, more and more assays could be reviewed, and hence helping the overall review of clinical assays. And expanding the menu of options for patients through customer and R&D efforts. So, they are trying to see how they can have more and more tests available. So earlier, as an organization, some organizations used to provide somewhere close to 4,500 tests made available, but most of these were required by the patient to actually go to the labs for a blood draw or any other sample draws. But now they're trying to see how they can have more and more lab tests available virtually without the use of a needle, and trying different technologies to try and see how this can be more useful for organizations and clinical trials. I'm sorry, can you just move that? Okay. The last one's about mobile labs. This is gaining a lot of traction and in the US and now in Europe. So the biopharmaceutical customers are able to actually take this forward and kind of fit for purpose built vehicles. Like you must have seen ATMs in a lot of Indian cities now, which is more mobile. So a laboratory is also now becoming more mobile, where blood samples and blood draws could be made available. An informatory message would be sent to patients, and probably if a mobile laboratory is in the locality, they could just go and provide the lab sample for that particular visit instead of having to travel a longer distance. Leverage in unique geographies. Some geographies actually do not have cities nearby. So they're just trying to see if you can actually make mobile labs available at a primary health center or anything like that, where it could be easy for the patient to just access it instead of traveling, say, a 50-mile or a 25-mile commute. Patient service center are also applications for various blood draws. Concepts in the early phase with late 2021 or early 2022 pilots. So some of the labs have already tried testing it, with the mobile labs, especially by partnering with some of the pharmacies, some of the local primary health centers, and more of clinicians and investigator sites more rurally. And we are talking about some countries in the US and Europe, but this might be a reality in other parts of the world as well. So let me move on to the next slide, please. Internet of Things. I've spoken to you about Internet of Behavior. So more likely, the new trend is Internet of Behavior. So you use a lot of smart wearables, and earlier it used to be just transmitting data, but now it is reading human behavior more from a linear algorithmic perspective and trying to see what can be prescribed using more information and more real-time review by clinical scientists. So coming to decentralized trials, virtual clinical visits, we spoke a lot about it. So the smart wearables is actually helping in terms of connecting all the data and bringing that together and making sure you could be transmitting the clinical trial data faster. Data storage in cloud assisting faster data cleaning and release. So earlier, some of the clinical trial teams actually had to go through a very heavy reconciliation process while they get source data from different systems and tools, just because they had to go through data review, data integrity, data cleanliness, data interoperability, you call it a lot of different ways. But the challenge basically was a single template or a single ledger or a single format was not available. But with now Internet of Things, they are trying to see how they can bring all of that together from a single source of data point and ensure clinical trials are completed faster. Another big advantage with decentralized clinical trial is bring your own device. So this is something which is gaining a lot of popularity with DCT trials, and you don't have to have a system which is being provided by the sponsor. Instead, the patient will be rewarded for bringing their own device. They just have to calibrate to the clinical trial needs so as to ensure that the regulatory requirements are managed. Data analytics, big data reviewed using analytical tools and algorithms. And this is actually given 70% time reduction in gene sampling. If a pathologist was actually reviewing 100,000 samples in X number of weeks, now it takes 70% lesser time, and to try and identify, which of the actual cell needs flagging, so that it could actually go through a more cell and gene therapy kind of a review, especially for oncology studies. Go or no go decision on clinical trial starts, saving up to $18 million every year for large pharma. So earlier, if an organization or a pharma company had to make a decision on a molecule, it took them several weeks to review the data. Sometimes they just went with their internal pipeline. Sometimes they had to actually bring in a different small pharma, buy their molecule and try to see if they can actually bring in a large value proposition for the big sponsors. So those decisions using Internet of Things, basically around the data analytics part of Internet of Things and smart wearables, has helped save big pharma up to $18 million every year. Clinical research training, it has actually reduced a lot, and that is because of the fact that a lot of tools are now becoming easier to manage and have lesser support needed by anybody in the site to help the patients understand what and how a tool needs to be used for. More natural processors use machine language and hence reduce time to train site staff and patients on devices. Training suited based on eye movements, automated bots replacing data entry, and reduced training. So all of this in a complete package, if you see the latest trends, some of them may be slower to adopt, but they are actually trying to see how this can help in getting clinical trials done faster. I hope this should be the last slide. Do we have another slide, Samprita? Oh, sorry. So basically, I just wanted to say, we've spoken about a lot of things, but honestly, when you're applying for roles or trying to look into the industry and see for roles, the basic expectation on some of the roles have actually changed in the new world order. So a CRC, or a clinical research coordinator, is also supposed to be a technology specialist to support patient queries on digital trial processes. So I mentioned, there is a lot of devices available, bring your own device, and all of these things, right? So honestly, these are actually going to change the way by which each of the roles are also changing. So just like I said, a patient might need some help, and the clinical research coordinator now has to be a technology specialist to help them understand how to use a particular device. Investigators monitor remotely the health of the trial. Earlier, you had to have somebody else review the data, but now a lot of remote monitoring or on-site monitoring is actually a role of a site investigator. Data managers, they need to be more data scientists to understand, review, and analyze data coming through unconventional sources. Earlier, you just get an data set and you provide a data set review, and based on the data set review, you query sites, and your role is over as a data manager. But now it's not just one source, one form of data. It is actually changing, and obviously a data manager need to be equipped with the skills to understand each of the data sets more clearly. And I'll try to keep telling you this, more and more technology advancements happen. Your domain expertise is what's going to help you and become a more domain expert, domain focused. I was part of what Hetal also said, try to attend a lot of conferences, workshops, read, understand, but more often, don't just stop yourself with the theory part of it. Try to talk to industry experts, try to understand, how things work. Your blockchain, fine. IoT, fine. It's not just about understanding the theoretical part of it. How do I apply? How do I make it happen? And that is more important in the upcoming world, where a lot of RPA is going to take up, mundane roles. So with that, I would also stop by saying, whatever you try and do, the key competency that is required to come into clinical research is always communication. There's so much roles, so many things happening, but the person with a good communication, able to understand what the other person is saying and making the needs of other person being met more easily, is going to be the successful person in any role in the future. So just remember domain expertise, communication, and seeing how you can apply all the new technologies from a actual work perspective. These are three things I would say anybody would try to actually learn for future roles. With that said, thank you for the opportunity, and I really look forward to see if I can answer any questions.
Thank you, Praveen, for the wonderful session on the latest trends in the clinical research industry. So the past 2 years have been very challenging across all walks of life, and we've been trying to embrace the digital world. So likewise, we've had a boom in terms of the technological advancements in clinical research industry, and we have been speaking about big data, artificial intelligence, natural language processing across various technical forums. And for we data management guys, we are no more called as data managers. Rather, we are working towards being data scientists. So as rightly said by Praveen, it's very important to equip yourself technically, and the key to success is communication. Thank you, Praveen. So we await questions in the chat box to Praveen. So I request all of you to post questions relevant to this session, please.
Okay, I see a couple of questions there. One is around the decentralized trials,
Right,
and won't the site face any difficulties? So, like just like I mentioned, what kind of difficulties are we really talking about? Is it more around patient care, or a particular sponsor or CRO can approach the patient directly? Does not mean you don't need the investigator sites. Obviously, you still need the investigator sites, because the CRO or sponsor can just identify patients. And let me give you some interesting statistic around the number of patients you would be requiring. Okay, so with decentralized trials and the number of trials that are actually currently running, it has increased by about 52%. And you actually need 2.9 million active patients to run clinical trials. So even if the sponsor or the CRO is going to try and approach a patient directly, the intel on where the patients lie, the number of patients that will be available for a clinical trial is still a subject matter or a domain expertise that the investigator site will have. So that's not going to change, at least for some years in the future. I don't think it's going to change. And again, a CRO or sponsor cannot be a third-party vendor for a investigator site to run a trial, because the roles are very specifically written and documented as to what can be done versus not done, as part of a contract. Probably, it would be interesting to see what else can a CRO take from an investigator site, if that is related to a data review or anything related to a data coming in from different sources, like wearables and things like that. But I don't think the role of a site is going to change. Their probable domain expertise is patient care, and also the number of patients they can make available for clinical trials. And with more and more clinical trials adding, I don't think that's going to change. And electronic informed consent process, I see that's the second question there. So, that is also something, the use of digital signatures, the use of blockchain ledgers are being tried as a possible measure to take informed consent. They are now trying to see, if you can actually have a video call to try and understand if a patient would want to sign up for a clinical trial and take that in electronic informed consent with a date in mind, stating that, whenever there's a physical visit that's going to happen in the future, they're just going to try and see if defrauding can be stopped. I think, the question is more around, okay, you take an electronic informed consent, but is the same patient going to visit me as the clinical trial progresses? And if that is the thought, I definitely will like to tell you, it's not going to be fully decentralized, because patient care is not changing. A lot of patient care is going to be more patient-centric and needs physical attention. And I definitely think, electronic informed consent could reduce the time for you to take consent in the first place, but you still need to go and validate the consent at some point in time physically. So, I may just try to be a little varied on my thought here by saying, your technology is going to be helping you, but to a certain extent being more regulated, we cannot stop. We definitely have to validate what we have as information, and that's still going to be the case. I think I've answered all the questions.
Yes, Praveen. I do not see any further questions.
Thank you.
And I completely agree with you that clinical care nowadays is not just about the patient survival, rather it is more about the well-being of the patient and how good the treatment has done in terms of the betterment of the patient's life. So thank you for the wonderful presentation, Praveen. So we move on to Anadia. Anadia, can you please confirm that you can hear me?
Yes, Samprita, I can.
Okay, so Anadia, we did have your introduction when you were facing this technical glitch. So I welcome you to this session wherein Anadia would be presenting to all of us on the introduction to the biostatistics team. So welcome, Anadia, and over to you.
Thank you, Samprita. Hope my presentation is visible.
Yes.
Okay. So first of all, welcome you all in the very basic and introductory session of biostatistics. It's very difficult to cover the things in half an hour, but I will try to cover it maximum for motivation purpose and couple of example which can tell you the relevance of small statistics and how valuable and informative it is going to be. So first of all, in medical research or any kind of research where there is any uncertainty involved and you are not sure how to take the decision, then in such places statistics come into place. It will measure the amount of uncertainty, and then once you know that how much amount of uncertainty is being involved here and how much I am sure, then your decision making process is going to be very easy. So what I'm going to cover some basic statistics and confidence interval and probably the sample size estimation, because these are quite very preliminary needed things before you start for any research. So to understand the statistics, we start with the data. We collect the data in a broadly two different formats. One is that qualitative or categorical, and one is quantitative or continuous in nature. For very sophisticated statistical analysis, we further classify the qualitative and quantitative data in a different terminology like nominal scale, ordinal, and numerical versus ratio and interval scale. These have certain benefits because at this point you won't see that why we are classifying, but these are having certain benefit at a very minute level of statistical analysis. Now, once we go for the descriptive statistics, you can see that there are mainly two things on which we are moving around. One is whatever you are measuring. You want to know where the central position of my all the data is. Means if you can hang the data at a single point where it will make a balance, something like that. So identify the central locality of data. We have a couple of statistics like mean, median, and mode. And then what we want to see that from the central position, how much variation is involved. So I'm not sure if I can use pointer. Yes. So suppose I am dealing with a normal data, and this is the standard normal curve you can see. So then on the central point, this is the central point of that. And in a situation of normal distribution, the property is that your central tendency, all mean, median, and mode will be on each other, means exactly in the center. If the distribution is little bit skewed or something different, then they may be little bit apart from each other, but almost they will be in center. So that's why you are going to measure the central tendency, and then you are interested that from this central location, how far my data is going to be. So some data may be very far extreme in one side and other side, and to measure those kind of things, we have many statistics, I can say. We understand from range that what is the maximum and minimum Then there is an interquartile range. We understand that 1/3 and 3/4, and then what is the difference between that? Mean absolute deviation. So from mean and from your data, what is the exact difference? So from your central, how much far the individual data points are lying. We measure the variance, standard deviation, standard error, and sometimes Z score we deal with. So these all are to understand that amount of variability in the data. Understanding normal distribution more, as I told that in a perfect normal distribution, mean, median, mode, both are perfectly aligned. But if data is skewed, then they may be left or rightly going to be to each other. Now, many times we calculate mean, and here, means my request from the researcher is that you lead the research, and if you understand the consequence or the challenge involved in particular analysis, and once you have the result you are interpreting, then what could be the consequence, then your overall decision making process will be very smooth and quite accurate. Otherwise, statistical techniques has their own assumption involved, merit and demerit, and if you will not be able to catch that up very precisely, then probably you will going to mess up the things which is your primary interest. So let's see it with a very small example with measure of central tendency. I am going to measure average. So here is the number of subject I have, and then their duration of stay in the hospital. It's a inpatient data and purely dummy data, nothing to worry. So just see that subject need to be stay in 6 days, 8 days, 4 days. This is something, but there is one subject who required to stay for 32 days. Now suppose either as a researcher or as a manager in a hospital management, you have to decide that on an average, how many number of beds I need to accommodate the demand. So what you take this data, you get this 11% data, you average it out. You got 8.36. So 8.36 is somewhere you need approximately 9 bed on an average. That's your peak demand, average wise. And this may be going to accommodate more than 50% of the demand or more. Okay. Now, this mean has a property like it is highly influenced by the outlier or extreme value. After reviewing this data, definitely you can see that this may be the outlier or extreme value. Now, if this will be the real situation, we cannot say this will be outlier, because just now you have observed a case in COVID management. On an average, you required 14 to 15 days stay, but there are a couple of cases who's required months and months to stay in the hospital to recovery. So things can go complicated, data can go very skewed. And suppose this is the situation real, then you have taken average 8.36. Now, just remove this outlier and take the mean for 10%. Now you got 5.9, means approximately 6, here 8.5. So how much number of bed minimum you have to maintain? It's a big decision, maybe, with a small example. So whether your mean is indicating you right or it is misleading you. So after removing this, you got 5.9. So which value you will rely for your decision making process? And absolutely here you are measuring the central tendency, the average number of bed needed. So going forward, and as you know that mean is severely affected by outliers or the extreme value, and in your data you have at least one example for such, then probably on the mean estimate you won't be rely very much. So what you do, you are going to take the next measure of central tendency, and that is median. Now, median is what? 50% of the information towards the left side and 50 was towards the right side. You are going to find the mid value exactly. So calculating median has a different process. You have to order the data, and then you have to pinpoint the exact pointer where you are going to get the central value. And after analyzing, we observe the sixth value in series is the 6. So it's approximately mean value of 10%, 5.9. So here, once you analyze the mean, you got 8.36. After removing the outlier, you got 5.9. But including this outlier value in your data set, analyzing with median, you got 6. So this is the average number. Now, probably you can go ahead with the 6 number of bed on an average to maintain. What will happen? It will accommodate on an average the number of demands. There may be a situation when certain extreme cases will come, then you have to manage it. But if you take in the first decision, like here, initially recommending 8.5, like 9 bed, then almost in your hospital, since the 6 is the average, almost 2 to 3 bed is going to be vacant. So this will be a kind of resource loss. So with this example, I would like to emphasize that how small statistics is going to be very, very valuable and helpful towards your decision-making process if you understand its assumption and if you understand what could be the consequence when generalizing. Okay, now moving towards the next level. Most of the time, statisticians educate that when you do the analysis, you need a normal distribution data. Any inference can be drawn on the normal distribution. So there is a couple of variables which we collected, and we try to put all the summary statistics for that. So this is a printout of SPSS. You can plug in the variables in one side. You can ask for statistics. It will give you. What's the problem here? Problem of data categorization. Okay, so I have calculated mean, standard deviation, median, all the variation of all the data what I have. That is not right. I have to apply the statistics based on the nature of the data. Like age is a continuous variable, so understanding average age of the subject is somewhere 50 years, there is standard deviation, and so on, so like. But having mean of gender is 7.6. It doesn't make any sense. My whole data is classified as 010101. Now, based on 01, computer don't understand that what you are plugging, what 0 means and what 1 means. But it just analyze and say that from your data, the average mean is 0.676. Now, here your cognition is required to understand the classification of the data, like how it is collected, what is its nature, and which could be the appropriate statistics for that variable if I want to measure the summary. HbA1c correctly, duration of diabetes, that is also a continuous variable. Somehow it can be explained. However, there may be good situation that time to event data can be go ahead with the median. There is an assumption for that. Now, this is by VPT that is also collected in 01. And again, you see software messed it up. It's just calculated 0.56 and standard error 1.01. Now, this value is not going to make any meaningful. But we have a tendency because of availability of software to plug and extract. We have to put a lot of cognizance there. And then we can probably arrive at a better conclusion. Okay, now trying to understand what kind of, how I can assure the normal distribution for my variables. There is a lot of techniques. You have to plot it, you have to check by the visuals, and highest level, you can put a statistical test to check the normalcy. So I have put a statistical test to check the normalcy. Okay. Now, what test is Sparrow-Wilk test? There are a couple of more tests. This is just I displayed. And then I have put this test on all variables. Okay. Now, from the property, I know that gender and net BPP is not a normal. It is a categorical variable. When applied test, what kind of p-value I am getting? Age 0.467, gender a very high number as a p-value, HbA1c 0.03, and so on. Now what I can conclude? My alpha level is 0.05. That's the default. So whether my calculated p-value is less than, probably for age, I can say yes. So then it is going to be a normal for me. This is very high value compared to this. No. This is also 0.06, slightly higher than 0.05, but still, if I can say, it may come to maybe the sample size is less or what. But for statistical test purpose, still I can go ahead with this. However, this is time to event data, so exactly what statistics I will use, that will depend. And this is absolutely no. So that's what you can apply the test and check whether it is normally distributed or not. Coming to the understanding the standard deviation and variance, we know that variability is going to be measured the dispersion of the data, and standard deviation is the square root of the variance. So what is the variance exactly? So the average of the square difference from the mean. I would like to again re-emphasize the average of the square difference from the mean. Read it from the right hand side. So difference from the mean, it means you have calculated, you have your data, you have calculated first mean, so you got the average value. Now for every single point in your data, you have calculated the difference. So you got difference for every individual data point from its mean. Then you square that difference, and then you take the average of that square. This is you called variance. This is the calculation step, but it will give you a lot of insight to understand the data. Now, these are the steps. The formula is something mentioned like that. And the good thing is that if you understand a normal distribution, you can apply 68, 95, 99 rule. It means from your central point, plus minus 1 SD is going to cover 68% of the data, and 2 SD is 95%, 3 SD is 99% of the data. This is again same, the graphical view, that how the data is covered if your data is normal. There are some other statistics which is mostly deal in the problem when there is a non-normal data, or there is a different unit. Like just a small example, you are measuring the height, and one person is measuring in centimeter and one person is measuring in meter. Okay, now who measure in centimeter, he is claiming that my average is somewhere around 165 centimeter, and whoever measuring in meter is saying that 1.65. And similarly, it will be very big difference in the standard deviation. Okay, so if one is numerically saying I have a value of 165, and one is saying no, I have a value of 1.65, numerically these are two different quantity. Looks like one is very smaller, one is very high, but operationally or biologically both are the same, exactly the same. So how to deal with the different type of measurement, and if you want to report certain statistics, would be unit independent. Like I get the information, and then whatever unit it's was measured doesn't matter, I am always going to conclude the same thing. So then it was proposed as a coefficient of variation, which will be kind of variation divided by the mean into 100, and then you understand the variability irrespective of its numerical quantity or irrespective of its the unit of measurement. So this will give a lot of other insight in different, like different lab value has been measured in NGML or MGDL or something like that. So that type of situation, how much variability is there in the lab value, can be assessed by CV. So choosing the right measure for a spread, this table can help you out that if nominal, then nominal data will have kind of a percentage and category. There won't be much you can do. Ordinal data, you can use range, you can use interquartile range, but standard deviation you cannot compute. And with the numerical data, I think you can compute many statistics. So that's the limitation with the data. Now, understanding which kind of data you have and how you can analyze the data. So if you have a numerical data, then numerical data can go to the parametric setup where you know it is normally distributed or some other distribution, or you don't know their distribution. So if you go parametric setup, then two group analysis, it's going with the unpaired t-test. If greater than two group, means 3, 4, you can go ahead with ANOVA. If you don't know the distribution, then you apply for non-parametric, and for two group, Mann-Whitney, and greater than two group, Kruskal-Wallis. Similarly, in categorical analysis, two group is going to be dealt by the chi-square test or Fisher-Exact test, and more than two group, chi-square are maybe complicated with the mental handles, something like that. Now, just to understand with a case, to see if there is a significant difference between the mean of BMI of the diabetic patient, pre-diabetic patient, and non-diabetic. So what I'm doing here, sample of diabetic patient I have, sample of non-diabetic patient I have, and everywhere I have calculated their BMI because my interest is in the BMI. So I have a 3 group, okay, and then I used to run a ANOVA for average BMI in 3 group. Case 2, to see if there is a significant difference between the mean of HDL of diabetic, pre-diabetic, non-diabetic patient with respect to the gender. So what I'm going to do, I'm going to introduce the gender concept also. So I have a 3 group, and then all the 3 group are further classified into the gender, male and female. So this is a quantifiable variable, the HDL which I'm measuring. So it is a again two-way ANOVA I'm going to use, where one classification is by group and one classification will be by the gender. Next level is that suppose I have a paired observation or dependent sampling, where my next observation is pretty much dependent on my previous observation. It's not like independent observation, it's a relative observation, a repeated observation. Then what the testing procedure will be. For the numerical, again parametric, then 2 group, pair t-test, more than 2 group, that repeated measure ANOVA I will use. And if it is parametric, then 2 group for Wilcoxon and more than 2 group Friedman test. Similarly, in going to categorical analysis, for 2 group, the McNemar test, and more than 2 group, Cochrane's Q test will be applicable.
Hi, Nadia. Sorry for interrupting. We'll just keep a tab on the time, and
okay,
probably you can adjust your presentation accordingly. Thank you.
Sure. So further to that, when there is a association I'm going to measure between two, so there is also parametric and non-parametric concept, and we can measure the association with the appropriate statistics. This is one example, like HbA1c I want to measure with as a dependent, and there are 3 independent variables I have collected. So in this case, the correlation can be measured with the Pearson, and I can perform a simple regression analysis to know that what is the effect of each variable on my outcome. Okay, this is going to give you the modeling concept like continuous variable, and then what will be the best model is linear regression, and this may be non-linear also, but mostly we go with that. If there is a binary outcome, then we go for logistic. If time to event data, then we go for Cox regression model. And what we are going to estimate is the beta coefficient in each model and how we will present our result in terms of odds ratio, slope, and hazard ratio. The same thing explained that how we are analyzing the difference between survival trends, time to event data. There is a small concept I would like to introduce about the sampling. And if you have a representative of the sample, a representative of the population, then your inference is going to be reliable. Otherwise, it may create a lot of problem. So it should be representative, external validity also should be holds, and then it will be generalizable. Okay, so this is a pictorial graph, like how my sample looks like, something very resemblance to the population. A lot of sampling methods are there, where one is like probabilistic sampling, where your chance is going to be controlled and you can measure it. Then is non-probabilistic sampling also, that is convenience or voluntary sampling. Here there will be problem of bias and we have to handle that. Okay, in the sample size calculation, there are a couple of things which we have to know, like when we plan for appropriate sample size, then it will improve our precision, it will give the adequate power, economical viability will also be assured, and then we can understand that how much ethically it will be right to do and what we can have optimal sample with this. What factor can affect my sample size? So my study, which type of that, what primary endpoint I am going to assess, power, how much I want, the variability on the measurmental variable, and the effect size. If I want to adjust for dropouts, attrition, then, and mainly that availability of the subject. Statistically, I can derive any sample size, but whether those subjects are available in the population or not, that is the next level challenge. So we have to make a balance in that. Hypothesis testing, this is pretty much standard. So we can go ahead with the hypothesis setup. As you know, the null hypothesis we have to plan, which is intended for rejection purpose. And once we reject, then we prove our hypothesis. There is an example I have with the placebo versus drug, and how I constructed the null hypothesis, and then how I concluded with that, I have explained. This is a very classical example when you know that what is the exact truth. There is a difference between the treatment versus placebo or no difference. And from the means my sample, when I do the research, what I am getting, significance or not significant. So two places I am doing the right decision, while two places there is a wrong decision. One is alpha and beta, and then all the statistical principle is moving around to adjust this. And from the type 1 error, which is fixed at 5% level, and if my error is coming less than that, that is my p-value, it will give the significance. Beta, from the sample itself, we will control that how much power we want, and hence it is controlled. And this is the example that how I am going to conclude the p-value. Based on the p-value, my end result. And couple of things, like when my p-value is 0.0215, then there is a 2.5% chance otherwise drug reduce the BMI. And this is kind of significance. If it is 215, it's 21% chance. By chance it is happening, and then it will be insignificant. So this is the pictorial diagram, and which will give you the best situation when you can confirmately take the conclusion. It's like with the lowest p-value. The same example I have explained in different situation of beta, where your confidence is pretty much high. So this I explained the very brief concept of testing of hypothesis. This ends my presentation. Thank you. I can now answer the question.
Thank you, Nadia. So usually everybody's perception is that the biostats team will come into picture only at the study closeout phase. In your presentation, it is very clear that biostats play a very vital role from the study startup during the conduct phase as well as the closeout. And I must say it had been quite a while since I really heard the words like the mean, mode, median, alpha, and beta. So it really took me down the memory lane of my maths classes. So thank you for the wonderful presentation and letting the team know as to how the biostatistic processes really work. So let's move on to the questions. So we already have a question from Abhilash. So he says, in trials, it is said that the power of the trial should be more than 80%. So why it is so and how it is related to sample size?
Yes, very, very relevant question about the power. And many of the time we thought that what should be 80%, 90%, and what is the logic behind that? So the logic is like that, suppose you are testing a treatment and your treatment is working, but due to the less sample or due to the less confidence, your sample is not able to prove that your treatment is working. In that situation, you are at great risk. Your whole financial and budget is at great risk. And that situation you almost want to avoid. So what situation it is, like my treatment is working, but my sample is saying treatment is not working. This is another type of problem, which we call type 2 problem. And then from the beta, you can fix it or maximize it, I can say. As maximize that if my treatment is working, it must be so in my sample, it is working. And how much confidence you want to put it? At least 80%, so that you are sure that if it is working, it will reflect from my sample. And thus the whole mechanism of sample size derivation will go, and with certain level of beta, it is going to. So if your power is less, your sample is less. If your power is going to high, your sample is going to high. Thank you.
Thank you, Nadia. Any further questions? Team, any questions related to the session, what we've had from Nadia? Okay, so I take it as a no there. So thank you once again for the thoughtful presentation, Nadia. So as per the agenda, we now move on to the next event. So I hope all these sessions have been very informative and really helped to bridge any gaps you all had in terms of understanding the clinical research industry. So I must say that we are done with baking the cake. So how about the icing to give it a perfect finish, guys? So yes, you got it right. So let's move on to the panel discussion on careers in clinical research. I'm sure this is the most awaited session through the day, and we have all our esteemed speakers as panel members, and the session would be moderated by me. Can everybody confirm that you are having the panelist access?
Yes, I can confirm.
Check yes. Chandana is added. I think she's the only one who may be missing.
I have it, Samprita.
Oh, she's there.
She's there.
Okay, great. Okay.
No.
So a quick check. We have Hetal, Praveen. We have Rutheja. Chandana is there. I do not see Payal, Aniket.
I'm here, Samprita.
Okay, I can see Nadia. Guess we have everyone. Yes. So are we good to start?
Yes.
Yeah.
Okay. So let me give a quick education background about myself to let you know as to what struggle I faced when I was fresh out of college. So basically, I hold an engineering degree in biotechnology from VTU, and ours was the first batch in engineering biotechnology way back in 2002. So all excited, I just joined the course and enjoyed the whole journey. And finally, when it was time to complete the course, I was completely clueless as to what's in store for me in the next few months. Of course, there were very few universities back then which were offering the master's degree in India, and there was no campus recruitment at all happening for the biotech branch. Of course, most of my friends did join IT companies like Infosys and TCS with no other hopes left, but I was clearly determined that I have to stick to my core subject. So, but then I'm confused whether I have to go do my higher studies or should I work? So with completely blank thoughts, I just reached out to my HOD who just randomly gave me some contacts and said, Samprita, please just go ahead and mail these guys. So thankfully, I got a call from one of the HR from the corporate sector, and I had to face an interview within next 2 to 3 days. So I go there with a completely blank face. It's my first interview, and I'm clueless. I was really not aware that clinical research industry existed. So then the interviewer started shooting questions, and I'm there with completely blank face and trying to mutter, trying to gather some courage and trying to answer, but then I failed miserably. But I came back, but then I was frank enough to tell my interviewer that I'm very new. I did not know about the industry, but then I can really give my best if I'm given a chance. And few days down the lane, I get a call from HR and voila, I landed up with my first job. And I can say that that moment has been historic and there's been no looking back ever since then. So I'm sure that you all can relate to what I went through a few years back. And that is the reason we all are here to help you, to support you all, to better understand as to what is the prospects of the various job opportunities within the clinical research industry. So with that, we would be covering the various aspects as to how you have to deal with the present situation. So we will discuss about the various career opportunities, be it in terms of your higher education, pursuing your PhD, or landing a job in the clinical research industry, as well as what are the skill sets you need to acquire before applying for a job. So with that, Chandana, before we really jump into the career opportunities, kindly share your thoughts as to how students have to go about first deciding whether they have to pursue higher studies or look out for a job. Chandana, I guess you're speaking on mute. Yeah.
Thank you, Samprita. And I think the journey that you related, all of us, perhaps, especially people who are in clinical research, when we stepped into clinical research, it was all a new field for us. Just like your faculty gave you some numbers. When I came back from UK, somebody invited me to join their team and asked me to join as a team member in clinical research. And I honestly did not know, I had not heard the name clinical research. And because he was a very senior respected person, I said, yes, sir, I will join, but please allow me the day I don't like it, what I am doing, I will leave that place, because I was happy in my own space. But today, after having stepped into clinical research, just like as you said, Samprita, you're so happy that you took the call that day and it was a historic day for you. I cannot be, saying less about the same for me, as when I joined, I did not know what it was. But as I joined and worked through the field of clinical research, it could not have been better and more excitement in the field that I'm working today. So when we talk about the life sciences graduates, when we have taken life sciences versus arts and commerce, when we had to down there after our 12th, we had to choose what we wanted to do. When we took the life sciences, we wanted to work for diseases, patients, science, etc. And when you get to work for patients, nothing better than that. And in clinical research, I feel that you can actually put your science into practice. You will be able to look after your patients, run the best of the protocols, the global protocols for your patients, and feel very happy. If one of your molecules also comes to the market, you are very happy and proud to say that, yes, I work for this molecule. And there were so many people who had taken part in that, and they all have their individual stories to tell us later. So when we think about a career for the students today who are with us, see, we are not looking at, of course, education, of course, adding to the knowledge, but then the whole purpose is to land us in a place, in a space, in a job which we enjoy doing, what we want to see ourselves tomorrow. We do not want to see ourselves as university students doing our graduation, post-graduation, our PhD and postdoc. Yes, that is a process. But then while we are looking at that process, we must also ensure that we have a goal to reach, we reach the goal, and then we learn as we are going in the process of our employment. So that is what I actually tell all my students, all the parents of the students who actually sometimes ask me, should I go for the next degree or should I, land in a job? There are both ways which are there in my space. So I would personally say that land in a job if you get a job to your choice and grow along with your job, so that tomorrow you don't have to hear from an HR person that you are too educated for the job that you are looking at. Whenever we join as a fresher. So let us go join as a fresher and learn while we are in the job and learn the specific needs of the job that is close to your heart.
Thank you. Very thoughtful suggestion, Chandana. Thank you so much. So the next is, Ullas, I see that you have an MPharm degree, and we all would love to know as to what was your approach towards the future steps, and if anyone really wishes to pursue higher studies. So what are the current options available for the students?
Okay, in my case, it was a little different, and I wouldn't want to go that path again, but because I did a lot of experimentation with my life. But that's again, what Chandana said. I think that's what that makes sense. You need to follow your heart. You need to ask yourself, because I think when you start your graduate education, like whether you're doing B.Pharm or engineering or whatever, biotechnology, first 1 or 2 years you may not have that clearer idea in terms of what you will become, which line you may end up in. But then I think as you progress, maybe the third year, the final year, things slowly start getting a little bit more clear. If not, then you should try and make them clear, and then you decide whether you ask yourself, do I want to go for a higher degree, or is it time for me to jump into the workforce, get some experience, and then come back to academics? Because nothing is stopping you. Once you start working, it doesn't mean you stop learning, you stop going back to education. Who said that? That is not at all the way, because it all depends on the which line you're in. Especially health science, if I have to speak, learning goes a little bit further. Until you earn at least a master's degree, you feel like, okay, you don't feel that confident. With master's degree, you feel a little bit more confident. But then it is very much case by case basis. It's very much subjective. If you have that clarity at the end of B.Pharm, jump into workforce. That's what I would say. If you think you need to, if you're interested to know about a particular field after learning pharmacy for 4 years, there are many subjects there: pharmaceutical chemistry, pharmacology, pharmacognosy, pharmaceutics. If any of the subject interests you really, like, so much that, okay, this particular subject I want to learn a bit more, dig deeper, if that is what your heart is saying, then go for higher education. But then always follow your heart.
Yes, I completely agree that especially the final year students, we are very naive, and things have changed now, wherein we have access to a lot of information. We have various events happening, like what we are presently in, and we also have LinkedIn. We have lots of communication methods. So we do have lots of options which the students can explore. So on those lines, Rutheja, if you can really tell us the present prospects of the various internship programs which the corporate organizations are running. So what is your take on it?
Okay, so I think the way in which you started, Samprita, it was really interesting. So I remember, again, I would like to go back a little to say that when we started our career, I think we were not very lucky to have the internship program ongoing. But now the things are changed as the clinical research professionals, we are growing on a day-to-day basis. The number of trials that are coming to India, the number of activities that are coming to India are tremendously increasing. And definitely, so the need of internship is not just for the colleges, but it's also becoming a need for the corporate world, for the organizations as well, because it's a time when organizations are thinking of nurturing new talents into clinical research field. I think today's session is one of the best example that I can say that the steps that are being taken by the organizations or by the society, the people who are working in clinical research in order to promote careers in clinical research. And definitely that starts with internship. That can be also university hires. So many organizations have started changing their approach. So not just going with the old, we want people with experience, but cultivating talent is something which has been taken up by many organizations, and it's been seen increasing on a regular basis. And efforts have been put in by organizations to train the people, to put on efforts, time, energy to train people who will be able to support in the clinical research field in the future. So yes, definitely, if I compare the trend, it's increasing. More and more organizations are coming in. It could be an internship, it could be a university hire program, it could be giving other opportunities. So that's definitely coming in, and the chances are pretty good. The way in which it has been kept, or the way in which the hiring happens for other fields, I think it's the same thing now, which is it's coming up in clinical research as well.
Great, great. So I'm happy to share that I have been part of the internship program in our organization for the last 3 years, and I've been spearheading that, and I get to meet with the various college students, and whenever we are interviewing them, they've done exceptionally well in the technical round, but when it comes to the HR round and the further discussions, we get to hear that, okay, so what do you want to do in the next 1 or 2 years? So they say, ma'am, I want to complete the internship program. I want to work for a year. Then I want to do PhD. So I question back as to, okay, so what do you think? How will your PhD program really help you? So I'm not sure about that, but then I still want to do my PhD. So I think Dr. Hetal can really help us to understand as to what is your take on students wanting to pursue PhD.
Samprita, my take on students who want to pursue PhD, I think just go ahead and do it because you already decided by then. The question if is whether you should or you're not sure whether you should be pursuing your PhD or not. Again, I think all of us are trying to say the same thing, that it will boil down on what career paths you are looking at, what are your aspirations, what are your areas of interest at work. So like from PhD point of view, if you see, there are a lot of roles that we spoke today, and not all of them need you to go through the process of PhD, and probably it may not help you to go back to take that to the workforce. So then probably it would be more to gather those work experience and equate it to the PhD training and take it from there. While some roles, like for instance medical writing or even pharmacovigilance or medical affairs, is where doing digging down further as part of the PhD might help you and have a very great added advantage, not just to kickstart your career, but also to take it further eventually once you have reached a plateau or a level. So that is definitely about it. One. Two is, I am yet to meet someone who has done PhD and repenting about it. So all cribbing and all complaining and all frustration is aside, but there's no one, at least I have met, unless you goofed up very badly, that you would repent doing it. So definitely that's something you will have to take a call on an individual level. Again, with the kind of information we are right now talking on, or the kind of exposure the student community has now, you can have multiple options, like jumping to your PhD directly from your masters, or doing some work for 5 years, taking up industry sponsored or collaborative projects, which like gives you the best of both worlds, like working and doing your PhD. So those kind of options you should explore. So to end it, I would say, and I always keep saying that to people who come back to me for, especially with this question, PhD is like a process of alchemy. The way the refinement or the transformation it will bring in you as an individual is immense. The certification is just like the icing on the cake and obviously has an added advantage. But what you should look for is the process, if that is going to add any value to you as an individual, as a professional, for what you try to do. Now, answering all this may not be possible at the start when you really want to decide. So don't be harsh on yourself. Take the best estimates that you have on your plate and give it your heart, as everyone says, and go for it if you feel like.
Sure, Hetal, thank you. So I guess we also have a lot of universities which are offering these integrated courses wherein they get to complete their masters and hand in hand they also pursue their PhD. So I'm sure the millennials do have a lot of options if they really wish to study further. So on the same lines now, we've heard about the options of higher studies. So when it comes to the life science students, we usually think that they are well fitted only in data management or pharmacovigilance or be it clinical operations. So Nadia, kindly share your thoughts as to what is the career prospects within the biostats team.
Yeah, sure, Samprita. It's nice to explain those concepts. Especially, I am from master's degree biostatistics, and after 15 years of my career in the industry, I decided to do the PhD. So now, from last 1.5 years, I'm doing it. So one is that your inspiration, and one is that means at this stage, if I do PhD or don't do PhD, it is not going to add any value further to my career progression or profession. But it is something like you are working in a very scientific environment, and it may strike any time in your mind to nourish further to the science, to cultivate further. And then you should not restrict yourself. You should go ahead with your desire and you should move ahead with your passion. If it is parallel option available, it's very nice and lucky to explore those. For biostatistics especially, it's not only for the clinical trial or research, it's a kind of techniques and tool which will inbuilt yourself up to a next level of confidence. If you understand the small values, how you calculate and how it can help you in your overall decision making process, it's not on the research. Your day-to-day activities which have certain priorities, you will able to figure it out. You will be able to execute it much more better way. Now, especially in the biostatistics sector, for career point of view, there are two different verticals is quite prevalent. One is the computational biologist, and one is that biostatistician consultant. So at very beginning, you have to decide that which vertical you want to take. Otherwise, at a later stage, it will be very difficult to swap. However, there are a couple of people who are good and experienced and well equipped to swap and very comfortable. But it's somehow I feel difficult because I swap in between. So one is like you become a pure statistician. You can contribute your whole research idea from the design level. And as a statistician, you will be able to suggest that what design can be better. And when I say the design, it means the clinical research design, like you want to do your research with crossover design or adaptive design or parallel design, whatever the way. And then what could be the endpoint, what could be the possible variable you want to collect, how you can analyze, how it should be collected in a sequential manner or is it independent. All those things as a consultant statistician you can advise to the research team. Now the next level is the computational biologist who do the computation. So if consultant statistician had advised certain things to do, there is a person who should be programmatically experienced enough who can perform those computation. And that's the next line career for the programmers, statistical programmer we call. And they are very experienced in the uh, software language. And nowadays, with a lot of scope in that area, especially with the advanced computation, advanced methodology, where you can have a new software who can integrate many things, can read data from multiple platforms, multiple standards, can summarize, integrate them, and present as a overall. A lot of good softwares like R Studio, Python, and these are very much integrating. Even these are helping you in the artificial intelligence and machine learning technology, where your machine is going to take the decision based on the whole computational algorithm. So those are taken by the programmers. So these are two vertical options available. However, there is a good integration in between them, and if you are having good exposure and skill, you can swap in career in between in either segment.
True.
That's from me.
True,
Thank you.
Anadya. So that is the same thoughts which crossed my mind when I was going through your bio sketch, and I completely agree that there is no age bar to learn more and more. So it is really not a hard and fast rule that you have to pursue a PhD as soon as you finish your master's, or you shouldn't do any further courses once you start working. So that is the reason we get to see a lot of executive master's programs, what is available in the market nowadays. And we see a lot of people like Nadia who are pursuing PhD, though they've had a good experience in their own field. So thank you, Nadia. So likewise, we also have another panel member with us who was a clinical practitioner for quite a few years, and now he is heading the safety team. So Aniket, I would like to hear from you as to, we usually see that the MPharm graduates flock the PV team. So we see that the concentration is more there within your team. But still there are various other departments within a CRO. And so according to you, where do you think that the MPharm graduates best fit in terms of the departments what they have they can choose, Aniket?
Yeah, thanks, Samprita, for that point. So I think for MPharm graduates, I think PV is first of all of the interest, because during their tenure of education, they must have definitely gone through either as a part of syllabus or maybe any parallel courses also. They must have gone through the knowledge of PV. That is one. And the other thing that from if you see, when a person actually enters into the domain of PV, one is the conceptual understanding, and second is actually the on job training. So I have also seen that MPharm graduates actually quite fit into the case processing profile. It may be at entry stage or also at a latter stage, because they have a good understanding of the concepts, and which actually very useful while doing any task or any process steps within the PV domain. So that's, I think, one of the point because of which maybe they have the interest itself to pursue the career into the domain of pharmacovigilance. The other thing is that I think if the students might have noticed, one of my slide actually was sort of keeping the spotlight on the requirement of a PVOI, pharmacovigilance officer in charge, and the requirements in India. So pharmacist is one of the requirement, and allowed, I would say, educational qualification for a qualified pharmacovigilance person in India for a company. So I think that's one more, I think, aspect wherein MPharm have a very good I would say career background behind them before they enter into PV, specifically in the clinical research.
True. Yes. So you do agree that the MPharm guys can really go explore outside the PV team?
Absolutely, outside PV also, yes. Definitely they can pursue. I think the key point here, just would like to summarize what Praveen also mentioned about the communication. So one thing that all students who are attending this, they need to understand, whichever field that you will enter, communication is very, very important. Not only to communicate yourself, but to understand what the organization has delegated you. So that's one thing. And whatever knowledge you have gained, you have to apply that accordingly. So that is also very important. So these two factors, if you learn, if you inculcate that, then I think it's you will get that, I would say, a boost to sort of have your journey on the right path.
Yes, right. So I guess it's all about evolution, I must say. So when we all were students, we started with a floppy disk, then we moved on to CDs, We moved on
Absolutely.
to pen drives. So having a 2 GB pen drive, I remember, was a big thing during our college days. So now I really don't remember as to what capacity pen drives are available though. So likewise, we are evolving in terms of technology, and on the same lines, even in our clinical research industry, technology is playing a very important role, I must say, and we clinical data managers are being addressed as data scientists. So though we are from the life science background, it is the need of the hour that we upskill ourselves in terms of our technical knowledge. So like how you said, yes, we did have this session from Praveen where he spoke about the various automation, what is available. He spoke about artificial intelligence, blockchain technology. So Praveen, from your perspective, what do you think that is this automation progress, what we are seeing? Is this really helping the newcomers? Do you think it's a boon or a bane for them?
Okay, thank you for that question, Samprita. So yeah, you spoke about evolution. It's a very interesting thing. I was just reading through an article about IBM in the 1990s. Okay, so it's like IBM asking the question about should I invest in software or hardware in the 1990s. Okay, it's like, see, automation is going to be coming up and it's going to get improved. And again, what is the core strength that a student is going to bring while he or she is going to join any organization? So IBM actually thought through, they made management changes. They thought it is hardware that was their strength. Okay. And they also said at some point in 2011, if you saw the story around Warren Buffett buying a lot of stocks in IBM at that time, he bought it because he saw that the management was changing and they wanted to make changes as part of the evolution, right? So they invested in cloud servers. They did not want to invest more and more on mainframes. Okay, so from a technology standpoint, they kind of changed themselves and they went into something called as IBM Watson. Everybody would have known a lot about it. IBM Watson is more of an AI-enabled tool or a database server that's available for making quick and analytical decisions. Okay, so you need to evolve. So basically, the answer is the industry is going to bring in a lot of automation. One, be prepared to identify what need to be automated and bring that to the table as a student. If you do that, you have a great career ahead. So that's one point that I want to make. The second point, subject matter expertise. I just mentioned it as part of the presentation as well. How do you communicate your subject matter expertise? Okay, the computer can actually communicate its own subject matter expertise by being very fast, by being very efficient, and by knowing a lot of things which are very important to make a clinical decision as part of a clinical trial. Are you that person? As a student, are you going to become a person more like, yeah, I'm building a subject matter expertise myself, but I won't communicate. Okay, so it's not going to help. Basically, it's like any talent that is not visible to others, it's not going to create value to anybody else, inclusive of the person who holds that subject matter expertise. So basically, I would advise that invest your time on communicating, invest your time on subject matter expertise, invest your time on finding automation, invest your time on creativity. That's the one last item that I want to leave with. Whatever automation is going to do, it is not going to replace creativity. And anybody with a creative mindset and a good communication is going to be very successful. In that note, automation is a boon for every student. How you use it is more important.
So that's some piece of good news, Praveen. And I guess the message is very clear from Praveen that how much ever we advance in terms of technology, end of it, what really matters is how we present ourselves and how do we take the right approach in terms of what career is best suited for us. So I would like to check with Payal as to, along with the technical skill sets, Payal, for the newcomers, what are the soft skills which they have to acquire or how do they have to prep themselves before facing an interview?
Thanks, Samprita. I think whatever I wanted to just say, Aniket summed it up really well. While on corporate, let me share my perspective more on the corporate and clinical research business. While it is imperative that you have a sound technical knowledge, process, compliance, documentation, procedures. However, I have seen a lot of this alone doesn't suffice. while delivering on role, and rather strong prowess on softer skills at times turn out to be equally or sometimes more valuable and important. On softer skills, I will put them largely in one big basket of people management, within which we can further delve into, communication, stakeholder management, time management, relationship management, and all those things. So, communication skills are very, very important. In our industry, we know, say, if I give you an example, as a CRA, if I'm going at the site, it is very difficult to catch hold of the doctors that we have, the investigators. Sometimes when you have to discuss with them a topic of, say, suppose 30 minutes, but they say that, we just have 5 or 10 minutes to give to you. So you have to adapt to those requirements, and you have to be very crisp in your communication. And a lot of this will also come through experience. And as I look back, as I look back around 10 years behind my journey, I have seen a huge transformation. So I think that's the major skill set that is required.
Yes, very true. So what I can say is, it's the first thing would be that the right approach and the what we choose, whether we wish to study or whether we really want to work, the skill sets what we acquire, how well prepared we are, and the key to everything is communication. So I would like to reach out to Sachin to sum up this whole discussion and share his thoughts, please.
Sure. Thank you, Samprita. I think it was a very insightful discussion. All of the panel members here brought in their own experience. How I looked at it is, basically, I think everybody said it's important for us to focus on the technical piece and also the communication piece. And this is how I see it. If you look at your career as a 30-year journey, in that, we are talking about the first few years, right? But the next 25 years, right? If you take the first 5 years now, is no different, right? So the learning is still continuing. You need to make sure that you have a better way to present yourself. You need to learn new things which are coming our way. So as Praveen was presenting, you have AI ML coming up now, and all of us are in that learning journey. We want to know how it impacts our work and how we can accommodate that, in our organization. So I think this learning journey is continuous, and I think that's the only one point, that you need to make sure that you keep an open mind, even if you join any organization or continue your PhD. I think that learning will go on. It's important for us to be open for it and then take all the opportunities, which we will have. So with that, I would like to thank everyone for this wonderful, insightful session. I thoroughly enjoyed, the entire day as well as, this session. So thank you.
Thank you, panelists, for the wonderful suggestion and your thoughts on this. So thank you once again. Now I request Ullas to conclude the session and share a few words, please.
Thanks a lot, all panel members, for your valuable time on a Saturday. First of all, I really appreciate you guys coming online to help this workshop, to give from your experience, from your skill sets, from your knowledge, share a few things so that these budding aspirants get benefited in whichever way they may want to go in their future. I'm sure all the attendees, whoever is there online right now and since morning, they would have made enough notes, and they would have had some ideas. If they were not clear before, might have been more clearer now. So, but as everybody said in the panel discussion, learning is an ongoing process, like Sachin said, right? So it never stops. And technology will keep coming, like Praveen said. We need to learn a new technology as soon as it is introduced in the market. So it is also, that's why they call it as disruptive innovation. So disruptive meaning, like, if something comes new, like, it is going to disrupt a few things, and we need to quickly adapt to survive. So that's how we all need to continue. And soft skills, like Payal also mentioned, it's really important. No matter how many things you know, how many degrees you have, if you are not able to use them in a proper way in the real life situation, when the interview you are at the interview desk and you are being asked a few questions, if you are not able to convince the other person sitting at the other side, no matter how many degrees you're having, is of no use, PhD or double PhD or whatever. So learning is what we all agreed on is very important, along with the soft skills. So thank you very much again for all the support. ISCR, I would like to thank wholeheartedly Sachin right from the beginning for all his suggestions and advice, and Samprita jumping in at the last moment and coordinating this whole event also, and getting me all the documents. So yes, please convey my best wishes to everybody, Sachin in the ISCR executive committee as well, and my please convey my thanks to them as well. And I would like to thank from this side the KLE University and their support as well, the Deputy Registrar and Professor and Head of Clinical Pharmacy Department, Dr. Ganachari sir, who is sitting next to me. And unfortunately, the principal had to go and attend to something. So Dr. Sunil Jalalpure, who is the principal of KLE College of Pharmacy, I thank them for all the support and lending the infrastructure to conduct this whole workshop since morning here. So I thank one and all the attendees. So with that, I think we are coming to this end of this session. In case we are going to administer the post session quiz, if that is still on the agenda, then we can continue with that and then finish it off.
Yes, Ullas. So we do have the post-session quiz. So I request the IT team to get started with the poll, please.
Yeah, and can I ask all the panelists to be on the camera? We'll just take a picture as well. Rutheja, Payal, Anadya. I think
Chandana, yes.
Okay, great. So let's take a picture. Thank you.
Thank you.
Thank you.
And I'm sure, even this is the beginning as well in more than one way. So I'm sure ISCR has been doing such academic workshops in the past as well. But then for me, it was the first one with ISCR and KLE University. I'm sure in future, we will continue to organize more and more sessions like this to help the academic fraternity as well.
Absolutely, Ullas. I think we do it often. We used to do it physically all this while, but now, with COVID, I think we have turned to online. I think this is a great way to actually reach out to many people. I think even if we start physical, we should have once in a while an online session where people from various parts of India who cannot travel maybe for certain reasons should be able to join. I think it's a great idea for us to continue the online session.
Yeah, sure. And the people online, the students, if they have any questions still, even after the workshop, so please reach out to us. There are email IDs, I think, or our LinkedIn IDs. Please reach out to us, drop in your questions or messages. And I'm sure everyone in the panel, all the industry leaders will be more than happy to answer your questions. Is it done? Do we keep it open for some more time or?
Yes, Ulas, there are 15 questions, so probably a few minutes more
Okay,
should really help.
Sure. And in terms of representation, I was just keeping an eye. Unfortunately, we won't be able to have the statistics of per session, how many people were online, because some were also on the YouTube, live YouTube streaming that was happening. There were some watchers there as well. But then whenever I saw, it was the lowest was 91 and the maximum I saw, I think it was 130 to
130, yeah, 130 something plus.
Around 130 to 140 was the thing. Yeah, higher side. Lowest I noticed was about 91, 92, I think. Someone has raised the hand. Yeah, maybe they want to say something.
We do have questions in the chat box. So probably I want to reiterate that you all can reach out to us wherein our email IDs are available, and we will surely give you a response, suitable response. So someone is saying, sir, can't see the question on the screen. So Kiran Patil says he's unable to view.
Can you guys see it here? Can you guys see?
No, the panel members cannot see it.
Panel members won't be, but I have some users here directly using it in the hall.
Okay.
Students, and they can see it on their mobile devices.
I think ask them to rejoin.
So the participant who is having difficulties in seeing the questionnaire, if they can rejoin, I think that may help.
Are we good to end the poll?
You guys could answer all the questions already. Okay. So I think the students here in the hall, they have completed answering. So I take it that maybe that's enough time for everybody as well.
Yes, Ulas. So will it be that just like the pre-quiz sessions, you'd be going over the questions, or will it be available for me on the screen?
Ramesh, will it be available on screen, or I will have to read it out? So the IT person is saying you should be able to see it now, Samprita?
Not yet, Ulas.
Okay, I think easy is I'll just go again. Yeah. Okay, I'll start reading. Question 1, ICH guidelines fall into which categories? Single choice question. Total responses 31. It was in terms of 60s in the morning. Now it's reduced to 30. So ICH guidelines fall into which category? Rights and well-being. 10% have answered out of 31. Quality, safety, efficacy, multidisciplinary. 32% have gone for this option. Both A and B, 58% have chosen this option. None of the above, no one has chosen it. So both A and B, option C is the 58%. That's the maximum.
Okay.
All right. Number 2, the key stakeholders of clinical trials are investigator, CRO, regulatory. 20% have gone for this. Sponsor, hospitals, IRB, IEC, biostats. 3% have gone for this. Sponsor, investigator, IRB, IEC, subjects, and regulatory. 67% of the respondents have chosen this, which is maximum. 20 out of 30. Sponsor and regulatory is only 10%. Correct answer is obviously C, and 67% of them have got it right. Question 3, during the maintenance phase of clinical trials, monitor plays a crucial role of site monitoring, which involves SDV, review of consent forms, checking drug accountability, provide training to site. 14% have chosen this. Next 14% have chosen the option site qualification, site initiation, prepare site master binders. 0% have chosen option C, which is final inventory, site master file documentation, checking of site binders. 71% of the responders have chosen D, all of the above.
Okay.
Right. Number 4, one of the key components of pharmacovigilance from MAH perspective are 72% of the respondents have chosen patient care and safety. 7% have chosen site management. 7% have chosen signal intelligence. 14% have chosen regulatory submission. Question 4 is done, right? Yeah. Yeah, obviously option C, signal intelligence is the yeah. But that hang on a sec. Hang on a sec. Signal intelligence is the correct answer. That means 7% of the total respondents got it right. Okay. The 4 elements of adverse events are 20% have chosen patient hospitalization product anomaly. 13% have chosen product dosing frequency recurrence. 20% have chosen patient product dosing an event. 47% have chosen patient reporter event and product. So which is the correct answer, and maximum 47% have chosen this correct. Next question, medical writers need to have 10% have chosen advanced understanding of drug research and pharmacology. 3% have chosen excellent language proficiency and computer skills. 0% have chosen ability to search literature. 86% of the respondents have chosen all of above, and all of above is the correct answer. Question 7. The formula for standard deviation is 79% have chosen square root of variance, and 21% have chosen sum of variance. So square root of variance is correct, and maximum of them have got it right. Probability and non-probability are the 2 sampling methods used. 79% say true, 21% false, true is the correct answer, and most of them have got it right. Next question. Dash are crypto-controlled ledgers for electronic health records and insurance information. 15% say geosynchronization, 41% say blockchain, 30% say digital phenotyping, and 15% say automated bots. B, blockchain being the correct option. Maximum respondents got it right. Next question. Dash is the person responsible for conducting clinical trials at trial site. 3% have chosen sponsor, 24% have chosen CRA, 10% have chosen monitor, 62% have chosen investigator. Investigator being the correct answer, 62% of the respondents are correct. Next question. Medical writers only write the scientific content as per the requirements. 66% say it's true, 34% say it's false, and false is the correct answer. So 34% got it right. Next one. Dash reviews and approves site initiation visit. 18% say it's investigator, 25% say it's project coordinator, 18% say it's trial manager, and 39% say it's CRA. C, trial manager being the correct answer. 18% of the respondents got it correct. The review of clinical trial data is performed by Dash team. 14% say it's biostats, 10% say it's clinical monitoring, 17% say sponsor, 59% say clinical data management, and clinical data management being the correct answer. 59% of the respondents were correct. Next one. Dash hold the complete responsibility of the clinical data being collected during clinical trials. 32% says data manager, 4% say clinical operations, 4% say sponsor, 61% say all of the above. Now C, sponsor being the correct answer. Only 4% of the respondents are correct in this one. Last one. Dash submission is completed prior to starting with clinical trials. 25% say new drug application, 0% say FDA, 75% say investigational new drug, 0% say none of the above. Correct answer being investigational new drug. 75% of the respondents got this correct. That's all.
Thank you, Ullas. So it is satisfying to see that most of the questions were answered correctly. So we all can go with a happy thought that this symposium has been really helpful for the students to really understand about our clinical research industry. So I take this opportunity to thank the dignitaries from KLE University, Ullas for being physically present there with the university team, Sachin for convening this whole event, our esteemed speakers who took time to present various topics, our IT team who helped us navigate through this whole event, and our participants who took time to be present for this session. So I thank you one and all of you take care and stay safe.
Thank you. Thanks, everyone.
Thank you, everyone.
Thank you.
Thank you. Have a safe weekend. Thanks.